Hello. Good morning to all. I welcome you all on behalf of Center of Nanotechnology, Indian Institute of Technology, Guwahati, on the second day of NEMS, MEMS and Theranostic devices. Today we will start our session with the lab demonstrations. The labs of IIT Guwahati has more than 30 major instrument facilities. For microscale patterning, we have the molecular printing system, mask writer, laser micromachining, and electron beam lithography. For material characterization and microscopy techniques, there are field emission scanning, electron microscopy, atomic force microscopy, confocal microscopy, and Raman spectroscopy. For thin film preparation, there is thermal and organic evaporator, oxidation and diffusion furnace, ODF, RF magnetron sputtering, and electron spinning device for the preparation of continuous fibers with diameters ranging from micrometers to a few nanometers. We have the RF and DC probe station for the electrical characterization of devices. So now I would like to call our first demonstrator to take over.
Good morning all. I welcome you all to this workshop. My name is Sunil Gone and I am research scholar in Morning all. I am Sunil Gone, a research scholar from Center of Nanotechnology, and I am working on mock sets for RF and I am a research scholar from Center for Nanotechnology. I am going to explain the operation of RF sputtering system. Sputtering is a technique in which high energetic ions will strike the surface of the target or material and will eject the atoms from the surface, and these ejected atoms will deposit on the substrate. And this happens in high vacuum conditions in the order of 10⁻⁶ mbar. So this is a vacuum chamber unit of the sputtering system, and this is the control unit and that is the chiller unit. So in this sputtering system, we can do DC sputtering, RS sputtering, and nanogen. DC sputtering is used for conducting target materials, and it is having a problem with dielectric targeting materials. Over the time, charge will build up on the cathode or on the target, and which affects the overall sputtering process. Whereas RS sputtering process, it alternates the electric potential of the current in the vacuum chamber at radio frequencies in the order of 13.56 megahertz, and this will actually avoid the charge buildup on the surface of the target at cathode. So since this is a high vacuum chamber, the main usage of this is to deposit thin films, and thin films are having wide range of applications. One is microelectronics. In microelectronics, for PMOS, NMOS, CMOS applications, we use thin film depositions, and in anti-reflective coatings, we actually use for mirrors, car tops, jewelry, and night vision goggles. And for solar panels also, we use thin films. And for oxidation resistance purpose, we use as cutting tools. In drug delivery system, we use thin film depositions. So the first step is to switch on the power supply. Now you see the vacuum chamber is in vacuum, so it should be always in vacuum, and it is in the order of -2, 10 power -2. Now the first step is we have to load the target or the material that we want to sputter. That will be done here, and the substrate loading will be done here. So since this is in vacuum, we have to bring down the vacuum to atmospheric pressure, that is 995 mbar. So for that, we have to insert the nitrogen gas, which is actually very pure and does not have any moisture, and by means of insertion of nitrogen gas, we will have the atmospheric pressure inside this. And by means of control unit, we will do the venting. So first, we'll turn on the nitrogen gas. So in the control panel, I am switching the vent valve. Now you can see we have done venting, and atmospheric pressure came down to 1 bar of 1000 mbar. Now you can see we have done vents. Now I can remove this. Now the chamber is in atmospheric pressure, so it is coming. You can see. So the opposite part is the grill part that we can see is the TMP, turbo molecular pump, which is actually used to bring the vacuum to 10 power -6 mbar. That will be helpful, and along with that, we use rotary pump for creating vacuum. So this is the substrate plate where we can keep the substrates. We can load the substrates. So I'm going to load substrates now. Kept the substrates on the substrate holder, and I'm making sure they're not falling, because we are going to keep the substrate holder like this inside the vacuum chamber. We have kept the substrate holder, and which is covered by the shutter. So whenever we want to start the deposition, then we have to remove that shutter, then only deposition happens. So I'm closing. We have loaded the substrates, and target is loaded here. Therefore, the first part is completed. Now it is in atmospheric pressure. Now we have to evacuate. So we have to create vacuum. For that, I have stopped vent valve, closed vent valve. Now I am going to open the vacuum valve, and so that we will start the rotary pump also, so that 10 power -2 vacuum is created, and up to the levels of 10 power -2 we have to create vacuum. Now I have started main chamber backing pump, and I have also opened the backing valve. Now within 40 minutes or so, this will evacuation will happen, and this will create vacuum up to the level of 10 power minus 2 mbar. After opening the backing valve, now the pressure reached to 6 times 10 power minus 2 mbar. So now we can start the turbo muffler pump. For that, we have to start this CPU. Now opening lay assist software. So this software is used to control the operation of turbo muffler pump. So this is the interface which we have. If we click, we can control we can set the frequency of the turbo muffler pump and other parameters, the current actual, everything we can set here, but this is fine. So we have to on remote control and we have to start. So now you can see slowly here the frequency will change. Now the frequency has changed and it will reach to the set frequency, and after that we can say that turbo muffler pump has started fully. And now if we can see the monitor here, the pressure value will change from minus 2 to minus 6. So it goes up to 6 times 10 power minus 6, and that is the pressure where we can do the deposit. So we have attained 8 times 10 power minus 6 mbar pressure inside the chamber. Now it's time to insert the argon gas flow. Once argon gas inflow is turned on, we are turning on the throttle value controller and also this MFC controller. Now this is pressure, this is full actually, and these are the channels for oxygen, argon, nitrogen, and helium depending upon the requirements. Now I have inserted argon flow. Therefore, by means of this, we can regulate the channel flow, argon flow inside the chamber. This is in SCM, that is standard cubic centimeter. So now this set point, this actually is at now minus 6. Now we are changing this. Before this, this throttle has to be kept on, since this is see, all these are in off, this is in on now. So we have on this. Now the gas flow is there inside the chamber. So by means of this, the argon gas is entering the vacuum chamber. Now you can see the pressure is decreasing down a little bit. So now this is at full pressure. So we are decreasing this. We are decreasing to 2 into 10 power minus 2 mbar. So here also the chamber pressure will come to that point. So we are increasing the argon gas flow now slowly. So in SCM we are increasing. As we are increasing the argon gas flow, you can observe the pressure is coming down. Now the set point pressure is equal to the chamber pressure. So it is blinking. It is coming to green from red. So that is this is the indication from where we can start the power supply. So now this is the RF power supply. This is the RF power supply. So we are turning this on. I have turned this on. So this is RF power supply, and this is on off. So I am turning this also to on. So red indicates on. Now this is on. Now I am slowly increasing the power in steps of. See, the power is increasing. I have increased it to 6 watts, 5 watts. Now for few seconds we will wait. Then we'll increase little bit again to another 5 watts to 10 watts. Now in total 10 watts we have increased power, and we can wait for a while. Now you can see here, you can see earlier there is no plasma. Now plasma appeared. Plasma appears because argon gas will be converted to argon ions and electrons because of the power that we are applying to the RS sputtering system, and those argon ions will hit the target surface and atoms will be ejected from the target surface, and they'll travel through vacuum and will be deposited in the substrate which is connected to the substrate holder. And in order to start the deposition, we have to move the shutter. Unless we move the shutter, you can see here there will be a shutter inside the chamber, which actually covers the substrate holder. And so if at all, if we remove the substrate holder, then only the deposition starts. We'll change the power. Here it is now 10 watts. Now we are changing this to 20 watts. So if at all, if you want to deposit the material at 20 watts, then 20 watts is set. And now see the plasma radiation will increase a little bit compared to earlier. And if we still increase, if you want to see, you can see the plasma radiation. There it is keep on increasing as we increase the power supply. Now in order to start deposition, we have to wait 1 or 2 minutes because that sputtering will not be uniform. Therefore, if we wait 1 or 2 minutes, it will be uniform. Then we can remove the shutter. From then onwards, the deposition starts. This is how the deposition actually works. So these are undeposited films. This is quartz substrate. This is P-type silicon substrate, and this is the sputtered deposited P-type silicon substrate. This is the quartz substrate, and this is the mask that we had put on the substrate while keeping on the substrate holder. You can see in the middle. So this is in the same color of this. So this is P-type substrate, and this is the thin film that has been deposited over the P-type silicon substrate. Similarly, with quartz, you can see this is the mass that we have kept while keeping on the substrate holder, and the thickness of these films will actually depend upon the wattage, the power that we are supplying, RF power, is 10 watts, 20 watts, 50 watts, 100 watts, and also on pressure that we are actually applying in the chamber while depositing the films. The thickness depends upon deposition time, pressure, and also on the power supply that we are providing.
Good morning, everyone. I am Rajan Singh. I'm a research scholar in EEE department. I am here to demonstrate mass writer and optical lithography process.
Hello everyone, I am Rajan Singh. I am a PhD student in EEE department of IIT Guwahati. I am working under Professor Roy P. Pelly. Today we are going to demonstrate photolithography process. So this is a clean silicon wafer, silicon dioxide wafer, approximately 500 micron thick and 20 nanometer of silicon dioxide deposited over, grown over it. So this is pre-cleaned via RCA cleaning, and now it is over the hot plate for removing any moisture. So you can see approximately 135 degrees Celsius we have kept around 10 minutes. So whatever water vapors are present, water humidity present, it will remove. Then we will go to this instrument, which is a spin coater. So you can see this is a chuck in which this wafer is going to sit. Then these are the vacuum pumps. So the pipe is connected in the back and the suction will come on the top. So vacuum will suck the wafer on its plates and this chuck will rotate. The rotation speed and the time we can vary over this panel over here. So we will place the wafer over it. We will put the photoresist and then spin coat it. After that, we will dry it again with this hot plate, and then we will go to mask writer, which is this machine for direct exposure, or this UV box in which we will do the exposure with the help of a mask. So we will show all these steps one by one. So first, so the preheat is over. So 10 minutes preheating of this wafer is over. Now we will keep this wafer to cool it down little bit, and then we will follow by HMDS coating, and then we will proceed for photoresist coating. So now we are going to keep this wafer on this chuck, and we have to make sure that it sits properly over the chuck so that no disbalance will happen during the rotation. Now I will on the vacuum, so the vacuum will hold this wafer over the chuck. Now we are going to put the HMDS over the wafer. Only slight is enough. And then we will load the program. And close this chamber so that nothing will come out. So you can see we are going to rotate about 4000 RPM for the acceleration of 5, and then time is 60. So now the HMDS coating is over. We will take the wafer out. We will switch off the vacuum first, and then we will slowly take it out the wafer and keep it over this hot plate for heating up. So it will be 135 degrees Celsius for about 5 minutes. So about 2 minutes left. So we are going to deposit photoresist after this. So there are two types of photoresist. As 1813, which is positive photoresist, and SU8, which is a negative photoresist. So we are going to demonstrate with S1813. And we are lifting this wafer from the hot plate and keeping it to cool down till the room temperature. After that, we are going to take the S1813 bottom from the fridge, and then we will repeat the spin coating process for positive photoresist. So now the photoresist spin coating is over. I'm switching off the vacuum and take it out this wafer and keep it for heating for 3 minutes at 120 degrees Celsius. So now the heating part is over. Photoresist is dried now, and you can see it is kept to cool down at a room temperature. You can see a slight reddish color over the surface, which is obviously because of photoresist. Now we are going to use lithography machines. There are 3 machines, 1, 2 and 3. So we can do the lithography with this machine, which is called a mask writer. As you can see, inside this machine, a laser source is there and then optical system is there. So the laser will fall from this head to this chuck. Here we can keep our mask or even the coated wafer, spin coated wafers for diet writing. So the laser spot size is about 2 micron and it will travel 10, 20, sometimes 1 mm per second speed. So the desired energy as well as the speed is selected as per the features which you want, and then the lithography can be performed. But this is a very slow process as the laser has to travel all the feature one by one, and therefore a lot of time is consumed. So in order to save the time, what we can do, we can use the mask, write the mask one time, and then use this UV box for the exposure. So our photoresist is sensitive to this UV radiation. So the UV radiation will fall and the patterns will appear over the silicon wafer. So this is the wafer. So this is the mask which I was talking about. This is a soda lime glass over which chromium coating is there, followed by the photoresist coating. So we can write the designs directly with this machine, develop it, etch the chromium, and you can see the final designs will appear like.
So there are many type of designs. So like there are some saw designs, some micro channel designs, and some designs which you will not be able to see are basically FET and chem register type of devices.
So you can see there are some markings. So we have to keep our wafer so that it will fit in between these markings. I am going to put the mask very carefully. The loading is complete. It is asking what is the thickness of the wafer. So it is 500 micron. Now masking distance. So for better development and fine features, we go for hard contact, or that's why the distance is 0. Now it is adjusting the distance between the mask and the wafer. So now loading is done, distance adjustment is done. So we move towards the next step. So I'm going to do a continuous exposure. So duration will be 4 seconds and power is 25%. So these values are already optimized. Now the exposure is being performed. Now the exposure is finished. I will take it out one by one. Very slowly. So I'm going to develop the patterns. So in this petri dish, the developer solution is kept, and in this one, the DI water is kept. The developer solution will expose over the surface for about 10 seconds, 10 to 20 seconds. So now I'm going to dip this wafer very slowly and start the timer and very gently shaking this. So I have developed it and then wash it with DI water, then again dry it using nitrogen. Now you can see the patterns appear over the surface. Roughly you can see there are some shiny things appeared. Examine these patterns, the distance and everything using the optical microscope.
If the pattern and their distances are up to the mark, then we can move towards the next step, which is metallization and liftoff.
Good morning. I am Vimal Kumar Singh Yadav. In this lab session, we will discuss about molecular printing system. Good morning. I am Vimal Kumar Singh Yadav. In this lab session, we will discuss about molecular printing system (MPS). In this figure, we show you the MPS laboratory setup. MPS is used for direct deposition of materials on targeted locations over a substrate. In this printing system, we can print nucleic acids, proteins, lipids, DNA, and also non-biological materials like metallic inks, polymers, graphene, etc. on diverse nature of substrates like silicon, glass, PVA, PMMA, and also flexible substrates like PET. The heart of this printing system is called surface patterning tool, a schematic of which is shown here. It consists of a supporting substrate, and the print head is microcantilever, which is made of thermally grown SiO2. It consists of microchannels, and the material to be printed is drop casted near reservoir in the form of ink. The ink particles flow through this microchannel of the cantilever and arrive at the opening gap or microcantilever tip. And when this tip touches the substrate, the ink is deployed and the printing is performed. Here we show you SPT30R. 30 stands for the width of the micro cantilever, and R symbolizes the round shape of the opening gap. This SPT30R consists of two inbuilt microchannels, each of it 5 micron, and the opening gap of this SPT is near 18 micron. Similarly, we show you here SPT60R, whose total width is 60 micron, and it also consists of two microchannels, each of width 10 micron, and the opening gap is near 30 micron. So using different kinds of SPT, we can print different diameters of micro spots. The XY stage range of MPS is near 50 mm cross 50 mm, and the J stage range is 45 mm. MPS also consists of motorized optical microscope having a maximum zoom capacity of 1000x with integrated video capture facility. Moreover, MPS consists of an inbuilt humidifier through which we can control the humidity of the printing chamber in the range of 25 to 80% RH. So before going to the actual demonstration of the equipment, let us briefly go through the printing process flowchart. So first of all, we clean the substrate and the SPT with acid piranha and DI water and dry them using dry nitrogen. To make the surface of SPT and the substrate hydrophilic to the ink, we treat them in ultraviolet ozone environment, and after that we put them in the print chamber. Based on a requirement, we set the humidity inside the print chamber using built-in humidifier. And through MPS software interface, we focus the SPT microcantilever and the substrate together. And once the desired ink flow is obtained and the SPT microcantilever touches the substrate, the ink is deployed and the desired features are printed. In the end, we clean the SPT properly for further experiments.
Okay, so let me introduce to the actual system which we call molecular printing system, which you can see here. We also call it as a nano enabler. So let me introduce this. So starting from the bottom part here, you can see that this is called substrate holder. So we place our targeted substrate on which the printing is intended to be done here after proper cleaning. The second part is called SPT holder, and this is the part where we stick our SPT, and after that we dropcast the ink over the SPT. So now once we have properly cleaned the SPT, using a proper tweezer, we will place the SPT on SPT holder. We tap the SPT gently so that it has to ensure that it has properly stick to the SPT holder. Now we will dropcast ink here for AGNP ink we will dropcast, for example, on the SPT reservoir with the help of a micro pipette. So we saw that when we dropcasted ink over the SPT without treating the SPT in the UV ozone, we saw that the ink was not spreading well on the surface of the SPT. So to make the ink properly spread on the surface of the SPT, basically the surface of the SPT is silicon, so we need to do UV ozone treatment to make the surface hydrophilic to the ink. So this is the UV ozone pro clear, which is used to treat the surface of SPT as well as the substrate. So here we can set the time, as you can see, from 10 to 60 minutes depending on the substrate and the ink, which kind of interaction we want, and then we on the system. So now we can see that the red light and the UV red light had turned off. So now it means that UV treatment process has been completed. So now we will, after the UV treatment, we will check the wetting behavior of the ink. In other words, you can say we can check the hydrophilic or hydrophobic nature of the SPT surface after the UV treatment. And now you can see here that we have been able to attain a proper spreading of the AGNP ink over the SPT surface. So now we will insert the ink loaded SPT reservoir inside the print chamber. The MPS system consists of a microscope whose objective directions can be controlled using these two knobs. Since the working principle of MPS is very much similar to AFM, we also have a laser source and correspondingly a detector for precise printing of smaller and finer particles. Moreover, to control the humidity inside the print chamber, we have inbuilt humidifier, which is placed here. So once we have properly placed the substrate and the ink loaded SPT inside the print chamber, we close the shutter. So in this section, we will show you the MPS user interface. We call it a nanoware. In the left hand side of this interface, we have controls for the X, Y and J movement of the SPT. Also we have the controls for objective. In the right hand side of this interface, you can see the cantilever of the SPT, which is right now in the focus, and the unfocused area is the substrate. In the bottom part of this interface, we have humidity control section, and according to the target humidity, the printing chamber tries to attain that value with the help of inbuilt humidifier. So now once we have loaded the ink over the SPT and placed the SPT on the SPT holder and then SPT holder in the chamber, so using this nanoware interface, user interface, we can now clearly see the presence of the ink. So now the ink particles, in short, you can say that ink has flowed down to the through the micro channels of the cantilever till the tip of the macro cantilever, and this is ready to be printed.
This video shows you the printing of AGNP micro pads on glass. By overwriting one spot or other printed spot, we can print continuous lines, and by printing partially printing a line over a previously printed line, we can print continuous micro pads. This video shows you the printing of microspots of buffering on glass. And similarly, by overwriting one spot over another spot, we can create continuous microlines as seen here. So after printing experiments, here we show you some of the printed results. In this figure, we show you AGNP printed microspots and microlines over cover slip. In this figure, these are the AGNP printed microlines on ITO. Here you can see the microspots of polyaniline on PET and PBA. This figure shows you the micropads of AGNP on glass. So if you are further interested to know about this kind of printing technology, you can refer to our publications. And with this, we end our presentation. If you have any query, you can ask us. Thank you for your attention.
Welcome everyone. So my name is Manish Kumru and I am going to demonstrate FESL, that is Field Emission Scanning Electron Microscope, and I am going to demonstrate how it works and how we can characterize our sample through the FESL. The Field Emission Scanning Electron Microscope is named because the electrons are generated from a tungsten tip which stays in the vicinity of an electric field. After a certain voltage, the tip liberates electrons which travels through the electromagnetic coil to be focused on the substrate. After falling on the sample as an incident beam, the diameter of the beam may be as small as 1 nanometer. Once the incident beam hits the sample, mainly three types of electrons are liberated from the sample itself. First one is the secondary electron, then backscatter electron, then the X-ray electron. Secondary electron, which provides fine structure topographical feature of sample's face, and the backscatter electron to discriminate areas of different atomic number element. X-ray electrons are used to identify the composition and measure the amount of the elements in the samples. All kinds of sample topography can be characterized except liquid and magnetic samples. Sometimes biological samples are also challenging. So the magnification that can go from 25x to 10 lakh x, accelerating voltage that can be maintained within 0.5 kilovolt to 30 kilovolt. The probe current also is maintained in between a few picoampere to 200 nanoampere. The image resolution is as high as 1.3 nanometer at 1 kilovolt or 1 nanometer at 15 kilovolt during the analysis. There is a gentle beam inbuilt characterization technique which allows us to characterize soft materials without destroying much of the sample, and that incident beam does not penetrate in that much, so that the sample will not burn. So there are certain main components are here, like this column where the electrons are generated, then the specimen chamber where we can keep our sample inside here, then the vacuum pump, then the monitor and the controller. So these are the main components over here. So today our sample is a small piece of butterfly wings. So this sample we are going to look at through the FESEM. So I have the sample holder, and the carbon tape is pasted on it, and I'll put that sample on the carbon tape. So now comes the next part. As this sample, it is non-conductive, so we have to make it a little bit conductive, so using the sputter coater. So we will coat the platinum on this non-metallic surface. So this one is our sputter coater. So you see, this is the chamber where we will put our sample. So once we close the lid, so it will get vacuum. We will switch on this instrument. So there are certain parameters, those are current and pressure and the time also. So according to our requirement, the thickness of the deposited platinum, we have to set the time as well as the current. So generally we use here 20 milliampere, and the time, that in second, we generally use around 90 to 120 seconds. After reaching a certain pressure, that is around 2.9, then we will start the deposition of the platinum on the sample. So that pressure has come to the 2.9, so we can start the deposition of platinum on the sample now. Okay. So now I am going to put our sample, that is the butterfly wings, inside the specimen chamber. So before that we have to vent the instrument, because the whole system that has to be in the vacuum inside. So once this specimen chamber, it comes to the ambient pressure, then we can open this lid. Once we keep our sample, then we again have to vacuum the whole chamber along with the specimen chamber. So once the specimen chamber's pressure, it comes to the near to the inside chamber, then only we can insert our sample from this specimen chamber to the inside. So while the inside pressure is reducing, when the pump is working, in the meantime we can set our voltage. We may start with the 5 kilovolt. That I am doing gradually decreasing. It was initially 15, then 10, then I set it at 5. And the other parameter emission current that is already automated with according to the acceleration voltage, and the probe current is also maintained at 6. So this schematic, it is showing the whole instrument. I mean the FESM, that column you can see here, and this is the specimen chamber. So this is now the color, it is gray color, and the whole system it is gray color. That means it is showing the inside pressure, it is all over same, and we can now insert our sample inside the FESM. So as now the pressure has come to our requirement, so we can insert our sample from the specimen chamber to inside the FESM. Once the sample is inserted, so we can see this dialog box where it is asking the specimen holder that we are using here. So this one we have used, so we can select it, and the offset we have to give, and the offset, that means the thickness of the sample, and in this case we are using the butterfly wings. It has much less thickness, so we don't require to give any thickness parameter. But unless if we are using glass slide or other any thick material, then in that case we have to give the thickness of the sample. So now I am going to take the stage little bit upward. So this one is the objective that we call, and this is the specimen that we have, and that specimen holder also. So we need to make it little bit closer to the objective. So in that case we have to reduce the working distance. So you may see that substrate holder, it is getting closer to the objective. It is not required all the time that we need to keep the substrate holder too close to the objective. So in this instrument, we maintain around the pressure that is less than 10 to the power minus 4, and once the pressure inside the chamber, it goes beyond 10 to the power minus 4, then only we can start the electron gun. So it has gone already, so we can start here. So this is a very fine instrument. So what you are looking at right now, it is the butterfly wings, and it has some pattern structure, that is some scales which are patterned. So with the controller, I can rotate the sample holder. I can make it move XY direction. I can make it tilted also in different angle. I can get zoomer and zoomer to get fine view of the smallest structure on the sample. So now we are basically detecting the secondary electron that is coming from the just top surface itself. Sometimes it is called as the primary electron. So you may see these are the scales which are patterned, and we may go mode zoomer. So with this FESEM, we can go up to 10 nanometer scale. So right now we are at 10 micrometer. So that is at 750x. But it is possible to characterize in around 10 nanometer scale. So I am doing a little bit zoomer on a particular scale. So you may see some fine microstructure on the scale that is very fascinating. So there is a whole new world in microscopic scale that is beyond our sight.
Good morning, everyone. I am Janani, research scholar from Biotech Department. I am here to demonstrate confocal laser scanning microscopy. Hi, everyone. Today, myself, Janani, and my fellow research scholar Tamana are presenting the principles of confocal laser scanning microscopy, followed by its demonstration. Confocal microscopy was pioneered by Marvin Minsky in the year of 1955. Confocal microscopy, an optical imaging technique, is used for increasing optical resolution and contrast of an image by means of using a spatial pinhole. Thus, a sharp image of a specimen is created that would appear otherwise blurred with the conventional fluorescence microscopy. This is achieved by enabling optical sectioning of the sample and eliminating out-of-focus light from the specimen, not from the focal plane. Pinhole helps in eliminating the out-of-focus light. Through this, a better image with appropriate contrast and less hazy is obtained. Interestingly, three-dimensional samples can be analyzed at certain depths, and also a series of thin slices of the specimen can be captured and assembled to generate a 3D image. A diagrammatic representation showing the light pathways and position of lasers, pinholes, specimen, and photomultiplier tube in confocal microscopy is shown here. The green line depicts the excitation light rays from the high laser power source, which passes through the first pinhole aperture, excitation filter, dichromatic mirror, and objective lens to reach the sample. By adjusting the pinhole in front of laser illumination, one can control the depth of field. Followed by the emitted fluorescent rays from the sample, which is shown in red line, passes through the objective lens, dichromatic mirror, and also detector pinhole aperture before reaching the photomultiplier tube. The second pinhole placed before reaching the PMT helps in elimination of background information away from the focal plane. Pinhole size is mainly used to control the optical section thickness. Here we can see that by increasing the size of pinhole, out of focus light increases and there is a compromise in achieving the better confocal image. So the pinhole size should be kept always small and the range of choices 1 to 3 AU. Through confocal laser scanning microscopy, the prime advantages are improved resolution image with better contrast, high signal to noise ratio, and controlling the depth of field and Z-axis scanning. It provides multiple benefits and applications. Main benefits are thick specimens can be clearly examined and reduced blurring of the image can be obtained from light scattering. Applications of confocal laser scanning microscopy are cellular processes like phagocytosis, internalization of nanoparticles, apoptosis, membrane potential, cell-to-cell communication, probe rationing, etc. can be studied through the confocal laser scanning microscopy. In our lab, we have Carl Zeiss confocal laser scanning microscopy model LSM880. We have 5 types of excitation lasers: 355 nanometer, 405 nanometer, 488 nanometer, 514 nanometer, and 633 nanometer. PMT2, GASP1, TPMT1 are the 3 types of detectors along with the microscope. 6 objective lenses 5x, 10x, 20x, 40x, 63x and 100x objectives are present. Mainly, halogen lamp is used for light transmission. The advantages of our confocal model are fine detailing of the specimen can be observed using both confocal and Eriscan super resolution mode. Both live cells and fixed cells can be imaged. LSM880 is also compatible for fluorescence correlation spectroscopy. Now, we are going to demonstrate how to operate confocal laser scanning microscopy, sample imaging, and image processing using the software interface.
Hello everyone, I am Tamanna Bhunya, Research Scholar from Center for Nanotechnology, and today I am going to demonstrate confocal laser scanning microscope. So to begin with, we have the main switches which we need to turn on. So this is the main switch and simultaneously we have to switch on all the switches, third and fourth switch to turn on the components connected with the microscope, and then we have the laser where we need to switch on the lasers. In total we have 7 excitation lasers to excite the sample, and this is the switch we need to turn on, and then simultaneously to adjust the laser power we can adjust this knob, and then we can set the laser power according to our requirement. Moving on, this is the system of the confocal microscope which consists of the detector, and this system also contains the pinhole, and then it is connected to the microscope where we can focus the sample after placing the sample by adjusting the light present below this knob. Using this knob we can adjust the light and we can focus the sample, and then it is attached to the laser which consists of 7 excitation lasers, and it is connected to the joystick. With the help of joystick we can move the stage where we place the sample, and further it is also connected to the objective setting of the microscope. The system helps to set the magnification of the objective lens.
I am Janani Ji, Research Scholar from Department of Biosciences and Bioengineering. I am going to explain how to place the sample and how to do the imaging part in confocal laser scanning microscopy. So we have already seen the basic components of the confocal laser scanning microscopy. So for placing the sample, we basically have 2 types of stages. One is for live sample imaging, where we can place our petri dish in this part, and we can set the knobs accordingly, and we can use this for imaging the migration assays, and we can do the live cell imaging. We can do uptake assays and all. And we have another stage for fixed samples, where the samples are already fixed in the cover slip and already fixed in the glass slide, and we can place one cover slip above it. This will be like very The thickness of the samples will be about like 10 to 20 micron thickness. Since it is inverted microscopy, we have to place the slide in the upward position, and this sample stage along with the glass tile, it will go to the sample holder. The sample stage goes to the sample holder. And after placing the sample stage properly, we have to close the chamber. And through this knob, we can focus the sample through the eyepiece. After placing the sample, we have to select the objective lens for imaging. 5x, 10x and 20x can be used for capturing lower magnification images, whereas 40x, 63x and 100x can be used for capturing high magnification images. Water immersion should be used for 40x and oil immersion should be used for 63x and 100x. We can see the different objective lenses used in our confocal laser scanning microscopy. For illustration, we are showing you here how to place an oil on the objective lens. A single oil drop is placed very carefully above 63x objective lens. Once after focusing the sample through the eyepiece, image and processing is done through the Zeiss software interface. To start with, the laser source is selected based on the fluorophore used for tagging the target in the sample. Then, intensity of respective laser and pinhole distance is set here. For each laser, it is important to adjust the master gain, digital offset and digital gain to achieve balanced intensity and also to remove background noise. The emission wavelength should be adjusted for each excitation laser selected previously. If we need to capture bright field image in the same focused area, PMT should be selected to allow the light source. For example, xylem of a plant is stained here for the cell wall using secondary antibody Alexaflor 488 and Alexaflor 561. Through split selection, individual images for each channel, bright field image and merged image can be exported. A 3D spectrum image showing the target distribution along X, Y and Z axis can be captured. Histogram indicating the distribution for each laser can be plotted using the software. Another example is shown here where the chick embryo fibroblast is stained and tagged with all three RGB markers. Nucleus is stained with DAPI and tagged with blue. Cytoskeleton markers is tagged with green dye and extracellular organelles are tagged with red dye. The split channels for each tagged dye and merged image is seen here for clear representation. Similarly, 3D spectral plot showing the distribution of extracellular vesicles in all the axes is shown for clear understanding. Histogram plot can also be plotted. Z-stack is used for recording images at different focal planes by incrementally stepping through the sample. First focal plane, last focal plane and steps distance can be set here. Few recent achievements using our confocal microscopy. A 2D bacterial biofilm is imaged here where it is stained with PI for red channel and AO for green channel. This research work is published in ACS Applied Materials and Interfaces in 2020. And another example is shown here. This is Z-stack imaging of a 3D silk scaffold cultured with kidney cells, where infiltration of cells into the silk scaffold is studied. Live cells are stained with calcium AM, showing green fluorescence, and silk is stained with ethidium homodimer, showing the red fluorescence. The 3D surface plot represents the distribution of live cells in X, Y, and Z axis. From the 3D projection video, we can see the individual stack image, which is later merged to obtain a Z-stack image. This research work is published in ACS Biomaterial Science and Engineering in the year 2019. Myself and Tamanna would like to thank Siddharth for his support in video recording. Thank you all, and it is now open for any queries from the audience.
Good morning, everybody. I'm Srimantapal. I'm a research scholar in the Department of Chemistry. Now I'm going to demonstrate Raman spectroscopy.
Welcome to the world of Raman spectroscopy. Raman spectroscopy is totally based on Raman effect. And this Raman effect was first discovered by our Indian physicist Sir C.V. Raman in 1928. So this is very special spectroscopy. Why you will learn this Raman spectroscopy technique? There must be some reason. There is lots of applications of Raman spectroscopy, like you can determine the nature of chemical bonds, symmetry of molecules. You can identify any molecules, any kind of crystallographic orientation of sample to observe low frequency excitation of the solid. You can distinguish between healthy and unhealthy tissues, medicine, therapeutic drugs. You can also use this Raman spectroscopy technique to detect explosives, lots of applications. That's why this Raman spectroscopy is very much important in today's life. Now, what is the basic principle of this spectroscopy technique? Suppose this is substrate. If you irradiate this substrate with light, then there will be lots of phenomena like reflection, transmission, absorption, and one of those phenomena is scattering. One photon out of 10 to the power 6 photons is scattered in this way, and Raman spectroscopy totally depends on this scattered photon. Now incident light comes and interact with the molecule of the substrate. So now this interaction can be explained through this picture. Suppose the molecule initially is in the ground state. So now if incident radiation excites that molecule from here to this virtual state, and if it comes back to its initial state, then it is called Rayleigh scattering. And if it comes to the first excited state, then it will be called as Raman scattering. But if the molecule is already at the first excited state, and if by absorbing light it goes to the virtual state, if it comes to the ground state, then it will be called as anti-Stokes Raman. To measure Raman spectrum of any sample, you need proper sample preparation. And for this Raman spectroscopy, this sample preparation technique is very easy. Sample preparation, and for that you need one glass substrate or silicon wafer, anything like this. So just put your sample mixture on this glass slide and dry it. After that, that will be ready for Raman measurement. Suppose this is our sample. This is Raman standard sample, and we have one silicon wafer. So we will measure Raman spectrum of this sample. To record Raman spectrum, you need one software. This is the software. We will use this software to record Raman spectrum. So now this software is ready. So to record Raman spectrum of this sample, we have sample platform. This is the platform where we will put this sample. To record Raman spectrum, we need one excitation source. Means that energy source will excite the molecule of this sample, and these molecules will go to the virtual state. For that process, we need one energy source. These two are energy source. This is 532 nanometer laser, and this is 633 nanometer laser. So these two laser source will be used as excitation source. So now we have different apertures. This is 5x, then this one is 10x, this one is 100x, and this one is 20x. Now how we will use these apertures, we will learn this thing. So first we will fix 5x aperture on the sample. Then we will go to the video portion of our software. Now if we go to this option, now we are going to start to acquire video. We are not watching anything.
So now we have one lamp. We will switch on this lamp. The light of this lamp will fall on the sample through this aperture. Now you can see light is falling on the sample and the brightness is increasing, and now brightness is decreasing. Now lamp is off. The use of this lamp is to see the sample location. Now the video is actually functioning, but we are not watching anything. The reason is that the sample is not focused. So for that purpose, we have to focus on the sample. So now you can see here the light is not falling on the sample. So to move this stage, we'll use this joystick. You can use in this way, just you have to move this joystick and your sample location will move. You can see the sample location is moving. I'm using joystick to fix the light on the sample. Now you can see that light is falling on the sample. You can see that video is totally white because the sample is not focused and light is too high. Silicon has reflecting characteristics. So for that purpose, we will reduce the light, and you can see the intensity of whiteness is decreasing. So now we will use this one and this one. We will use these two knobs to focus the sample. We have to use in clockwise or anti-clockwise direction, means these two will act along the z-axis, and that sample stage with the help of joystick, that thing will act on xy plane. To focus the sample, we will use these knobs. This is for slow movement, means from A to B. If the distance is too high, then we will use this knob, and if from point A to B, if the distance is very less, in that case we will use this knob. Now I am focusing the sample. You can see now clear video is coming. You can also see some particles on the sample because this might be some dust particle. Now this is fully focused. Now we are ready to record Raman spectrum. Before starting recording, we have to readjust some parameters. First, we have to stop that video acquiring. Video is stopped. That means your sample, aperture, everything is fixed. Now we will switch off this lamp. Here you have two options, 532 nanometer and 633 nanometer. These are lasers. You will use these lasers according to your sample. We have silicon wafer, so 532 nanometer laser will be much more appropriate for this sample. So that's why we will use 533 nanometer source. Now this is filtered. If your sample is easy to burn, in that case you will decrease this percentage like 10%. If you increase this percentage, in that case intensity will be much more higher. So that's why every time we have to increase this percentage, but we have to keep in mind that sample might get burned if we use higher percentage of laser. Here this is the objective button. From 5x to 100x we have these objectives. Now the sample is under 5x objective. Now this is the range, means some range where you can expect peak position of your sample. And then And this acquisition time means the time to acquire Raman spectrum for each window. If you increase this acquisition time, the intensity of peaks will increase. And if you decrease this time, then the intensity will decrease. And accumulation means this is the average of acquisition of spectrum. And there is another important parameter, that is spectro. Every time you have to put the middle value of your range. Now all the important parameters are ready. We are ready to record Raman spectrum of silicon sample. So now we are clicking this start button. Now we are clicking this spectra button.
Finally, we have recorded Raman spectrum. In this way, we can use Raman spectroscopy. Hope this demonstration will be useful. Myself, Srimantopal, PhD student in the group of Professor Urun Chattopadhyay at IIT Guwahati. Well, thank you all.
Hi everyone, my name is Amir Suhail. I am a PhD scholar in the Center for Nanotechnology. Today I am going to demonstrate the laser micromachining workstation. Hi everyone, my name is Amir Suhail. Today I am going to demonstrate the ultra-fast laser micromachining equipment which we use for our microfabrication applications. So before going to operate the equipment, let me explain a bit about the process as well as the equipment. So this is a general purpose ultra-fast laser micromachining workstation. Micromachining is the process of making small things in micrometers 10 raised to minus 6 meters by material removal. We are using an ultra-fast laser as the tool. So ultra-fast or ultra-short time scale, that is the time scale in femtoseconds to picoseconds. Laser beam waves diameter, that is ranging from 5 to 10 micrometer. So we can make structures as small as 5 to 10 micrometer. So the minimum spot size with the laser, that is also 5 micrometer. The pulse repetition rate of the laser source. So that is like how many pulses are coming out of the laser per second. And the another parameter is the average power, that is the energy per unit time. So how much energy is coming out of the laser per second. The third parameter is the scan speed, that is the speed at which the scanner is moving in the working area to machine the sample. So that is the third parameter. So now let's move on to the parts of the system. There are 3 parts for the laser system. The first part is a femtosecond fiber laser. The second part is a second harmonic generator. The third part is a galvanometer scanner connected with an f-theta lens. Let us see this first part, that is a femtosecond fiber laser. So in this femtosecond fiber laser, the first part is a fiber oscillator. So in this fiber oscillator, light pulses of 1 nanojoule energy at a wavelength of 1030 nanometer is being produced at a repetition rate of 25 megahertz. So this energy is very small. So for our micro machining application, this energy is insufficient. So we need to use amplifiers to increase the energy of the light pulse. So we will be using multiple number of amplifiers, and also we won't be needing the 25 megahertz repetition rate for our micro machining applications. We will be needing from 200 kilohertz to 1 megahertz. So for reducing the repetition rate from 25 megahertz to this desirable repetition rate, we will be using a pulse picker. So after that, we will use a main amplifier. There is a preamplifier here. Then we will use a main amplifier. After that, we will be achieving a pulse energy of 10 microjoule. So the initial pulse energy was 1 nanojoule. So we have amplified that to reach a value of 10 microjoule. So at the output, we will be having 10 microjoule, in which the amplification throughout the system, that will be 10,000 times. Now let us see the second part of the system, that is a second harmonic generator. So this actually is used to double the frequency of the femtosecond fiber laser output. So here the wavelength was 1030 nanometer. So by using the second harmonic generator, we can convert this wavelength to 515 nanometer. So this process is actually a highly inefficient process where we will be combining two photons with 1.2 electron volt energy, and we will be achieving a single photon with double the value of energy. So this process is a non-linear process and it is highly inefficient. So because of that, we will be achieving only 30% of efficiency. So you can see that in the SHG input, when we will be applying 10 microjoule of pulse energy, we will be getting only 3 microjoule of energy at the outlet of this second harmonic generator. So there is a question, why we are using this second harmonic generator? So there are three advantages. The first advantage is that when we will be machining some high band gap materials such as glass with some, for example, 10 electron volt of band gap. So when we will be using photons with double the value of energy, we can get a lower order non-linear absorption. So the order of non-linear absorption will be reduced by half. So that is the first advantage. So it will be easier to machine materials such as glass or some other dielectric materials. So the second advantage is that we will be getting a smaller beam waist diameter of 5 micrometer. That is what we will be getting, and we will be getting a higher peak intensity. So these three advantages are compared with the direct output of this femtosecond fiber laser. So for getting these advantages, we are using the second harmonic generator. The third part is a galvanometer scanner which is connected to an f-theta lens. So this galvanometer scanner is used for beam scanning in the XY plane while we will be machining. So the focal length of this f-theta lens is 130. And the working area with this galvanometer scanner f-theta lens setup we will be getting is 40 by 40 mm. So this is the maximum area of the sample which we can machine. So another part is the beam waist diameter that is possible with this galvanometer scanner, and this focal length is 5 to 10 micrometer as told above. So most importantly, the scan speed with this galvanometer scanner can be varied from 1 meter per second to 1 mm per second to 1 meter per second. So this maximum speed of this galvanometer scanner is so high. So our productivity, our process capability will be very good. So there are some other accessories also we will be using while machining. So first is a power meter. So we will measure the average power. For calculating the pulse energy, we will measure the average power with this power meter, and we will be using a spectrum analyzer for viewing the oscillator spectra, for viewing the spectra of this oscillator, and this oscillator is generating these small pulses, ultra short pulses, by a technique called mode locking. So we need to have a spectrum analyzer for verifying the mode locking of the oscillator. So another part is an oscilloscope. We need to view for viewing the pulse train. So we will be using the oscilloscope. By that we can view the repetition rate. How much is the repetition rate? The fourth part is the Z stage, which we will be using along with the galvanometer scanner, FTL and setup for adjusting the focus of the sample in the Z axis. So now let's see the parts of the system. This is the laser. This is like there is oscillator inside the laser, and there are amplifiers, and which the outlet of the laser will be through this side. And this is the controller. This is the controller which will be giving signals for functioning of the laser. This is the second harmonic generator, where we will be doubling the frequency of the output of this particular laser. So with this second harmonic generator, we will be getting 515 nanometer beam pulse laser source with maximum energy of 3 microjoule. So let's go into the front side and see the parts. So the beam from the second harmonic generator will come through this mirror 1, 2, 3, 4, 5, and into the galvanometer scanner. So there is like we have connected the power meter here for measuring the power, and this is a Z stage which is used to move the sample and to keep the sample and to move the sample in the Z axis. So on the bottom of this galvanometer scanner, we have connected an f-theta lens to get a larger area of working area of 40 by 40 mm. So essentially inside this galvanometer scanner, there are two mirrors which will bend the beam to move the beam in the x and y axis. So that's all. And here there is a tablet interface. We will be using this tablet interface for communicating with the controller, with the laser. So currently the laser is active, and we can change the pulse duration by changing the compressor position, and also we can change the average power also by using this tablet interface. Also we can adjust the pulse repetition rate. So we have currently optimized the system from a value of 200 kilohertz to 1 megahertz. So we can set the repetition rate values also. To control this galvanometer scanner motors, we are using a software which is installed to this PC. So this software is from this company called ScanLab. So we will be running our we will be importing or drawing the geometries inside this software, and we can use this camera to see the machining in real time. So now let us see the procedure for starting the system and to load a geometry and to do the machining. Let's see that. So first of all, we have to turn on a preamplifier, the preamplifier. Then we have to turn on the final amplifier, and then the galvanometer scanner. So first let's go and check the preamplifier one. So we have to rotate this particular key to turn on the preamplifier. So after turning on the key, we have to go to the tablet interface. The tablet interface here, and we have to start the laser here. Now we have already started the laser, and it's active. So we can set the parameters such as pulse duration, the average power, and the repetition rate with these three controls. So after setting all these parameters, we have to keep our sample in the stage at the required location, and then we have to open this particular software. So after keeping the sample in the Z stage, we have to go to this software, and we have to we can either draw the geometry or we can import the geometry. So for importing, we can use file and then we can import, and we can select the file. Like I have already selected a particular file, and then we can select the scale, and we can check the aspect ratio of the frame also, of the geometry also. Then we can sender the geometry into the working area, into this particular sender, and then we can click OK. Now the geometry we have drawn in some other 2D CAD software will be imported here into this particular interface. So we generally used to import DXF files. So now the geometry has been imported. So this is a micro channel which we are going to fabricate. We are going to machine on a stainless steel sample. So after importing the geometry, we have to place the geometry at a particular location. So for that we can use this, we can right click and we can click the place option. So we can place at different locations, and we can do so many things. We can move, we can rotate, we can delete, or we can put some offsetting, we can do the filling. There are several functionalities which we can use. So after doing all those things, what we need to do is we need to open this particular shutter, and after that this is a mechanical shutter on the outlet of the laser. So after clicking this open shutter, the mechanical shutter will be opened. Now we can go into this laser control. So laser control in this right hand side, and this pop-up will be coming, and then we can click manual marking. So we are going to do the marking manually, and you can see that through the camera that the machining is happening. So after that we can take our sample, we can close this shutter, and we can take our sample. So this sample has already been machined. So this is the micro channel which we have fabricated. So this might be having a depth less than 100 micrometer. So if we want more depth, so we may have to use the particular stage. We may have to move the stage upward. So after machining our sample, we have to stop the laser. We have to switch off the final amplifier by clicking here. So after, we have to wait for some time. After the final amplifier has been switched off, we have to go and switch off the preamp. So now you can see that we have switched off the final amplifier. So now the screen is showing that it is ready to start. So the preamp is already on, the final amplifier we have switched off. So now we can switch off the preamp. So let's, by rotating this, we can switch off the preamplifier. So now both the amplifiers are off. The oscillator which is inside this laser will be on every time. So we don't have to switch off the oscillator. So that's all. Thank you.
Thank you all the demonstrators for the very informative session. Now we will take a few questions from the participants. So our first question is asked by Romeo Sagalsem. The question is from FESM, so I request Kanishka Murmu to be online. The question is, can we see the cross-sectional image of nanostructures, nanowires, nanocolumns, etc. in 200 nanometer range using FESM?
Hello. Yeah, this is Kanishka here. So thank you for asking that question. So whether we can characterize the cross-sectional area of any nanostructure, yeah, it is possible. As the range is 100 to 200 nanometer, so it is easily possible to characterize the cross-sectional area. But in order to get the cross-sectional area, you have to place the sample, I mean the cross-sectional area you have to place towards facing towards the objective so that you can easily see the cross-sectional area. Thank you.
Thank you, Kanishka Murmu. So our next question is from Angana Bhattacharjee. The question is from LMM. So I request Amir Suhail to be online. The question is minimum gap in between two lines in MMM. Also, what is the minimum width of a structure in LMM?
So the minimum gap.
So first question is what is the minimum gap between two lines? So it can be in the direction in which the scanner is moving, and it can be in the direction perpendicular to the scanner motion. So in the direction in which the scanner is moving, we can get it as low as like 500 nanometers, or like even less also, and we can get it up to 5 to 10 micrometers also. So for getting for optimizing, for tuning that particular gap in the scanner motion direction, we have to optimize the pulse repetition rate and the scan speed. So with by choosing these two parameters, we can get the value of dx, that is the distance between in two pulses in the scanner motion direction. And in the perpendicular direction, we can vary that in the geometry by giving different hatch width during drawing by filling. So and the minimum width of structure in LMM, that is possible, that will be the that will depend on the beam base diameter. So our beam base diameter can go down to 5 micrometer. So we can make structures as small as 5 micrometer. That's all.
Thank you, Amir. So we'll take our next question. The next question is from Material Printing System. The question is asked by Ramesh Babu. By using MPS, can we print some biomarkers? I request Vimal Kumar Yadav to please answer this question.
Yeah, thank you, Ramesh, for asking this question. Yeah, definitely we can print biomarkers like isoigens like GST and different kinds of proteins and lipids using biomarkers using this MPS. Yeah, definitely the channel dimensions are in micron, so that we need to take care of that. So yeah, the answer is yes. We can print biomarkers.
So we will take one more question. The question is asked by Emily. In RF sputtering, can substrate heating be done during the deposition and up to what temperature? Sunil, you can please address this question.
Yeah, thank you, Anushat, and thanks for asking this question. Yes, substrate heating can be done in RF sputtering system, and it can go up to 800 degrees. See, in RF sputtering, it depends upon the material which we are going to sputter. In order to increase the crystallinity, we can go for substrate heating. Yes, thank you.
Okay, so we'll take our last question. The question is on laser micromachining. How much time you have taken to make the above structure? Amir, you can address this question.
So hi. So it will depend on the number of scan passes you will be doing also. So to do one scan pass, it will take approximately or around 10 minutes. So comparing to other techniques, even with other lasers also, this is a rapid prototyping technique in which you can get a high process capability. That is because the pulse repetition rate can go up to 1 megahertz and the scan speed can go up to 1 meter per second. So generally, the time taken for the machining will be significantly smaller. Thank you.
Thank you. So now we will move on to our next section. Our next speaker is Dr. Divyendu Das. Dr. Das is an assistant professor in the Department of Chemical Sciences of Indian Institute of Science, Education and Research, IISER, Kolkata. He received his MSc degree in organic chemistry at the University of Calcutta in 2005, PhD in supramolecular chemistry from Indian Association of the Cultivation of Sciences 2010, and postdoctoral training from the Emory University. From January 2017, he is working in IISER Kolkata. His lab is actively working in the field of systems chemistry and driven self-assembly. Some of his recent achievements include the DST Swarna Jayanti Award 2020-25, joining the Early Career Advisory Board, ACS Chemical Reviews 2021, and selection as an associate of the Indian Academy of Sciences, IASC 2019. Today, his topic of talk is system chemistry, how complexity emerges from chemistry. So now I would like to request Dr. Divyendu Das to please take over.
Thank you for the introduction. I hope you can see my slide. I'll take that as a yes, right? You can hear me. Okay.
Yes, sir, we can hear you.
All right. Yes, great. So thank you for the introduction. Thank you, Professor Uttam Manna and Professor Miwan Mondal for the invitation. It's always a pleasure to be with IIT Guwahati in person rather than online, because the campus is so beautiful. But anyway, I will start with a topic which is we are doing for the last few years, and this is a very new topic of chemistry. It's called systems chemistry, and it has relevance in various fields. I will talk about that. I'll begin with the line or quote of Schrodinger, which is living matter evades the decay to equilibrium. It's a very interesting quote. A physicist quoting about life and living matter. Life is a bit more complex, but living matter definition is something that he has attempted, and I will try to shed some light on that. Okay, so systems chemistry, this is if there are students in the audience, this is a phylogenetic tree where life started around 3.5 billion years ago and took three domains, bacteria, archaea, and eukarya. And we are somewhere here, human, right? But what exactly happened here is something that is under the purview of systems chemistry, that whether we can understand how life began, and more importantly, whether we can create intelligent materials out of that, because the most intelligent material would be living matter, right? And then this guy comes in. This is coronavirus, in a way funky cartoon, which is living or dead. Some confusion is there. It can show evolution. It can not produce its own protein. So this is something that is stopping the world currently. But anyway, so this is interesting. In between that, we are going to talk. Our lab is interested in, and even beyond that, can we create life 2.0? So that is a tantalizing question. That is something that it's not anymore a science fiction. We have some data. The team of systems chemistry have some data on that, which can show that indeed some intelligent properties can emerge out of non-equilibrium systems of chemical systems. So I'll talk about that. And in fact, these two are interrelated. If you can see that the origin of life and creation of living matter or life-like object, if you may say. Okay, so this is the review of a meeting which happened recently. So you can, if you want to go for it, you can read that. That talks about a recent, one of the first conferences of systems chemistry. You can have a look if you want, if you're interested in that. There are many people who talked about it. But before learning supramolecular systems chemistry, we should talk about last 6 decades of supramolecular chemistry, right? So systems chemistry, as you can, from the name, or some of you might know, that is intricately related with supramolecular chemistry. And supramolecular chemistry last 6 decades has given us a lot of products, like, for example, liposomes. The liposome market is growing. It is useful. One of the first marketed nanotechnology, you can say. From supramolecular chemistry, we can design enzymes in a very simplistic, minimalistic structure. We can design cellular matrix. That is also interesting. And obviously, Nobel Prizes have been won on supramolecular chemistry on 1987 and 2016, right? So supramolecular chemistry has been useful even as we speak. For example, in this COVID-19, this remdesivir has been known to be a very important drug, and it was solubilized by beta cyclodextrin, which is a cup-shaped molecule in a way where you have the drug inside, and you can do that. Gilead's, one of the leading drug company, used it. So there you have it, a textbook application. So cyclobeta-dextrin, beta cyclodextrin is something that many people work on. So this is the drug, and this is the cavity molecule. The point I'm making is that supramolecular chemistry has been useful, has been mimicking life, has been trying to create product for the last 6 decades. And for example, this 2 snapshot I've given from other, this is not our reviews. For example, nucleic acid based supramolecular systems, which has been useful for drug delivery, filtration devices, sensing and whatnot. Peptide based structures, which has been useful for various functions. But these are inspired in a way from nature. Again, MOFs, molecular crystals, micelles are all examples of supramolecular chemistry. But if you look at the Gibbs free energy versus reaction coordinate plots of these materials, you will see that this is the Gibbs free energy, and this is the starting condition, and this forms a micellization or a nanostructure or a MOF. Usually they are downhill or in a kinetically trapped state. So this is a reaction on the x coordinate. You can think about a reaction progress or reaction coordinate. So here it is downhill and is stable, excuse me, or it is kinetically trapped, still downhill, depending on the barrier. But living matter, there is a profound difference, and I will come back to the term of the quote of Schrodinger. Living matter, although these systems are inspired from life, but these are actually profound. There is a profound difference between these and living matter. So I'll talk about that. For example, systems level. At the systems level, we have temperature increase by some means. We have sweating. We control the temperature, right? So there is an equilibrium. There is a homeostasis, we call it. So there is a reaction. There is a response. There is a counter stimuli that controls the system to equilibrium. This blood pressure increases, vasodilation happens, it comes back to normal pressures. Cold, shivering happens, comes back to normal pressure, a normal temperature, thermoregulation basically. This homeostasis, this non-equilibrium system in a way, so you can understand I'm going away from equilibrium. So I was trying to give you the example of equilibrium. All these systems are under equilibrium, but these systems of biology, all of cell physiology, are driven by non-equilibrium events, non-equilibrium oscillations. These are always under energy. We're eating food. I'll come back to that. I'll eat food to talk. So the point is that all these systems are driven by energy. All right. So, and one of the classical quintessential example of a cell division. So I think I have a movie on that. Yeah. So this is a cell division. I'll show you a movie in just a bit. So these are the microtubules that are forming and separating two cells. These are also energy driven, and when the energy or the fuel is gone, is expended, the cell division will not happen. So this is again taken from a Nobel Prize site where ATP is required as a currency of our systems. All right, so this is the movie I was talking about. This is something that WBC is trying to run after a foreign object. This is driven by energy, and this is the cell division where the spindles are forming in a matter of 2 minutes, is forming in a matter of 2 minutes, is dissolving. So this is only possible when you break the constraints of equilibrium. So last 6 decades of equilibrium supramolecular chemistry has given us materials, stable materials. But if you really want to create acutely sensitive materials, acutely adaptive with temporal control of function, this is temporal control of function because this is happening only when it is dividing. At that time, it is burning energy. It is burning energy in terms of a fuel. A fuel is a chemical fuel. It's basically a thermodynamically activated molecule that is driving these processes, and this do a function act in a temporal manner. So we are not always using all our energy, otherwise we'll be burnt off. There's so much of energy. This is done in a very smart way, and this is done only when you break the constraints of equilibrium. So our goal is that whether we can mimic that in a chemistry lab, in a systems chemistry lab, where we can create material, chemical systems, small molecules, which can do something like this and can really help us design advanced materials of future. So that was the idea. So give you a quick recap. So this is the equilibrium system of supramolecular chemistry for the last 6 decades. This is the legendary dialogue of Schrödinger, living matter evates the decay to equilibrium. A steady state system in equilibrium where no work is done is death, actually. So inanimate. All the supramolecular chemistry products are inanimate in a way. But when you see the cytoskeleton proteins, which is seen across life forms as an evolutionary strategy, these are all driven systems. These are all breathing dynamic systems. So like lifelike dynamics is there. So this is the equilibrium gives free energy plot, and this is the x-axis reaction coordinate. Here you have to add energy for the monomer to reach to this polymerized state, and this is there as long as you're adding energy. When you're adding the energy is expended, this comes back to equilibrium. So to give you an idea, so I will come back to that in maybe in a detailed way later on. So this is the green filaments you saw in the movie, where the filaments were forming very transiently, doing the job and then dissolving away. This is something like that is going on, where you are adding a GTP, which is driving the polymerization. These polymers are basically these green filaments. In the polymerized state, the GTP is broken to GDP. So in a way, if I draw this diagram in presence of this diagram, you will see that these are the two same diagram, where the building block in presence of an energy source like a GTP drives it away from equilibrium. The assembled state breaks the GTP to GDP and it returns to the equilibrium. So this is a non-equilibrium dissipative state, and this is the equilibrium state. So this is where the fun begins. This is where the burning of energy begins, and this is where we can control functions in a temporal way and really have acute adaptability. So this is the difference between these two, where energy you're adding to go to the polymerized state and you're coming back to the equilibrium. So to give you in detail in a very small way that the building block starts, this is the polymer that is forming in presence of a GTP, which is, we all know, a phosphate bond with high energy, which is basically thermodynamically activated, kinetically stable molecule. This drives the polymerization. That is okay. In the polymerized state, this GTP is broken to GDP. So the polymer is a catalyst. The building block is not a catalyst. The polymer breaks the GTP to GDP, and that return crashes to equilibrium. And that's why we call it a feedback-driven system. Why? Because after the assembly is formed, then only the catalysis begins, and then only the GTP is broken to GDP. So that's where the very interesting part, or one of the classical quintessential examples of non-equilibrium self-assembly happens in biology. And if you have to plot the rate, you will see a spike of activity, which is almost a smoking gun proof that indeed there is a dissipation happening from the assembled state. Dissipation means by a cup of tea you take, and that heat is dissipated from the surface. Just like that, this energy of this GTP around 12 kilocal per mole is dissipated from the assembled state, and that does a function. Muscle movement, this cell division, this WBC, the motility are all done by this. So these are the buzzwords: acceleration catalysis from assembled state, and a negative feedback due to an acceleration catalysis, and dissipation from assembled state. So GTP to GDP is happening from the assembled state. So we were not the first when we started our work in ISER Kolkata to start the field. There were two seminal works. I'll briefly talk about them. They are very simple works in a way. They took an organic acid. They started with an organic acid. Then they added a methylating agent, which rapidly methylates this organic acid to decrease the repulsion, and then it rapidly forms a polymer. The fun part begins when they have a hydroxide in the medium, a sweet balance of hydroxide, which breaks the ester to the acid. So what happens is that there is a transient formation of these nanostructures, which looks like exactly like a microtubule. A microtubular cytoskeleton protein I showed you. There are videos. If you have time, you can have a look at the movies by this paper. They look like to be really forming and breaking. But then we ask that, is this really a negative feedback? Is this really an assembled state catalysis like the biological cytoskeleton protein? The answer is no, because they are using hydroxide rather than a catalysis by assembled state to break the ester to the acid to return to the equilibrium. So this is the equilibrium. This is the away from equilibrium structure. Similarly, next year, one fascinating paper came up from Prince Group, which showed that amplified in presence of ATP now can form rapid vesicles. So that was okay. That was expected. That can happen. But they had an enzyme concentration, again, a sweet balance of enzyme concentration, which is slowly breaking the ATP to the AMP. And that resulted in a transient formation of the vesicle. Because this rate of hydrolysis of ATP was slower, the formation was fast, and hence you can see a population of vesicle. But again, the feedback was not from the assembled state like the cytoskeleton proteins or microtubules, but it was from an external source. In fact, Prince came up with a review next year or a year after that and criticized both these works, that these are good works, fascinating works, but these are not true mimics of non-equilibrium self-assembly of biology. So if I have to juxtapose the two diagrams, this is the science paper by Vanish, and this is the paper this is the actual real scene that is going on in the human system, or the living system rather. So here we see that GTP is driving the polymerization, and the polymers are breaking GTP to GDP and it's coming back to building block. In this case, you are adding a hydroxide externally, right? So there is some cheating going on, but that's okay. They are getting a transient polymerization here. So at this point, we came in and these are the works we have shown over the last few years, that indeed it is possible to show enzyme-like catalysis at the assembled state. And then we can show a substrate that can drive a polymer, and the polymer or the assembled state can break the substrate to return to monomer, right, or building block. So that is the key we have shown. We have taken two major steps. One is a cooperative catalysis, that is like this, where two histidines are very close and one act like basically proton shuttling. The other one is a binding pocket generation, where in assembled state they will be binding pocket and they will break the substrate. So this is the story, and I will begin with the first story. This paper has been incidentally cited almost 50 times last year paper, because this was the first contribution from our work, which is where Shubhajit started. Shubhajit was the first author of that paper. What he did was very simple molecule. You can see one lipid-based, this steroid histidine, basically this is. He added in 20% DMS of water. He found no networks, no hydrogel formation, nothing. But when he added this 30 millimolar, very less amount, 12 millimolar of this surfactant, amplified in a way, and added a nucleophile, which is a phenol, and an EDC, which is a coupling agent, he mixed them together, he found a gel to be formed. So we expected that, although there is a subset, this is skewed concentration, it starts with 12 millimolar, we are adding only 4 millimolar and 4 millimolar. We expected that there is some co-assembly going on, that this EDC is basically a coupling agent for people who are not chemists. This can form a coupling and form a ester, a kinetically stable ester, a thermodynamically activated ester. So this was forming a gel. But what was shocking was that the gel was transiently formed. So it was sol initially, then we added this EDC and PNP, it formed a gel, then after 30 minutes it becomes a sol again. So this is a fuel, we can call the EDC as a fuel, which is for driving the generation of this gel, and that gel is then dissipating to access the sol. So you can do a TEM of that. We have done ACM, we have done cryo-EMs extensively to make the reviewers happy. And we have found that indeed there's some transient out of equilibrium structures are being generated. We have seen movies also. I'm not including the movies for sake of time, but these are forming structures They're transiently formed just like microtubules and then dissipating away. The key part here is that we are not adding any external reagent which will result in this disassembly. So we have done spectroscopic technique, not just a visual confirmation, a bulk spectroscopy like a UV and an NMR we did, where we observed that indeed transient formation of structures are happening, but in this case a control where there is no self-assembly properties, it is not doing the same. So that is very cool and very interesting to see that we are indeed able to have a transient gelation. We could do refuel, just like biology. Biology does refueling. It can use the same system over and over again, as long as the individual survives. So it is like a GTP is breaking to GDP, and then GDP is again converted into GTP by various energy sources. So here we added EDC again and again, and we found that this transient phenomena was continuing for as long as 3 cycles. After that, the waste, it is a waste product. Biology, living systems can excrete the waste, but here a poor vial cannot. So we cannot do beyond 3 times, but we can prove the proof of principle was there that indeed this can be recycled multiple times, at least 2-3 times, to show that this is acting like a refuel driven system. And we did investigation of the activity, and we found that indeed 2 histidines are coming together. This is a very simple experiment. So this is a pH based experiment where we found that 6.5 the highest activity was there, which is kind of a very classical example of a cooperative effect where 50% of the histidine will be protonated, one histidine will act like a nucleophile, other histidine will act like a proton source. So that was the first story. So this is, if I have to again compare these 2 systems, this is the natural system which is happening without the presence of any external deactivating source. Here also we are getting a system where we are without in the absence of any deactivating source, we are getting disassembly. So this was very interesting to see. Then Pavel came in subsequently with an idea that we will remove the EDC out of that, and whether we can do a reaction where we are adding directly the fuel and to generalize the system basically. We have taken the same surfactant, if you notice, it's just drawn differently. It's a steroid histidine. We added an ester directly. So we added the ester. This is the cartoon of the ester, and this is the surfactant. We mix them together, and 2 hours we see the fun. So what happens if I take the amino acid alone? We don't see any gelation. We take the this ester alone, we don't see any gelation, in fact precipitation. When we mix them together, this surfactant and this ester, we find a transient gel. Gel is forming a weak gel, and then after some time it is just crashing out. So it was very interesting to see. Now we have just removed the complicacy of EDC and we have just added ester, just like biology. Biology adds GTP or ATP. ATP fuels actin polymerization. GTP fuels microtubule polymerization. In this case, our poor small ester can actually fuel this self-assembly in a in vitro system. So this was very unique to see that. And then we did rheometry, we did CD, we did CD, and we did fluorescence experiments where we saw that indeed in bulk spectroscopic methods or mechanical strength measurements, we could see an increase of transient G prime strengths. For example, the gels are forming and then breaking away. The CD is showing a formation of an emergent supramolecular structure, which is over time then slowly crashing out. So we found that when we did the TEM of this system, we found that a helical structure was being formed or accessed when we added this fuel to the surfactant. So this helical structure were formed, and that was actually creating the this was dissipating the energy, and this was temporarily formed. And if I did we did a cryo-EM, and we found that helical structures are indeed formed. So this was quite fascinating that yes, we are also getting an emergent morphology. So we did a control, and we call it a driven control. So what we did, we used the same surfactant. Now rather than adding the fuel, we waited for 12 hours. It formed some structure. Now this we are calling it equilibrium structures. After the equilibrium is reached, if I add the fuel here, we are not able to access the same helical structure. We are not able to access the gel. See, this is the actual image where we take the vial, we add the wait for 12 hours. It becomes a bit more thicky, not a gel. Then we add FNP, which is a ester or a fuel. this fuel is not able to drive the gelation. It can form some color. Obviously, some hydrolysis will go on, but it's not able to form the gel, and importantly, it's not able to form or access this unique helical structure. So we call it negative feedback is not there. This emergent helical structures was giving a negative feedback to its own growth. So it was degrading on its own, right? On a kind of autopoietic system. So so this is creating the resulting in an equilibrium. In fact, we did a TEM and we found that no helical structures were formed. So one of the best part was that when we did these two systems, this is the equilibrium system and when this is the non-equilibrium system, from beginning we started the surfactant and waited for 12 hours and then added the fuel. This was the plot of the rate. But when we mix them together, the fuel and the surfactant, we found that there is a spike of activity after some time, which is a smoking gun experiment, we call it in the lab, where it shows that indeed there is an acceleration of catalysis after the assembly is formed. And that results in the dissipation of energy. The energy is dissipated, energy of the bond of the ester, and that results in the return to equilibrium. So this is the example, if I have to compare between these two. Again, this is the living system to show you that we are not using any external system, external reagent. We are getting the dissipation of energy form on its own. So then Chandru and Shubhajit came in and they thought that let's extend the idea and make it generalized to the extent that we can have actually a peptide-based system which can form a catalyst, which can form a better catalyst, because peptides are known to be better catalyst than a simple amino acid. And then we can see whether we can simulate similar structures in a complex amyloid polymerization. So what he did, they started with a big sequence. Obviously, they did not take the entire sequence. They took a nick of it, KLBFFAE. This is 7 residue nick. And this sequence they took, and then rather than E, they mutated E to L. And then they mixed them together. This was 90%, let's say, green arrow, and green arrow with a red marker or brown marker was 10%. When they mixed them together, they found a gel to be formed. All right. So then what they did was this lysine was mutated with a histidine to make it catalytic. And this glutamic acid, in this case, rather than a leucine, they used a paratophenyl ester. Now they mixed them together, and indeed they found, as per expectation, that, so this is the system that I was talking about. This is a natural sequence that is all in our brain. This is a human brain sequence. We are taking the nick of that, nucleotide code of that. When we mix them together, we find the gel to be formed is stable for 24 hours. And when we took this separate mutated gel, we started with this histidine-based gel and then histidine with a ester mutation. When we mix them together, we found out that, well, this was forming a gel, but after some time it was crashing out. So this was forming this non-equilibrium polymerization of amyloid. So this was, we are talking about now polymerization, when amyloid is usually known to be a rogue protein and it goes downhill equilibrium trap of these structures in the human brain, and that's where the disease starts in. In this case, we are seeing that it is not going there. It is in fact taking a completely new state or a steady state of, you may say, steady state in the energy thermodynamic surface or a thermodynamic terrain. It is going to a non-equilibrium state because we are adding energy and this coming back to equilibrium. So if I have to see the data in terms of the spectroscopic techniques, we found out that the turbidity is increasing for the control sample, which is the equilibrium gel, is increasing and stabilizing after, let's say, some time. But in this case, the equilibrium, this non-equilibrium samples, the turbidity is increasing and remarkably going down on its own. So that was, again, very interesting. And then we found out that the CD also, obviously, these are multiple data to prove the same point, that drive home the same point, that this is accessing a non-equilibrium structure. So this is the equilibrium structure of CDP going up and stabilizing. But here the CDP goes up and then comes down after some time to return to equilibrium. So this was very interesting, and this was actually, I mean, this is something provocative also in a way. When we added Congo rate, and Congo rate is known as a gold standard for knowing the interaction with amyloid plaques. So when the Congo rate was added to amyloid sequence, And for example, this was the control sequence of the equilibrium samples. We found that indeed there was a shift as per expectation from 520 to 526 around. A red shift is there, which is again known in literature. In this case, the congruent is showing a transient binding, a temporal binding. So the congruent shows an increase of lambda max and then decrease of lambda max, and it comes back to again equilibrium structure. So this binding of this guest is also controlled, and that actually forms the basis of our sustained equilibrium non-equilibrium systems that we will do in future actually. So to give you one more data which is actually shockingly similar to cytoskeleton proteins, where we have taken that in cytoskeleton proteins, it's known that when we add GTP, it forms these transient polymers. But when we add rather than GTP, a less hydrolyzable analog of GTP. So GTP is stable but can be hydrolyzed. GMP CPP is a less hydrolyzable analog. It cannot be hydrolyzed very easily by these polymers. So when we added this GMP CPP analog rather than PNP, we added ester which is more stable. We found out that the ester was forming a gel. The gel was not breaking. So the point is that when we added a more thermodynamically activated but kinetically slightly more stable ester, it is not able to form the transient gel or transient fibers, just like we see in a cytoskeleton protein. So that was the interesting part. And in fact, I will show you that we are just scratching the surface in terms of non-equilibrium self-assembly, in terms of the functions, because it's very this non-equilibrium, this bond breaking and bond making is not only present in cell biology, but it's also at the heart of Darwinian evolution, right? And from there, we can actually create materials which can redefine actually chemistry. These are not our words. These are the words of very futuristic reviews by various people out there. So what we did with this system, we started with these peptides and we did casting of this on we collaborated with this with Ratish Vijayaraghavan. He has done this conductance studies. We found that this peptides was showing a transient control of the conductance. It was increasing, decreasing the conductance, and that resulted in a temporal conductivity, which you can understand, which can open up possibilities of very smart security devices, where you can add a marker or a molecule. The security device will be active for, let's say, after 2 hours, and then deactivate on its own after 2 hours after that. So it will be temporarily functioning. It can be refunctioned if I add the same trade secret molecule which can do that. So this is very interesting what we have received done this time. We have controlled enzyme activity also by this temporal systems, and we have also done very recently we have done AND gate and a concatenated AND gate, where we can sense very small simple molecules by these transient systems. And finally, we have also done what we call, this is a red line which shows a sustained delivery of drug by hydrogels, but in this case we have done a non-equilibrium gel which shows a temporal control of drug. So we take a drug every 12 hours or every 24 hours. In this case, the material will be released and then held back and released and held back temporally. So that will be done because these systems are forming in a function of time. Normally, all this supramolecular chemistry or the products of supramolecular chemistry are stable, right? They don't change with time. But in this case, we are having a function of time, and hence the function will change slowly up and down. Okay, so I will finally wrap up with the last story of the day, last two stories of the day. Actually, I have some 10-15 minutes, it seems. So if you can excuse me, I will just 1 second. So I had to turn on my fan because it's getting too hot here in Kolkata, Kolani rather. Okay, so this is the second part of the story, which is showing that the systems chemistry can also give us ideas of how enzymes evolved. Okay, so this was the same slide what I showed, how earliest enzymes evolved, how covalent catalysis can be shown. Covalent catalysis is a technique used by advanced biochemistry to break very difficult substrate or do metabolism. In fact, cascade catalysis and substrate channeling is also something that is very interesting. So what we did, we are trying to create short peptides which can act like enzymes. So if you take any enzymes from the bench or you buy from a company, it is at least 100 amino acid long, and how nature found this sequence from an astronomical pool of sequences is anybody's guess, and we might not ever be able to answer that. What we feel, our contribution is that we think that short peptides like amyloid might have been the earliest protein folds which can pack and create functions, not only simple hydrolysis oxidation reaction, but it can do beyond that. So what I will show you is that a recent data from our lab that short peptides forming assembly can do metabolism, kind of like reverse retroaldolase and forward aldolase. This is a snapshot from a Wikipedia page of an aldolase enzyme, which with the lysine has been used as a enamine to react with one more aldehyde, or it can do a retroaldol by the same lysine can do, where the CC bond manipulation has been done in various metabolic cycles. So what was there in the report was this computational work where they have shown that this substrate can be broken by a de novo enzyme, a computational designed enzyme, by to this aldehyde and this ketone by a process of retroaldol. So what Antura and Pavel did, they started with a peptide lipid sequence, and then they found out that it forms a nanotube and nanostructures. I will skip these parts. This small peptide forms these structures and can bind to small molecules. As you can see, it can binding to small molecules. Importantly, when they added this sequence, this peptide, to this aldol, it was rapidly getting cleaved. It was rapidly getting cleaved in a very good rate, one of the best rates so far reported by any short peptide. So it was very unique in that respect. Secondly, we could see that this HPLC profile, we could see that it was able to select one substrate of the other. So it was showing, like just like an enzyme, a very nice selectivity of substrate. It was showing also some enantiomer selectivity, which I'm not showing here. Some enantiomer selectivity was there. So this short peptide, which is a fraction of an aldolase. Aldolase are 60,000 molecular weight. This is around 800, so very small. But these are doing functions which are like aldolase. Also, when we added some, just like aldolase uses tyrosine, we added some tyrosine and found that an active site like can be generated, where in presence of tyrosine, the activity even increases further. So this is very interesting, and this is the POC of the paper to show that when we mix all of them together, there are 1, 2, 3, 4, 5 ketones, one aldol adduct. We found that exclusively one aldol product is forming. There could be 8 different products, if I am not wrong, that can be accessed. But from HPLC and mass spec, we could see only one product is forming. That shows volumes, speaks volumes about this short peptide acting like a very advanced rather enzyme. So then the final part of the story is that we went ahead and we found out that what else it can do, whether it can do a covalent catalysis. Covalent catalysis is not reported by chemical system. Covalent catalysis is something that where multiple amino acids join hand to break a same substrate. For example, the serine proteases seen in nature, where serine, histidine, and the glutamic acid are coupled together, and one acts like a covalent bond. Here is a covalent bond, and the other breaks the other amino acid breaks the amide in this case. So this is very interesting. This is called covalent catalysis. This is not reported in literature by systems, chemical systems, very simple chemical systems. So this is even in the eye, we have this reversible covalent bond, which has been used to for the cis trans isomerization, and that can result in signal transactions cascades. And also this kind of a enzyme where two residues like a lysine and a histidine has been used to do a job. So basically, one hand grabs the substrate, other hand breaks the substrate. That is how enzymes sometimes get these phenomenal rates over the other enzymes or over simple catalysts. So we wanted to mimic that, whether we can mimic this covalent anchoring. This is called covalent anchoring. You can see the lysine, which is basically a primary amine, is forming this imine bond. This imine bond is transiently formed, and then the reaction happens, and then again this imine bond is relieved. So it is almost like a sacrificial right hand and left hand doing the job, right? So whether we can do that, can we use such covalent catalysis in assemblies of short peptide? And that's where we published it this year again. Boishaki and Oyon started with this peptide. I will not go to the sequence of that, detail of that, This is the crux of the story. Peptide showed that a lysine was there. The lysine was forming an imine bond. A histidine was there. The histidine was slowly breaking the ester, and that is resulting in a high rate, substantially higher rate than a control which had exactly same sequence. Morphologically indistinguishable nanotubes were formed. So the nanotubes, whether you can see, these were exactly same whether you take lysine or arginine. Similar nanotubes were formed, but the arginine is not able to break or not able to actually form this imine bond because of obvious reasons, doesn't have a primary amine. In this case, the activity was substantially lower. So both these catalysts have the same amino acid exposed, catalytic amino acid, but a secondary amino acid, which should not have any earthly business with this ester hydrolysis, takes part in a very nice way of covalent anchorage and breaks the ester bond. So in this case, what we did, we found out that indeed the lysine-based these nanotubes were showing the highest activity, while the arginine-based nanotube was substantially low activity. Although, again, I will remind the audience that both have the same imidazole exposed. One has a imine. One can form an imine with a substrate. The other cannot. We did some other controls, and we did one more control with an ornithine, where rather than lysine, ornithine has 1 carbon less residue. So I will just show you the sequence. So this is a lysine, this is an ornithine. So 1, 2, 3, 4. It has 1, 2, 3, 3 carbons, 1 carbon less. So when ornithine was used, because it can cover less surface area on the surface of the nanotubes, the activity was substantially low than this one. So it was following a very nice structure function relationship and showing us that in this designer friendly peptides can access catalysis just like a very advanced serine protease or a covalent catalysis seen in even the human eye to the dehydroquinase. So this is very important in that context. So with this, I will close. This is done majorly the works of talked about by Shubhajit, Pavel, and Antora, mainly these three, and Chandro. Others works I'm not talking about. Obviously, for the lack of time, we are funded by Nanomission, Inspire, and DBT, and obviously Aizer Kolkata. And this is the campus of Aizer Kolkata. This is a brand new 1,500 seater auditorium, one of the biggest in our place, we can say. Our campus is not as pretty as yours, but it's not bad also. So I would love to welcome you all when we have time and things get back to normal in future. So this is the logo of the lab where we are talking about systems chemistry from information transfer to reactive materials to structure confinement, scaffolds to function, which is emergent catalysis and chemical networks. With this, I thank all of you. Thank you. Thank you. I'm done. Thank you.
Can I ask some questions here? It's
sure,
a great
sure.
talk. It's a great to hear you. You know, it's amazing work. It just in a different purpose. It's what I understood that all of this system mostly works with the water, right? It's form hydrogel, right?
Yes, some of them form hydrogel, some of them form just nanostructures, like soluble nanostructures. The amyloids form nanotubes.
Right. No, but the first two system where you took this lipid kind of, right? So those are formed the hydrogel, or it can be also prepared in using the organic media.
So we are using 20% DMS of water. We have not tried with toluene and other things. It might be forming a gel. Usually, a surfactant can have the ability to form an organogen and hydrogel depending on the moieties it has.
Okay.
Right. So to answer you, we have not checked it. We have not checked organic solvent. Yes.
Okay. So do you think that if you can prepare this like organogel, they will remain there, the ability remain intact, or the kind of this transient gel formation?
Yes. Right. So that depends again how much water is there, because we are using a hydrolysis reaction. We need to have some water for the reaction to happen. So, but the transient organogel is not yet reported. That will be interesting to see in the literature. But I doubt that if we take 100% organic solvent without dehydration, dehydrated organic solvent, I doubt the hydrolysis reaction will work. But there will be something else that will open up. For example, there could be a CC bond cleavage or something like that can happen.
Exactly. Another thing is like just I don't know, like if it is work, then it would be like another interesting application you might thought about, like where basically catching some oil phases, like so my mind goes there, right? And then recovery of that oil, so where basically you just add to just accelerate the hydrolysis, you just add a little bit of the mixture of water ethanol, and then through there, so that ethanol will help them to go there, and then it accelerates this hydrolysis, and that we can collect or recover the oil phase. Similar to that kind of
Right. Yes, that is interesting actually. All the functions that are there for the equilibrium systems, for example, this oil recovery, can be accessed here autonomously, sort of. These are autonomous systems. So it will automatically release the oil at a certain point.
Exactly.
Now the lifetime will be important. How do you change the lifetime? So you want it to be gel at the time when it wants to trap the oil. You want to be sol. So you can change it as per the fuel concentrations. But I think it's a great idea. Yes.
Another thing is like how difficult to synthesize these things, like particularly the initial two molecules.
So these are one-step reaction actually. So what we did, we took acid chloride of a long chain acid surfactant. Let me just show you acid chloride of a stearic acid, and then we added histidine. So we didn't even protect it. Can you believe? So initially we started with protection and coupling, but this is reported actually. This surfactant is not the unique one. We have the concept is new. So surfactant is reported by others. By C18, C16 has been reported, I guess. Yes.
Okay. Sounds good. Great. Probably I'll catch you later.
Thank you. Yeah, sure.
Thank you, sir. Thank you for the interesting talk. So participants, if you have any more questions, you can ask. So I think there are no more questions. Thank you, sir.
So can I ask another one?
Yes, sir. Please go ahead.
As the participants are not asking, so so in your another system where you're showing this enzymatic activity where lysine play a crucial role, right? Lysine and histidine, I guess, right?
Yes.
Two functional groups. One is basically kind of dissociating, another is basically kind of reacting, right? So if you can go that slide.
Yes. This one, right?
Right. So the position of those two functional group also should play a crucial role, right?
Absolutely.
So sequence is playing a huge role, probably, right?
Right. So the substrate length, for example, the substrate length and the two residues should click together. So based on that, but the advantage is that Uttam, these are breathing a bit. So we have seen from various studies the degrees of freedom, and they are having some water in the bilayer, so they breathe a bit. So the accessibility is a bit more. So we could actually cleave ester, these two esters. So this is an imine can form here. If you can see, the lysine can form here, and limine, and this ester will be broken. And then we did this one also. This is pretty far actually. This one was the imine, and this was. So both can be broken at almost equal ease. The point is that the distance matters, but not to that extent. But if we had a very long substrate, that would be difficult. So that's the advantage actually. That makes it to be
give
specific.
the selectivity. It gives some selectivity.
Yes. Yeah. Absolutely.
Great.
Yeah.
It's amazing.
Thank you. Thank you.
Thank you, sir.
Okay, so should I stop
now,
presenting? Yeah, yeah.
Yes, sir. You may. So now we would like to move on to our next talk. Our next speaker is Dr. Swapnil Sinha, founder, director, and CEO of Bioaptagen Laboratories Private Limited. Dr. Sinha has over 10 years of experience in oncology, biopharmaceutical drug development, and epidemiology. She earned her PhD from CDRI Lucknow in 2009 in malaria epidemiology and population genetics. Postdoctoral fellowship from IRCM, Canada, and Colorado State University, USA. And her research was focused on various aspects of gene regulation during cancer and cardiovascular diseases. Dr. Sinha worked as a DST women scientist at NAPER Guwahati from 2014 to 2017. And here her research interest shifted to biopharmaceutical drug development. In 2018, she founded the startup Bioaptagen Laboratories and secured the necessary early stage funding from BIG, that is Biotechnology Ignition Grant, sponsored by the Government of India. Today, her topic of talk is development of DNA-based apta sensors using cell SELEX and nanotechnology. So now I would like to request Dr. Sinha to please take over. Please, Dr. Sinha.
Yeah, thank you for the introduction. And still, I think it's good morning to all. So I know since the conference started, most of the time you'll be hearing, because I know that you're in the audience, you are mostly from the chemistry background. So I'm sure this talk will be really different to your ears because I'm a biologist by heart. So I will try to make it what you call it user-friendly talk, which you will understand, and then you will understand what we do and how we incorporate our ideas with nanotechnology and with electronics also. So, okay, my slides are remotely handled. So Moshmi, can we go on the second slide? So like you have heard in my introduction that I have a company, it's a startup. So we are based in IIT Research Park, IIT Guwahati. And the sole aim of our startup is, this is kind of an innovation driven startup. So we make, you must have heard about biosensors. So when we talk about sensor, is something that senses something, right? And so biosensors are, when bio is attached in front of sensors, it means it is sensing something which is of biological origin, right? So what it's in the simplest of the language, you can understand in this way that if there is a biomolecule, so what you want to detect in say any biological sample, be it a urine, be it a serum, or cerebrospinal fluid, anything which is of biological origin, and you want to detect some biomolecule, some metabolite in that sample, right? So what you do is, of course, these molecules are small, you cannot see them, so you need some kind of sensing mechanism. So what we do is we develop something that will attach to this biomolecule in a very specific manner, and then it produces some kind of signal, be it a color signal, it could be a fluorescence, it could be a light signal also, and that is amplified by different procedures, and then which you finally read, it is read in a inbuilt device. So in a simple way, that's how you can understand electrochemical biosensors. Moshmi, can we have next slide, please? So this is what we do. So let's move on the second slide. Next slide. So what we do is, so before telling you what exactly we do in our company or in our startup, so you can detect these, you can make many biomolecules into and develop them in the form of a biosensor, right? You can use simple DNA molecules, you can use RNA molecules, you can use antibodies. So most of the diagnostic tests that are available in the market for most of the diseases, they are antibody antigen based. So antigen is something which is there in your body, and antibodies are something which recognizes your antigens. So you might be hearing a lot about COVID rapid antibody testing. So that is how it works, right? You have an antigen, which is your spike protein of the coronavirus, and the antibodies are always there in your testing test, which you are using, and that detects, that actually captures the spike protein of the coronavirus. So antibodies, then you have a lot of other biomolecules that could be developed as biosensors. They're hormones, different kinds of enzymes that are produced in your serum, in your urine, and many other biological fluids, and many other metabolites, right? Can we have the next slide, please? So if you can see that we have this DNA molecule, right? So our company is basically, we are developing DNA biosensors. And why we opted for DNA biosensors over many kinds of protein biosensors that are already there in the market, and a lot of people are working on that. See, DNA have a lot of advantages, right? First of all, it is highly sensitive. So whatever diagnostics that you are developing, the first thing that should come into play is that it should be really sensitive, meaning it should do what it's supposed to do. It should detect the disease or any biomolecule in a very precise, in a very specific manner, right? So that is the first aim of any kind of diagnostic test. And then your response time should not be more, right? It should be quick, meaning when you're testing it, your test should give results in few hours, not in days. Like there are some tests which takes like 2-3 days to come up with the results, but you don't really need that. You need something that technologically advanced that should give you results in at least in an hour or so, right? And then the operations should be very simple. And that's how you can make any kind of sensors user-friendly and point of care. Because India is a very different tapestry, right? You have villages in which you don't have trained personals who use those diagnostic kits. So your diagnostic kit should be that it should be used by any simple community healthcare worker or any Anganwadi worker. It should have simple operations, not much directions on the manual. And of course, it should be low on cost so that it could be affordable by lowest level of people who are not economically that much advanced, right? So that is how you make everything point of care. It should be, although you want to make something point of care, but in the back of your mind, you cannot ignore the fact that your test should be highly sensitive and it should be very specific in nature, right? Can we have the next slide? So this is what actually we do in our lab. We develop these DNA biosensors for many types of diagnostic, diagnosing many types of diseases. So what is the DNA in your sensor? So let's talk about, so when I say DNA biosensors, so what I know most of the people in the audience know what DNA is. So I just want to give you something in our diagnostic test, what we do, what is that DNA that we use, okay? Can we have the next slide? So just a little recap. This is, so you might be knowing that DNA is always double-stranded, right? That image that you always see in all the, everywhere, the DNA image, it is always double helix, right? Most of the time when we develop biosensors, be it a DNA-based biosensor or RNA-based biosensors, we use single-stranded DNA. RNA is anyway single-stranded, but when we use DNA, we always use it single-stranded DNA, okay? And this single-stranded DNA, it recognizes your target in a specific manner. Your target could be anything. It could be DNA itself. It could be protein. It could be any organic molecule. It could be any other biomolecule like vitamins. And that is the versatility of DNA molecule, that it is not just confined to binding to any protein molecule as it is in the case of antibodies. So antibodies are only developed, mostly are developed against the any protein molecule, right? That your antigen is most of the time is protein. But that law doesn't go with the DNA molecule. It could bind to anything. You can develop this DNA molecule for any kind of molecule. And when it binds it, we develop some kind of signal. Like I said before, it should give any signal, be it a color signal, luminescence, or fluorescence, which finally is transduced into an electrical signal. Can you move on to the next slide? So just a recap that what is really DNA and RNA molecules are in terms of their chemical structure. They're made up of three moieties, which one is your nitrogenous bases, and then your phosphate backbone, and your ribose sugar. So these are the three components of a monomer of a DNA, which you call as nucleotide. And these monomers, then they with the formation of phosphodiester bond, they form the DNA chain or the polymer of the DNA. Right. And they are arranged in such a way that this phosphate backbone, it is called as a backbone, which is always exposed outside. So that is how this DNA and RNA molecule, they get their negative charges. Okay. Can you move on the next slide? So let's now talk about what are DNA aptamers. So the company name is BioAptogen, right? So what we do is we really develop aptamers. So that is why we kept the name of the company as BioAptogen, because we deal with aptamer molecules. So aptamers, they are a class of DNA molecule, right? So aptamers it comes from a Greek word which means aptus mean to fit. So any DNA molecule which actually fits or binds to a particular ligand, and this ligand could be anything. So that is what it is called, why it is called as a DNA aptamer. And these molecules are not long molecules. DNA molecules are polymer, as you know. It could be huge. It could be because your entire genome is made of DNA, right? So, but these aptamer molecules, they're very small molecules, and they are always single-stranded molecules. They're not double-stranded. They're not double helix. So in this, the range of this aptamer molecule is from maybe 50 bases to around 120 bases. So when I say bases, that means, remember when I was showing you the structure of the DNA molecule, you have nitrogenous bases. So when I say bases, I mean the monomers. So a monomer molecule of 50 bases, a monomer molecule of 120 somewhere around. That's a range of a DNA aptamer molecule, right? And when they are single-stranded, so if you see these two figures, they are in a form of a conformation. So that actually happens because there is a hydrogen bonding that always exists between the nitrogenous bases, right? So they have a ring structure, these nitrogenous bases, and they all have, so they are made of either purines or pyrimidines. So they have a ring structure and they form hydrogen bonding among themselves. And that actually gives a unique conformation to any kind of a aptamer molecule. And that totally depends upon the sequence of that aptamer molecules, or the sequence and the way these monomers or the nucleotides are arranged on this polymer, right? So, and this actually gives a unique structure to the aptamer molecule, meaning that they uniquely now bind or dock to a particular ligand, okay? Because, and that totally depends upon the conformation of this DNA aptamer molecule. So when you leave DNA in a water medium, just water is there, they don't really fold into this conformation. So they need this folding to get into this conformation. They need some kind of buffering. So you provide buffers in the form of tris EDTA buffers and any kind of buffer, biological buffer of a particular pH, which is generally set at biological range, which is like 7.5 to 7.8. And at that pH, these molecules, they generally fold into a particular conformation, which is unique. And then this uniqueness is then counted on when they bind to their ligand. Okay. Can we have the next slide, please? So, like I said, that is the versatility of these DNA molecules. They can bind to any kind of target. It could be metal ions, it could be simple amino acids, peptides. It could even be whole mammalian cells, whole viruses, and the bacterial molecule. So if you look at this list, you will understand that when I say total whole viruses and whole mammalian cells, it actually provides a very good free hand when you are developing any diagnostic kits, which is a DNA-based kits. So we should, for developing any kind of DNA aptamer molecule to bind to a particular ligand, you don't really need to know the conformation or the structural components of the target. What you need to know is your DNA molecule. And that actually provides versatility for developing these DNA molecules, which doesn't go with the antibody and antigen. For that part, when you are developing antibodies for any kind of protein molecule, you need to know the structure of a protein molecule. You need to know what is your protein molecule. While in the case of DNA aptamers, you just take the target, for example, like there's one kit that we are developing with in collaboration with IT Guwahati. I'll come back to that later. We are developing this kit for coronaviruses, for COVID detection. So we just take a virus, right? And we keep on incubating. So I'll tell you, we have a lab session also where you will see how these molecules are developed, DNA molecules. So what you need to know is you don't really want to waste your efforts in knowing your target. What you need to know is the entire procedure that your target should bind to your selected DNA molecule in a high affinity and high precision, right? Can we move to the next slide? So I was talking about this a lot. So this is just a glimpse of what we are at present, what we are doing at our company. So we develop DNA aptamers on routine basis, and we integrate that with molecular biology, synthetic biology, and nanotechnology for developing affordable diagnostics. So right now we are developing these 4 products. These are all aptamer-based products. So we already have in place a UTI detection kit, which is urinary tract detection kit, which detects the bacterial presence in the urine. We're also developing cardiac malfunction kit, which is basically for myocardial infarction. And then vitamin D3. So if many of you might be knowing that vitamin D3 is the kit that is there in the market is really expensive, somewhere around 3,000. So we're just trying to make it very affordable. It will cost around 500 something. And then we are also developing a COVID-19 detection kit. And all these kits are DNA aptasensor based kits. So we develop DNA aptamers, we develop these DNA molecules which bind to our target. And then we convert these aptamers into aptasensors or into biosensors. And this conversion, we generally, right now, we are using gold nanoparticles. We'll be telling you that in the lab session also. Can you go on to the next slide? So let's see how these aptamers are made. So I have tried to make the rest of the part of this presentation very simple for any person who's not from the biology background to understand it and to make it interesting. Let's see if I have done a right job. Let's move to the next slide, please. So yeah, so this figure, I don't think so it looks complicated, but I'll just tell you, understand this way. You have a library, right? Say, for example, you have a library of DNA molecules, meaning that you have 10 to the power 15 DNA molecules, right? And these DNA molecules, you don't know what these molecules are. All you know is that all these DNA molecules have a similar size, say, for example, 70 bases, right? And the sequence of this DNA, the way in which these monomers are arranged, you don't know that. You just know that it is a library of DNA molecule. Now, among that library, you want to know which one of these library members, which one of these unique DNA members will bind specifically to your target. This is kind of something when you go in a book library and you have taken 2 or 3 kids with you, and one kid says, I only want a book which deals with fighter planes, right? And I don't want anything else. And the other kid says, I only want a book that is something on Avengers, nothing else, right? And you don't know that among that 1,000 books in the library, which one is a Avenger book and which one is a what was the other one I said? So the phytoplane book is, right? Um, so you take all these books and start giving it to the kids, right? They know what they want, so they will specifically point out that book. I want that book that deals with Avengers. So he just sees the cover of the book and he says, yeah, that's an Avenger book. I want that book. So you will take that book and go away. So that is how you are going to hunt DNA aptamers which bind specifically to your target. So that is how this entire aptamer are made in the laboratory, and this procedure is called as systemic evolution of ligands by exponential enrichment, or in short we call it SELEX, right? So we take a DNA library molecules, you see there are a lot of colored molecules here, right? So these are all DNA library aptamer molecules, and then you incubate these molecules with your target. Like I said, your target could be anything. So the molecules which bind specifically to your target, so in this huge sea of DNA, there might be few thousand sample the DNA molecules which will bind to your target. Okay, and then what you do, so you let this interaction happen in a solution, in a buffer solution, where all these DNA molecules will go and attach to your target. Okay, and then all the unbound molecules, you need to get rid of them. So you just throw all the unbound molecule library members, right? But here is the thing. So when you have done this interaction, DNA molecules, they are not like books, right? That okay, yeah, this is the book and the kid can take the book. This molecule is very small, right? So you don't know which molecules are binding to your target, right? So you need to, and there are 10 to the power 15 molecules. By that I mean that there is one molecule, just as there are all single molecules, right? So whatever is binding to your target, they are the single molecules that are binding to the target. You need to enrich these molecules. You need to make thousands and millions of copies of this selected DNA molecules. And how you do that? You use a simple technique for amplifying these DNA molecules. You do polymerase chain reaction, or PCR, right? If you have heard of it. The PCR is a simple technique in which one DNA molecule is made into many copies of the same DNA molecule, and these copies range from from 1,000 molecules to million molecules, and they're all same. So that's what you do in this next step of the selects, right? So once you have multiple copies of the selected DNA molecules, so you again use these DNA molecules. So when you do PCRs, all your DNA molecules become this classic double helix DNA molecule. But when I said DNA aptamers, they're always a single stranded DNA. So you're done with a PCR, and then you convert these double stranded DNA molecules into single stranded DNA molecules. There are techniques to do that. We'll be detailing you that technique in the lab demonstration, right? So you convert them into single-stranded DNA molecules, and then you again incubate with your same target. You keep on doing this cycling for like 10 to 12 cycles. And why we do this iterations? Because we want, in the end of the SELEX procedure, we want those molecules, selected molecules, which are the best binders to your targets, right? So that is the sole aim of doing this iteration. Can we move on the next slide? So once you are done with all the SELEX procedure, say 10 to 12 cycles, you come up with few thousands of DNA molecules which are uniquely identifying your target molecule. So when I say few thousand, that's still a huge number. So among those few thousand, so you come from 10 to the power 15 molecules to few thousand, and this few thousand is from 1,000 to 2,000, no more than that. But still, fishing that aptamer, which is the best binder to your target molecule, is still a ginormous task, right? So before experimentally testing, you cannot test 1,000 molecules experimentally one-to-one with your ligand. That's logistically impossible. So what do? We have a algorithm. It is a So before moving to that, you need to know the sequence of this DNA, meaning you need to know what is the arrangement of the nitrogenous bases or the nucleotides on that selected few thousands of DNA molecules, right? For that, you use a technique which is called as next generation sequencing. It is simple. It is a simple method by which you know the sequence of the DNA, how these 4 nitrogenous bases are arranged on the selected DNA aptamers, right? So, and this actually, so generally we move when we keep on selecting these DNA molecules, this output is generally from 200 to lesser is good, because if there are less molecule from this sequencing output, which means your selection is really very good. If there are more molecules, it's not that your selection is bad, but still you have a lot of diverse space, right? Which you really have to hunt which one are the best binders. So anyway, when we do NGS, we get output from around 200 to 500 unique DNA aptamer sequences, which are coming from your selects procedure. So anyway, we are down from 10 to the power 15 molecules to say 1000 molecules, right? Can you move to the next slide? So like I said, we cannot test actually these 1000 molecules with your ligand. So what we do is, before going for experimental testing, we do a little bit of machine learning algorithm. And so we have this AI and ML algorithms which actually do the aptamer candidate optimization, meaning they from that NGS data of say about 1000 or 500 molecule selected DNA molecules, we need to come down to 20 such candidates which are the best binders to your target. And the parameters that on this we do the selection ab initio, it is structural stability, how stable is your DNA molecule, then your binding affinity, how with what affinity this molecule is binding to your target, what is the specificity of binding, and and what is the conformation. So all these things are taken into account for selection of the candidates which actually bind to your molecule. So once we are down to somewhere around 20 DNA aptamers, then we test these 20 DNA aptamers. We synthesize these DNA aptamers in real, and then we test individually for binding to its target, right? Can you move on the next slide? So this is the last step of entire aptamer candidate optimization procedure. So we take those 20 candidates as a result of those AI applications, and then we individually synthesize them from any commercial facility, and then we test this binding experimentally or in the wet lab. So, and we test on specificity, we test on affinity, we test on how stable are these molecules in solution, and when they are bounded to your ligand, if these biomolecule which we are testing, if it is found in urine or in serum also, we do all these things. First we do this binding in a buffer solution, then we do the actual binding in the biological fluid, serum, urine, or whatever it is. And we see that how stable is this binding is in all these fluids, and how well they are binding, what is the time they are taking to form the best conformation or the best complex. So all these things we keep on assessing before we come down to the best binder. So at the end of the entire procedure, we come up with 2 or 3 best binding DNA aptamers, which we finally take it for the apta sensor development, or they are finally taken up for the next step, which is incorporating them with the gold nanoparticles, right? So can you move to the next slide? So these best candidates are then taken. Now we have to develop sensors, right? So we develop apta sensors. So what we do is we conjugate these DNA aptamer molecules with the gold nanoparticles. We see, I'm giving this example. You can, I'll be showing you a few of the examples from different workers around the world. This incorporation can be not just gold nanoparticles. You can do with nanofiber, any kind of nanomaterial. Right, so so there are generally 2 methods for this DNA, for forming this DNA aptamer and gold nanoparticle conjugates. The first is your physical absorption. You don't use any kind of so the attachment of DNA on a gold nanoparticle, it is not covalent. you just incubate it in some buffer conditions, and then these are physically adsorbed. And sorry, so once this DNA and gold nanoparticle conjugate is incubated with your target, these DNA molecules, they actually leave nanoparticle and they bind to their target, and these nanoparticle, on the addition of some kind of salt, NaCl, for example, they aggregate and the color change happens. One is another, you can bind these aptamers covalently on the surface of these gold nanoparticles, and that is generally done. We use thiolated DNA aptamers. We attach thiol groups at the 5' end of this DNA molecule, and then this interaction is done. This is the covalent interaction, and when the ligand comes, it is a change of they bind to the ligand, and there is a change in the UV spectra, which is observed by your any kind of detector device. Right? Can you move on the next slide? So I have taken few examples before actually telling you what we do in our lab or in our company. So I have pulled few examples at how these DNA aptamer molecules, or how these DNA as such molecules, they are integrated with any electronics applications or with nanotechnology. So I've taken this one. This is basically an Ebola detection biosensor, and this is already there in the market. It has been not extensively, but yeah, it is actually doing some clinical trials in Africa. So they have taken these screen printed electrode, and on this they have used thiolated DNA aptamer molecules, right? And this DNA aptamer molecule, which is actually detects the DNA of the Ebola virus, and this hybridization. You can see this fuchsia colored another helix. So this is basically an Ebola genomic DNA. So again, Ebola is an RNA virus like the coronavirus. So what they do is they break the Ebola viruses, take its RNA out, and this hybridization is basically so they convert the RNA into DNA. Just don't go into that. What this pink part is your Ebola DNA, right? So which is detected by your DNA apta sensor. So these DNA aptamers, they basically hybridize like this way with your Ebola, right? And that is what it gives. This hybridization, it gives signal, and all these chemical reactions takes place, which is amplified by your electrodes, and you get a quantitative reading. Okay, can you move the next example? And this is another example that I pulled out. This they have used this DNA apta sensor in this. So what they do is they have taken this again. They have taken thiol labeled DNA aptamer, like I told you. And so if you know the DNA structure, it has two parts. One is a 5' end, one is the 3' end. Why they are called so? This prime is basically the number of carbon on the ribose sugar. So one end is always the 5' of the ribose sugar, and the other part of the DNA chain is the 3' of the ribose sugar, whereas the incoming next monomer attaches, right? So at the 5' end, so they have thiolated the 3' end, and at the 5' end, if it is not binding anything, it is in close proximity with this methylene blue, right? So this is kind of a reaction which takes place when there is a ligand. What happens is proximity breaks down, so there will be a little, there will be a difference in the electron transfer. And so what I want to let you know is, there are in many ways you can manipulate this DNA molecule, and you can develop any kind of sensor using any kind of applications, any kind of nanomaterial, any kind of electrosensors. And that is the beauty. You cannot do all these things with a protein molecule, right? And you can modify this DNA molecule in any way you like. And whatever way you modify, it's not going to affect much on its specificity or binding specificity to its target molecule, right? Can you move on the next slide? So this is another example I have pulled out. So they have used, again, they have used screen printed electrode, and this is a biomarker. This is an aptamer molecule, which is specific to a cardiac biomarker, which is BNP32. And this is just, again, another example which you can, so they have taken, so these gold, they amplify the signal. once this aptamer binds here, or when it gets to it, this is your aptamer molecule, and if it gets this biomarker, which is your cardiac biomarker, there's a change in the redox couple, and that is test. So this binding is always very specific, and when your cardiac biomarker is not there, if it is not present in your, or it is present in a low amount in your serum, you don't you get different kind of signal, and when it is present, you just get a quantitative signal out of it. And one another thing with this DNA molecule is that all the protein, if your test is protein based, if you're using antibodies, so they are very much thermostable, right? Which meaning they need a particular temperature for transit. And so, in a very popular terms, you say that all these kits, whether it is ELISA kit or any kind of a kit which requires antibodies, they require cold chain, because from transiting from one place to another place, they need a particular 4-degree temperature for, and for storage also. If you are not using that kit, and if it is not point of care, and you need to store these kits somewhere, right? And the storage temperature cannot be more. It should be stored at a particular temperature, at a cold temperature, say about 8 degrees or 4 degrees. All these things are not required for DNA molecules. DNA molecules are very thermostable molecules. So that is why for transit also, for storage also, they don't require any cold chain. And DNA molecules are very inert molecules, right? And so that actually increases their shelf life. In the case of protein molecules, in case of antibodies, your diagnostic kits, they cannot long more than maximum 2 years. While if you're using DNA molecules, any kind of so these DNA molecules basically can be stored for like 20, 30, or even 40 years. When you're making it in the form of a kit, the shelf life is generally, it's somewhere around 7-8 years, because after that, your buffer conditions change and a lot of other things happen. So that is even more than 2 or 3 years in the case of protein molecules. Can we go on the next slide? So this is what we are doing right now. So among many other kits that we are developing, this is just an example what we are doing in our lab. So this is we are doing with IIT Guwahati with Dr. Tapas, and we are doing it with Dr. Partho also. So we are developing these DNA aptamer molecules which bind to the spike protein of the coronavirus. We already have the spike protein in our lab. We don't use live viruses because you cannot, but we have the spike protein. So we develop aptamers which bind specifically to the spike protein. And then this is what we do in our lab. And then IIT Guwahati, what they do is they convert these aptamers into aptasensors. So what generally in this case, we are using thiolated aptamers. And so, when there is a coronavirus, if it is present in the sample, this will bind specifically to it. There will be a color change. And when there is no target, they won't bind, there will be no color change. And this color change is then amplified. And it is, it is read in the form of electrical signal. And we have a device in place that we have collaborated with another startup, which is based in IIT, which is Primary Health Tech. So they already have a device in place where this device can be attached to your smartphones, and we can get the reading, right? Can you move on the next slide? So this is my team. That's me, and so this is the core team, and then we have a set of advisors also. So Moshmi is the lead researcher in our team, and Sahil. So Sahil has another startup, Primary Health Tech, which is also based in IIT Guwahati. So he's basically engineer. So he develops the device part that I was talking about. Then Dr. Ranadeep Gogoi, he's a scientist at NEMS right now, and he basically coordinates all the scientific biological part of our startup. And then we have a set of advisors. Dr. Srinath, he is basically a business advisor to us, and Dr. Mohan, and we have collaboration with IIT Guwahati and a few other institutes, which, we keep on for, the part we are biologists, we need engineers, we need chemists who actually, so when you meet all these three people, we can come up with very nice and very path-breaking kind of a diagnostic. So that is what we're trying to do still trying. Can you move on the next slide? So that's it. If you have any questions, this is our contact details. And anyway, I am ready to take up questions now.
Thank you, ma'am, for your valuable talk. So our first question is from Devasri Devasmita. Her question is how to select the aptamer library. Is it commercially available or do we have to isolate it from some source? If so, then
Okay,
kindly
yeah,
explain the process.
That's a very nice question. So no, Debashri, this library, they are commercially available. What you need to tell those commercial people is that you need to tell them that, so when I say PCR, when you're doing PCR in one of the steps, so for PCRs, you need to know the part of the DNA molecule for which you design your primers. So what you tell the library, people who actually develop these library molecules, if you can go back to, I think, Moshmi, can you go back on the slide? Okay, they are playing the, just, can you open the presentation? I'll just explain the library thing. So what these commercial people, they do is you give a little sequence of the primer, say about 15 bases, you tell the sequence, right? And they make this 15 bases sequence, they attach to it a matrix, and then they randomize, meaning they keep on adding nucleotides without knowing which nucleotide they are adding, right? And then they randomize this entire part. Okay, yeah, can you go back like in the beginning? More when I was explaining. So yeah, go back. Yeah. No. Move forward after. Yeah, that's beyond this. So if you see this image in which I have written 5' PBS, then this 40 more randomized and aptamer sequence, and this 3' PBS. PBS is basically primer binding site. Now, why you need this? Because when you are doing PCRs, you need this a little sequence which is somewhere around 15 to 18 bases. You need to know the sequence where your primer gets attached and when you do the PCR. This entire 40 bases, you don't know that sequence. And so what they do is they keep on adding nucleotides without any order. They let this randomization happening. So that is why you come up with this huge space of 10 to the power 15 molecules. So you have all these molecules which have a little part 5' end and at the 3' end whose sequence are known to you because you have given that sequence. This 40 part is unknown to you, right? So these are basically all commercial libraries. You can make this library in-house also. We have a patented method of doing this where we use an enzyme called terminal transferase. So this enzyme, it also adds nucleotide without any specific manner. It just keeps on adding enzymes, right? Nucleotides. You can do that at your in your lab also. But this is actually a cumbersome process because once you make this molecule, you have to purify them, and you have to go on those HPLCs to get pure preparations of your DNA, which is a bit cumbersome. So most of the time, almost all of the time, we opt for the commercial library. Does that satisfy your answer? Query? Okay, I can see one another question. Can aptamer-based detection be used for cancer detection? Of course. In fact, there are a few of the kits which are in the clinical developmental stage. One kit is, I think, in Germany. So they are developing a kit. So like I said, when I was telling you what are the targets against which these aptamer can be developed, one such target was the mammalian cells. So when you are, in fact, in our lab, when I was working in NIPER Guwahati, and so we have a lab session in which your resource person, Shravan, so he's the guy who actually developed these aptamer molecules against cancer cells, meaning that these aptamer molecules, they bind specifically or identify specifically a particular type of cancer, right? So of course, you can use this. They are already in clinical trials in Germany. So they have taken, and you can find huge literature in PubMed also where they have developed these aptamer. In fact, the first cell selects that procedure was optimized, that was optimized using cancer cells. So they take cancer cells and then they do incubation steps where they develop DNA aptamers which can specifically bind to your Because cancer cells, what happens is they develop some kind of receptors which are actually not present in your normal cells. And that is what is identified by these aptamers. So that is what these aptamers, they only bind to your cancer cell. They don't bind to your neighboring, other kinds of normal cells. And if this things, if this happens, and you can develop a very advanced sensor, you don't need to use biopsies. And that is the main aim of cancer diagnostics these days, that you need to bypass the biopsy step for identification of cancer, right? Any other question?
Yes, ma'am. One more question is there.
What is
how does an aptamer bind to a protein or metal ion? It is a nucleic acid sequence. So how can a nucleic acid bind to an amino acid or metal ion?
Yeah, so this binding is never covalent, and like I said, when it is single-stranded, they are in a form of a conformation, right? So this conformation, and when there is any ligand is there, any ligand, so the interaction is always electrostatic interaction. So that is why not every aptamer can bind to any given target. So if your metabolite is, for example, vitamin D, right? So which is not or any protein molecule or any enzyme molecule. So this interaction is always electrostatic interaction that totally depends upon the arrangement of the conformation of your. So that is always electrostatic, any interaction depending upon the conformation of the your aptamers. Okay, I think I have one more question from Simranjeet. Are these sensors reusable? No, they are not reusable, meaning, and you don't really need to reuse them because, so we can have a reversible binding, but then to break this binding, you need to again, okay, so you understand in this way. So you have your gold nanoparticle attached to your aptamers, and then they bind to your ligand, and this interaction is, even though it is not covalent, this is a good interaction, right? And you want to break this interaction, you have to heat it, right? So anyway, when you heat it, your protein molecule or the ligand molecule will denature, or so there are many other steps are involved here, right? If you heat it or if you break this interaction, you have to get rid of the ligand molecule, which is again a cumbersome procedure. DNA molecules are not that much expensive to make, right? When we come on making these DNA molecules in a kit, when you put for commercial, for synthesis of the DNA, per base cost is generally 20 to 30 rupees per base cost. And you need very less amount of DNA on when you are doing these kind of binding studies, not huge. So we need huge DNA when we are optimizing some kind of experiments or in the binding optimization or kit development procedure. Once you're actually using it, you don't need that much of DNA, and DNA price, the synthesis of DNA price is way more cheaper and way less cumbersome as compared to the antibodies. Antibodies are expensive and they're always made in animal system. You don't need an animal system in the case of DNA molecules. So we don't really need to reuse them, right? So that is why we can, theoretically, we can reuse them, but then you have to break the interaction, get rid of all the junk, and then again attach it to the gold nanoparticles and then again use it. Okay, I have one more question. What if more than one aptamer binds to the same target? That we check always, right? So that's an excellent question. That is how we decide the specificity of these DNA aptamer molecules. So once we are done, remember I was showing you the various steps of the selects, in which when we are done with the selects, we know the sequence, we do all those machine learning algorithms for candidate optimization, and lastly we do the experimental specificity of these aptamers. And in that step, we take a range of, say, I'll tell you in the form of example, suppose you are detecting something, a biomolecule which is found in a serum, right? Some biomolecule X, and you have developed this aptamer binding this X biomolecule. When you're actually testing this aptamer for the aptasensor development, you take this DNA aptamer molecule and put in the serum, and you detect, is this aptamer is binding to any other metabolite that is present in the serum. If it binds, we absolutely never move that aptamer forward for aptar sensor development, which means that this aptamer is non-specific binder, right? So we do all these steps before we actually develop these aptamer molecules in the form of aptar sensors. We make sure that it doesn't. It can bind, it always binds, but then we need to make sure that it is not binding. So that is what we do a lot of counter selections. So when we are doing the select procedure also, I have shown this image, if you see in the slide, this is a very simplified select procedure, right? In this procedure, when we are doing these iteration rounds, we do a lot of counter selections, meaning we take these selected DNA aptamer molecules and we incubate with non-targets, similar non-targets. Like if we are using spike protein of the COVID-19, we use the spike protein which is expressed on the SARS virus, on the MERS virus, which are similar in structure. We use all the spike proteins from these viruses also during the select procedure. We do counter selection and we make sure that this DNA molecules of which we are selecting, they are not binding to the spike proteins of any other viruses, right? So that is what we do candidate optimization before we actually move on to the kit development procedure. Okay, and I have one more question. Is aptamer replace antibody conjugation here? So in this, what I have explained you just now, we don't need any antibodies. And I don't really understand it. So you mean that if we can replace these antibody or we can use this antibody conjugated to gold nanoparticles, is that what you want to ask? This question is from Himadri. You can conjugate antibodies with gold nanoparticles. In that case, so that is, see, there are two types of methods. One is using antibodies, the other is using aptamers, right? So of course you can conjugate antibodies with your gold nanoparticles and you can develop any kind of sensor in that way also. That always you can do. So this is what I say, they're kind of two divergent paths. One is with the DNA molecule, the other one is with your antibody molecule. Okay, if that satisfies your query. Do we have any more questions? Okay. So I think we move to the lab demo, and I have some other engagement right now. So I'll leave you now with my team members. So we have in the audience, we have Shravan with us, who is the main resource person, and he's actually, like I told you, he's the main guy who actually developed DNA aptamers a lot, and who actually with me have optimized the select procedure in the lab. And then we have Maushami with us. She is the project assistant in my company, and she will walk you through the detection procedure. And so I'll just leave you with these two guys now, and thank you for your attention.
So we will be telling you about the procedure that I've already told you in my talk about the select procedure. So that is systemic evolution of ligands by exponential enrichment. So since you guys, what I know is from the chemistry and electronics background, so I'll be telling you, explaining you what the procedure is. I'll be telling you the sequential steps that you do in the lab to develop DNA aptar sensors. Or so there are two parts. In one part, we develop DNA aptamers or the DNA molecules. And the second part, we convert those DNA aptamer molecules into aptar sensors or biosensors. Okay, and then these biosensors can be integrated to any of the electronics device, which will read your parameters that you're going to know with using those aptar sensors. Okay, so to start with, I'll just outline the procedures. So easily, how will you understand this procedure? We have one ligand. Okay, and then so our procedure is target molecule or target cell against which you have to search the DNA molecules with identify and bind to this ligand with high specificity and high affinity. So it's the same principle as you might know the antibodies and the protein interaction works, or antibody antigen interactions. The difference is here the antibody molecule is the protein molecule, but in Cellex these recognition molecule are the DNA molecules. Okay, so how we are going to search this, and since antibody selection and antibody production is a biological process, so that takes place inside your body, but here we are going to do everything like the searching for those DNA molecules which bind to your target molecule with high affinity. We are going to do that procedure entirely in the lab. Okay. So for that we use a library which is your DNA library. Okay. Now what is this DNA library? Consider there are 10 to the power 15 unique DNA molecules whose sequence, so DNA molecules, the students who have some background on chemistry, they might be knowing that DNA is a macromolecule. It's a polypeptide, it's a macromolecule, it's a polymer. And what is this polymer? It is made up of 4 nitrogenous bases, A, T, G, and C, or adenine, thymine, guanine, and cytosine. Okay? So these are the 4 nitrogenous bases of which any nucleic acid molecule is made of. Now, so this DNA molecule, the sequence is all what gives any DNA molecule its unique conformation and specificity, affinity, or any of the DNA properties, right? So just consider this DNA library which has these many unique molecules, meaning they have different the way these 4 nitrogenous bases are arranged. Okay, so there are these many huge molecules with different sequence, and we want to know among these 10 to the power 15 molecules who are the best binders which are identifying and binding to this target molecule, or which is our target ligand. Okay, so that is what the entire Cellex procedure is all about. So the first step what we do is we incubate this DNA library with the ligand molecule, and we let these two things interact with each other. So in the first step, what you will get after this incubation, this is your ligand. So this is your ligand. Today, the ligand which we are selecting is the bacterial cell, right? The bacterial cell which we are using today to show you is E. coli or simple Escherichia coli. That is the lab bacterial molecule that we use. This is the bacterial cell, E. coli molecule. And after the first step in which you are incubating this DNA library with this bacterial molecule, among those, remember 10 to the power 15 molecules, you will get some 100 molecules which will attach to this bacteria molecule, bacterial cell. Okay. So they will attach like this. Right. So among those 10 to the power 15 molecules, here are few of the 100 molecules that will finally attach. Now, these are those molecules which are identifying this bacterial cell in a very unique manner. Okay. Now, you want to take out these single-stranded DNA or these DNA aptamer molecules. So that is our next step, in which we just isolate only those DNA molecules which are identifying your ligand, or in this case, the bacterial cell. How we do that? This is very simple. This interaction is not covalent interaction. Okay, this is simple binding which is happening by electrostatic interactions. Okay. So the best way to break this binding is to heat it. Okay. So that is the next step, in which we collect these single stranded DNA aptamer molecules by simply heating it. When you heat it, this interaction will break. Next step, so we have to break this interaction, which is simply you heat this suspension. So when you heat it, you'll get rid of this bacterial cell, okay? And all you are left with is those DNA aptamer molecules which are binding to your bacterial cell. So if you convert this in form of a tube, so we have this tube, and then you have, so what you do in the first step, you have these many E. coli cells, and you have this DNA molecules attached to it, okay? And you heat it, and then you centrifuge it, spin it. So what will happen? All these cells will settle in the bottom of this tube, like this, and all you are left with in the solution are those DNA aptamer molecules which you have selected. Okay. Now, remember I told you there are 10 to the power 15 molecules, and DNA molecules are very small molecules, even though they are polymers and they are macromolecules, but you cannot see them with your naked eyes, right? And these are single molecules. Okay. So identifying one single molecule among a sea of DNA molecules is logistically impossible. So what you do, you need to enrich these molecules, meaning you need several copies, several 100 million copies of these selected DNA aptamer. Okay, so that is our next step, in which we amplify or make hundreds and thousand copies of one single aptamers which are selected, and that you do by PCR. Okay, so for people who don't know what PCR is, PCR is your polymerase chain reaction. Okay, so it's a chain reaction in which this DNA molecules gets amplified into as many copies as you want. So this reaction is basically a exponential reaction. So if you put it in a curve, what will happen? This is your starting DNA, which is say about 10 nanograms. Okay, or in DNA molecule number, if you say, if we are considering one DNA aptamer molecule, this is one molecule. Okay, then after this reaction takes place, it's a chain reaction. This will go like this, and the curve will go like this, and this is that phase in which all your ingredients which are there in your PCR reaction, they are used up. So it's kind of a stationary or dead phase, and this is the phase in which this one molecule gets divided in each cycle. So generally you put it for 10 to 15 cycle of amplification for DNA aptamers. So this will result into like somewhere around 10 to the power 10 molecules. So this is how one single DNA molecule you can get this many yield. Okay. So now there's enough DNA for you to select these DNA for the subsequent round of select. So I told you in my talk that select is a cyclic, it's iterative procedure. There are a lot of iteration rounds which happen. You select some DNA molecules and then you reiterate the entire procedure to get the best and the high affinity binders. So this is your next step. So once, now there is one catch here. When you do PCR, so you might be knowing, some of you, what PCR is. So just not to complicate things, like I told you in the beginning, PCR is a reaction by which your DNA molecules get amplified, right? Now when you amplify these DNA molecules, they become double-stranded. Now what is this strand thing is? So you must have seen all those glamorous picture of DNA that this is always double-stranded. This is the image that you generally see everywhere, you know, in movies, in t-shirts and whatever. This is double-stranded, right? So generally the orientation is, for the chemistry people, this 5' to 3', and this is 5' to 3', like this. What are these prime numbers? These are basically, so DNA molecules is made of 3 moieties. One is your ribose sugar, one is your phosphate molecule, and one is your nitrogenous bases. Okay. Now these numbers are the numbers of the ribose sugar. Okay. So 3' is the third carbon of this ribose moiety, and 5' is the fifth carbon of this ribose sugar. And the orientation of this macromolecule is always 5' to 3'. Okay. And the second strand, which so DNA, that is why there are 2 strands of DNA attached to each other. So that is why it is called as a double helix. Okay. And the second strand is the anti-parallel strand, we call it, and that's an opposite orientation. It is from 5' to 3'. So this is what it makes a classic structure of the DNA. Now when you do the PCR, so aptamer molecule, I told you already in my talk that these aptamer molecules are the single-stranded DNA, or ssDNA we call it. ssDNA. So they are single-strand DNA molecule. This is in this orientation. Okay. Now when you do this PCR, this will become double-stranded. So and remember, SELECT is an iterative process. We need to do the PCR, enrich our binders, and then re-incubate it with your ligand molecule or the target molecule. Now, target molecule incubation, again, when you do it again and again, it can always be done with the single-stranded DNA molecule, not the double-stranded. Okay. So that is how your second step is. You do the PCR, you get the double-stranded molecule. Now you have to convert this double-stranded molecule into single-stranded DNA molecules so that you can do subsequent rounds of SELECT procedure. So how you do it? So when you do PCR, just not to complicate and bore you, this is your template molecule or that molecule which needs to be amplified. Okay. So here you use one enzyme which is called as DNA polymerase. This enzyme just adds nucleotides, or we call it DNTPs. These are the building blocks or the monomers of any DNA molecule. So this enzyme, what it does, it polymerizes these monomers into a polymer, which is your DNA molecule. This DNA polymerase enzyme, it uses a primer molecule. Okay. This primer molecule, we simply call it primer. It cannot work without this. Why am I telling you this? When we use this primer molecule is labeled with a protein which is called as biotin. Now, the chemistry people are, just for the sake of this procedure, biotin is a protein molecule which binds with another molecule which is called as avidin. And this interaction is one of the most strong interaction found anywhere in the biological system. This is a huge high affinity binding. And the KD values are generally very good values because they're the highest affinity bindings found in any biological system. So your primer molecule is labeled with a biotin molecule here. Okay? And then you use magnetic beads. These are magnetic beads. They are of, I think, 10 micron sizes magnetic beads. And these magnetic beads are coated with the avidin molecule, or we generally call it streptavidin. Okay, so you have these magnetic beads coated with streptavidin. Once you do the PCR, you load this double-stranded DNA on these beads. Okay, so just see this. If this this is quite easy to understand. So this is a primer and this is labeled with biotin. When PCR will happen, your DNA polymerase will make the second strand. Okay, like this. So this second strand has a biotin and the first strand doesn't have a biotin. You load this double-stranded DNA on this streptavidin magnetic bead. So they will load like this. So this biotin molecule will attach to the avidin molecule. Okay, and your DNA molecule will be like this. like this, is labeled with biotin, and it is attached to the streptavidin of the magnetic bead. And the other strand is just like that because of the DNA double helix, hydrogen interaction. It is forming double stranded with your DNA. Now what you do, you simply put it on a magnet. So this will be separated, and then you simply add salt molecule, high alkaline concentration, which is you add NaOH, right? So you add NaOH, what will it do? It will break the hydrogen interaction between these two strands, right? And then you take out, separate this magnetic bead on the magnet. So what will happen? And then simply this reaction. So we will show you how this thing takes place in the solution. So just imagine that this is your tube, okay? And this is your magnetic beads. And here is your DNA attached to it, like this, okay? And these two strands, they are attached with your hydrogen bonds. That is how this double-stranded DNA is formed, okay? And now what you do is simply you put it on a magnet. These are magnetic beads. So when you put on a magnet, what will happen? We will show you how this takes place. So what will happen? All this magnet will be attached like here, and your DNA molecules will be like this, right? And now you have to separate that DNA strand which is not labeled with biotin. So what you do, you just have to break the hydrogen bonds between the double-stranded DNA. So what you do, you just add a alkali. So you add simple alkali, which will break the interaction, break the hydrogen bonds. So in the solution, what will happen? These double-stranded DNA now will become single-stranded. Okay, and this is attached to the magnet, that biotin label, and your single strand will come in the solution. You just simply pipette out the solution in a fresh tube, and then all you get is, you remove this. In a fresh tube, you will get all single-stranded DNA aptamers, which you have selected against your ligand. Now, so this is the entire one cycle of selects, and what you do is you again incubate these selected DNA aptamer molecules again with the target molecules, and you repeat these procedures, repeat these cycling for around 10 to 12 rounds. Keep on doing this, okay? So why we do this again and again? Because we want to be sure that whatever DNA molecules which we are getting, they are the high affinity and high specific binders against your target, right? These are molecular recognition molecules, so they recognize your target with high specificity and high affinity after these many rounds. That is why we do these iterations round again and again. So once you are done with these rounds, so up till this part, how you do one cycle of selects, we will be demonstrating in the lab session. What you do after that, I think I have detailed that in my lecture already. So then these DNA aptamers, when you are done with selects, you know the sequence of the DNA. I have already explained you that. So you develop that now into apta sensors or biosensors. So what you do, you load these DNA aptamer molecules on gold nanoparticles. Because now you want to use these for diagnostic purposes, say for example. So that is what our company does. We develop apta sensors. We make DNA aptamers and then we develop them into apta sensors, and then we use for diagnostic purposes. So now these DNA molecules, they are now loaded on the gold nanoparticles. Okay, and this loading is generally physical absorption. This is physical interaction. So what we do is we take these gold nanoparticles and then we incubate them with the aptamer molecules, right? And it's a reaction, I think it takes place around 5 to 6 hours. And then these DNA aptamer molecules, they get attached to this gold nanoparticles, okay? And then, so this interaction is, like I told you, this is a physical absorption. You can also have this interaction covalent. In that case, your DNA aptamer molecules, they attach covalently to your gold nanoparticles. And for that, what you do is you modify one end of this DNA aptamer molecule and you attach a thiol group here. Okay, and this thiol group then you use for covalent interaction with the gold nanoparticles. So we won't be doing that today. We generally use physical absorption method. So we also do thiol, but anyway, so today we are not going to do that. So we just absorb this DNA aptamer molecules over these gold nanoparticles. Now these aptar sensors are ready to sense your target ligand, cell, or whatever against which these aptamers are designed. Right, so what we do is now we take these gold nanoparticles and then, so that's just this huge image. So these are your gold nanoparticles with your DNA attached to it. Let's say AUNPs. Okay, my handwriting is really bad, but anyway, bear with it. So now we do, we put this E. coli cells now, because now they have to sense it, right? And so we have one control in which we have just water and no cells, right? So generally these AUNPs attached with DNA, so we call it DNA AUNP conjugates, they are in wine red color, okay? So now we let this interaction happen. So this is around a 30-minute reaction. So we have a proprietary buffer in which this reaction takes place. So, and we let these two interact. So what will happen? These molecules will go and attach here, right, like this. They will leave the gold nanoparticle and these DNA molecules. So this is physical absorption, right? So this DNA, as soon as they get their target ligand, these DNA molecules will go and attach here, and the gold will be separated. Here in this control one, no such interaction will take place. Now you add some salt, NaCl. So in the presence of this salt, these gold nanoparticles will aggregate. Okay, and here no such aggregation will take place because of the presence of these DNA molecules. They won't aggregate. So once they aggregate, the color changes into purple for these sample and not for this. So that is your indicator that your aptar sensor is working and it is identifying your target DNA, any target, which could be a cell, which could be a virus, which could be any protein molecule, any organic molecule that I've already detailed in my talk. Okay, so this is what we are going to show you today, step by step. Okay, and now I just want to introduce the team members or the resource persons who will be telling you how to do the entire procedure. These three will be your resource people today. They will be demonstrating the various steps what I have just explained. She's Moshmi. She's a project assistant in my company. And he's Kundal. He's another scientist in our company, and that is Shravan. He's the main resource person, and he's very instrumental when we were optimizing this select procedure. He was the guy who was actually doing all those steps. So he's the main resource person today. So say hi to them, and then over to them. Okay. So now I leave you guys with these three people.
Here we are going to make aptamers against E. coli. So our target is E. coli cells. So we are taking here overnight culture of E. coli cells. We are making aptamers against E. coli cells, and we are doing a bacterial culture group. In that video, I am taking bacterial cell culture, that is overnight grown. After the collection of 1 ml of culture, then we have to centrifuge it at 6000 rpm for 5 minutes. In the centrifugation step, we will get the pellet of E. coli cells. We can do it normal room temperature. This is centrifugation at 6000 rpm for 5 minutes. After the centrifugation step, we will get the pellet and we have to discard the supernatant, and we have to wash this pellet, bacterial culture pellet, for the removal of any metabolites or excess of media. Here I am taking phosphate wafer saline, 7.4 pH. Here you can see the pellet and upper the supernatant. Just we have to remove that supernatant and we have to wash that pellet with the PBS. Again, we have to centrifuge it 6000 rpm for 5 minutes. Then we have to, after the centrifugation, we have to remove the supernatant. To the bacterial culture pellet, we have to add library. In that library, we have the molecules of 10 to the power of 15 different type of molecules. That is single stranded DNA molecules. That is around 10 to the power of 15 molecules. These are the library molecules. This library we can make in a lab, and commercially we can also order it based on our design. Already MEMS explained in that video. In the time mixing the pellet with the library molecules. In that we have to add that library molecule in the binding buffer. We have to use here binding buffer with the library molecules. After mixing those pellet with the library molecules, then we have to incubate at room temperature at 45 minutes and gently rocking. For 45 minutes, we have to incubate and shake them at room temperature. After the completion of incubation step, we have to heat these library bound molecules with the cells. We have to heat at 95 degrees centigrade for 10 minutes. Then after, we have to centrifuge again. In entire procedure, we are using same centrifugation that is 6000 rpm and for 5 minutes. In the centrifugation step, what will happen? In heating step, that cell bound library molecules will be separated. And in centrifugation step, what will happen is that cell debris and cell will be go down to the bottom of the tube. And in the supernatant, we will collect single cell molecules, that is single stranded DNA molecules. Without disturbing the pellet, we have to collect that supernatant carefully. In that supernatant, we can get that molecule, single stranded DNA molecules. The single stranded DNA molecules we will use for the PCR, for the amplification in the further steps. Here in this PCR, we will use two primers, that is forward primer and a reverse primer. In this reverse primer, we will use a biotin primer. We are using 10 to 15 cycles in the PCR. Here we will use so many buffers: PCR, tag DNA polymerase, DNA premix, magnesium, and primers, and DNA templates. The template is single strand DNA molecule. This is the composition of PCR, and we will set that cycle. In this, three are there: denaturation, annealing, and extinction. These steps are there in PCR. In 10 to 15 cycles, that our single strand DNA molecule will be get amplified. After the PCR, what we will do, we will add NaOH buffer to that double stranded molecules. In that PCR, that single stranded going to be double stranded. We will bake those molecule bond with the NaOH. We will hydrolyze those bonds, hydrogen bonds, and then for the conformation, we are running the gel. This gel electrophoresis in this. Here in first ladder, I have added first well, I have added a ladder and 50 base pair, that is, and remaining are the samples. And here you can see that bands also afterwards. The separation step we will show you in further how we will separate the. This is the power unit. This is, and we have to give 70 volts, and for I kept 1 hour. And this is the agarose gel. The gel electrophoresis unit. This is called gel electrophoresis. Based upon the charges, DNA will be moved to the positive side, because DNA having the negative charge, and it will run towards the positive side. And here we can see the bands. First one is the ladder, that is a 50 base pair ladder, and the remaining are the bands. So we confirm that we got our single stranded DNA molecules. So the purification step is this is the magnetic bead stand. Here you can see that pellet, that is magnetic beads. In the reverse primer, that is biotinated, and the beads are iodine coated beads. We can see that biotin iodine conjugation. After that, this is the amplification step. Here I am taking that. Here we selected that molecules, single stranded DNA molecules. Then we are going to perform the detection method. Here I am taking same again E. coli cells, and I am centrifuging at 6000 rpm for 2 minutes here, and we will get that pellet, cell pellet. Against to that cell pellet, we will going to detect our DNA molecules, aptamer molecules. That is cell pellet. Here I'm adding here I'm taking 2 tubes. One is control and one is test. In this test, we have E. coli cells, and control is nothing. There is no cells, and this is the gold coated nanoparticles. Hello, Mosami. Hello, Mosami. Hello. Hello. Hello, Mosami. Hello. Hello. Hello.
Itu Pranjali no is no. Hello. Hello, Shravan sir. Is my
Hi,
voice
yeah.
audible to you? Yeah,
Yeah, right now I can hear you.
So we are
The
moving
detection
a
method you
little
can
back with the
explain.
yeah. So here we have the app installed in the mobile phone, which is provided by Primary Health Tech Private Limited, which is a partnership company. So like the OTG connection has been done to the host devices. After this, we'll take the gold nanoparticle in the cuvettes as a blank substrate, almost with almost 200 microliter of the gold nanoparticle. And after this, the main E. coli samples, which will be almost 20 microliter, will be added with the gold nanoparticle and will put in the detection system. Then, when the OTG is connected, we'll use the app, and there will be some basic several readings which will be optimized. So this readings takes almost 10 to 12 seconds to get optimized. As you can see, there are several readings. So the final reading is what we consider for our detection procedure. So this is the main cuvette where we are taking the detection main sample, the E. coli cells, almost 20 microliter, and we'll use the same procedure method for the detection. So this is how the final readings comes across. You can see the difference between the two qubits readings. Thank you.
Hello. Yeah, if anyone have any questions, they can please unmute and then ask the questions. Or even text in the meeting chat. Yeah, so we have one question from Arshini Paruthi. So the question is, what is the limit of detection in concentration for the sensor developed?
Actually, so right now the LOD for the demonstration that we have showed you, can you hear me? Can you hear me, guys?
Yes, ma'am.
Yes, ma'am.
Yeah.
We can hear you.
So yeah. Yes, so the LOD right now that we have developed for the sensor that we have shown you in a demo. So this LOD is in the terms of the number of cells that E. coli cells that detection system can develop. So right now this is around 10 to the power 3 cells, which is like 1000 cells is the lowest limit. And that is very good because the clinical level that your urine, if it has say 10 to the power 4 cells, that is right. So that number of cells, 10,000 cells. So if that number of cells are present in any clinical sample, that actually makes it clinical. That makes you that you are suffering from UTI, right? So that those are the guidelines given by WHO. So our detection system, when detects around 1000, if there are 1000 E. coli cells, it could detect it. So it is well below the clinical limit. So that is really good. Okay, so we have another sample. Yeah, 1000. No, Archini. So 1000 cells in 220 microliter. We multiply it. So whatever. So we have a CFU range that we have made. Which shows that against whatever reading that we are getting from the detection device, with that reading, how many cells correspond. So we have made a range of CFU range that we call it, or colony formation unit range. So we extrapolate it. So if we have, say, if we have an OD of for example, if we have an OD of 4 something absorbance reading, so just for the example's sake, I'm saying, so if this 4 number is the output of from the device corresponds to, say, for example, 10 to the power 3 cells. So that is how we extrapolate. We don't have 1000 cells in 20 macroliter. We have a reading, and that reading corresponds to the number of cells, that range that we have made, if it satisfies your query.
Okay. Okay. So thank you, ma'am. So now we will have a break, and the next session will start from 2 p.m. So welcome everyone to the day 2 afternoon session. So welcome everyone to the day 2 afternoon session of NWNTD 2020. We now have with us Professor Pavel Levkin. Professor Levkin is the Professor of Multifunctional Materials Systems at the Institute of Biological and Chemical Systems at KIT, Germany. He graduated from the Institute of Fine Chemical Technology, Moscow, and obtained his PhD in Organic Chemistry from the University of Tübingen in Germany, followed by postdoctoral work at the University of California, Berkeley. Professor Levkin is the co-founder of ScreenFect GmbH and AccuArray GmbH. His research focuses on the development of functional and responsive materials and surfaces for biomedical and biotechnological applications. Now, I would like Professor Levkin to take over. Sir, we welcome you to join us and give your valuable talk.
Thank you very much for the introduction. I will share my screen and start my presentation. Can you see my screen?
Yes, sir, it's visible.
And you can hear me, I hope. Yeah?
Yes, sir, yes, sir. Yes, it's audible.
Perfect. Thank you. Thank you for the introduction again, and thanks for inviting me to present to you at this conference. My group is located at the KIT Karlsruhe in Germany, and the topic for my talk today is basically so-called chem-bias, so marine chemistry with biology by combining on-chip synthesis with cell screenings. And in my group at the KIT, we are interested in the development of novel functional material systems, and we approach this problem by working at different kind of material science aspects. We work on photochemistry, on surface functionalization. We work on superhydrophobicity and omniphobicity. We develop novel porous polymers, liquid infused interfaces, and also dynamic and soft materials and interfaces. And then we use these methods that we develop to make functional materials with complex functionality sometimes, which we then apply to solve biological questions. So, for example, we're interested in how cells interact with interfaces, how we can control cell behavior using functional materials. How can we control cell patterns or make cell patterns or bacterial patterns or gradients using functional materials, functional interfaces? How can we control biofilms and bacteria on surfaces? And finally, also, one of the active topics at the moment in the group is how to accelerate drug discovery, how to accelerate biology, biological experiments using functional materials and functional interfaces. And today I will talk mostly about this last part, about how we can use functional interfaces to solve some of the biological problems, especially cell screenings, drug discovery, personalized medicine, and how to enable novel types of biological experiments using functional materials. And this brings me to the topic of superhydrophobicity and hydrophilicity, which we have been working on quite some time. And we developed a number of methods to make either superhydrophobic surface, which is extremely water repellent surface, or superhydrophilic or highly hydrophilic surface. And these two interfaces, they are very interesting by themselves because this is two basically completely opposite functionalities. It's not possible to make a surface more hydrophobic than a superhydrophobic, so with 180 degrees which is not accessible but close to it. It's not possible to make it more hydrophobic, and it's basically not possible to make a surface more hydrophilic than a surface with zero contact angle. And they are all interesting by themselves, but if you can combine these two interfaces into the same surface in precise micro patterns, it can lead to additional functionalities that are kind of unexpected, or that cannot be produced using simply super hydrophobic or hydrophilic surfaces. Here's one example that we showed for quite some time ago to make such microfluidic devices, basically by making hydrophilic pattern on a super hydrophobic background. It's a 2D flat surface, but it behaves like a microfluidic system that can potentially be used for diagnostic applications. Another example is shown here. These are aqueous droplets of complex shapes that are formed by filling hydrophilic patterns of different shapes on a super hydrophobic background. So here you can see 3 different droplets that are separated by a super hydrophobic barrier, just 100 micrometers wide. And these patterns can be used, for example, to make 2-dimensional patterns of cells by culturing cells in these compartments next to each other, or we can make patterns of particles using this method, or patterns of hydrogels, which are difficult to pattern using other methods. So this is already useful, but then we also discovered that if we take this method and make a microarray of hydrophilic spots on a super hydrophobic background, we can make thousands of nanoliter sized droplets in every single hydrophilic spot. For example, by this method, so-called discontinuous dividing. So if you just roll a droplet of water, you can form thousands of nanoliter droplets in every hydrophilic spot. And this is interesting because you can use, instead of water, you can use suspension of cells or particles or bacteria to form in one step thousands of compartments that are isolated, that can be used for individual experiments. And also, since we can make the hydrophilic spots of any different shape, we can control the shape of the droplets, we can form triangular droplets, we can change this, the size of these droplets, and also make very high density droplets. In addition to the discontinuous dividing process, this method can be used with a liquid dispensing, and then you can control the volume of the droplets, as shown here on the right, from few nanoliters to microliters. And we can also print cells using a cell printer into the droplets. And in this case, we can use these droplets for biological experiments, for cell experiments, where we deal with tiny volumes, 100 nanoliters or less, and also very small numbers of cells. And this is very important for biological experiments where you have to use cells that are not really widely available. For example, cells from biopsies. Yeah, you have a few thousand cells from a biopsy. You want to analyze it. You want to do a screen with different drugs, and you cannot use microtiter plates with microliters, because there you need thousands of cells for one experiment. Here you can use 5 cells for one experiment, or 100 cells for one experiment, and also very low quantities of expensive chemicals and reagents. And over the last few years, we have demonstrated different applications of the droplet microarray platform in biology, for screening of cancer cells, primary cells, stem cells. So currently we work on human induced pluripotent stem cells. Showed one example on single cell screenings. and also some more kind of exotic examples that are also shown here schematically. So here you can see an example where we use droplet microarray in a hanging droplet method, where cells that are usually form monolayers, they can form 3D structures, 3D cell spheroids, and then we form basically a microarray of single cell spheroids in this in this way. In this case, we use droplet microarray to form hydrogels to gelate these droplets, also containing cells, to form such hydrogel particles of defined shapes, where the shape is defined by the shape of the droplets. And since we can control the shape of the droplets, we can make hydrogel particles of different shapes. And here we use droplet microarray to form an array of actually whole animals. In this case, this is a zebrafish embryo, and every droplet contains one embryo of a zebrafish, which is important because zebrafish embryos, they are used for toxicological screens. It's a big difference from using cells, because this is an animal model and the results sometimes are more predictive, more relevant than results in 2D cell lines. Now I want to come to one separate project from last year, which is called actually ChemBios, which is abbreviation from chemistry and biology together. And the motivation behind this project was and is shown here on this slide. Motivation is that the inefficiency of drug discovery in principle at the moment. So at the moment, to develop one drug, it takes on average 20 years and about $2 billion US. And this is it's extremely inefficient, yeah, because if you think how many experiments you could do with $2 billion US as a research group, you can imagine that this, if you had that $2 billion US for average research group, you would have 1,000 years to work without additional third-party funding. Just simple example. Of course, there are different reasons for these expenses, but one of the reasons for why it takes so long and why it's so expensive is that to develop drugs, pharmaceutical companies, in more than 60% times they do cell screenings. So they screen millions of compounds with cells to identify drug candidates, to identify hits. And this process is extremely expensive, and it's only affordable to pharmaceutical companies. It's schematically shown here. So you have to screen thousands of millions of compounds using cells, and to synthesize all of these compounds, chemists use methods that have been developed 100 years ago. They basically synthesize them one by one. For this reason, this process is very expensive, and it took us many years to collect these libraries that we have now. But you cannot resynthesize 1 million compounds using the old traditional methods. There's another problem, is that the chemistry is disconnected from biology in this process, and chemists use their methods, biologists use their methods, and you have to transfer chemicals off from chemistry labs to biology labs one by one, and for 1 million compounds, that's really expensive, inefficient. So the whole process is very slow, expensive. Only a few pharma companies can do this. And to solve this problem, to improve this process, we have to kind of unify chemistry with biology. We have to miniaturize the whole process to make it much cheaper, and also we have to parallelize it. So The idea behind this project is to develop on-chip synthesis process, on-chip characterization, and on-chip biology. Only in this way we can potentially improve, accelerate this process and make it more efficient and help drug discovery to become more efficient. The problem is, if we want to use droplet micros, that superhydrophobic surfaces are also oleophilic because they are porous or rough and hydrophobic, which makes them oleophilic for organic solvents. So chemistry is difficult if you have to use water. So in my lab, we recently developed a method to overcome this problem by making omniphilic patterns. And this was done by going from a rough surface to a flat surface without defects. We developed simple chemistry to make omniphobic surfaces flat on flat glass surface, and this surface became indeed omniphobic with low contact angle hysteresis and low sliding angles even for low surface tension liquids. And then by patterning this substrate, it is possible now to confine even organic solvents in small droplets, even liquids with very low surface tension, such as even hexane, which has very low surface tension, but we can form droplets in precise patterns. And this allows us now to do chemistry, organic chemistry, in these droplets by controlling the atmosphere to avoid fast evaporation, obviously. But to demonstrate this, we designed a combinatorial chemistry approach, which was developed by Mija Nurmolo from Calcutta University, who's now at the university, who was von Humboldt fellow in my group. So he developed a method which is a 3-component reaction based on amines, thiolactone, and pyridyl disulfides that leads to a lipid, a cationic lipid in one step. And then by changing building blocks, thiolactones, pyridyl disulfides, and amines, we can make a library of such lipids. And these lipids can form liposomes, can then interact with nucleic acids, and potentially can transfect cells, can deliver this nucleic acid to the cells. So we wanted to synthesize a library of these lipids on chip and screen it on chip with cells to show how we can do the synthesis, characterization, as well as biology on chip. So this was done, the synthesis was done in the following way. So we had 2 arrays, one with amines and another one with a mixture of thiolactone and pyridyl disulfides. So by sandwiching them together, we start the reaction in every single droplet to form basically a droplet microarray with different lipids in every single droplet. And an advantage of such droplet microarray platform is that we can take this droplet microarray and we can kind of sandwich it onto a multi-plate and then use this multi-plate for multi-mass spectrometry to analyze the molecular weight of every single compound that has been synthesized on chip in parallel without kind of transferring compounds one by one to vials as it is usually done in chemistry. And this works. So here you can see some spectrum, multi-spectrum. So then we took this array of lipids. Also, what you can see here, it is possible to do on-chip parallel extraction, which is interesting and important because when we miniaturize chemistry, purification becomes very challenging as well as characterization. And parallel purification is important. And this demonstrates that it is possible to do indeed parallel high throughput mini extraction on chip. But the next step was to take this library of lipids that we have synthesized on chip and we sandwich it with droplet microarray containing aqueous droplets with plasmid DNA that we want to use for transfection. And this process leads to the formation of liposomes in one step, as well as lipoplexes. So liposomes complex with nucleic acids. So at the end of this sandwiching, we have a droplet microarray or microarray of different lipoplexes, where each lipoplex is formed from the same nucleic acid but different lipids that have been synthesized by this process on chip. So here you can see characterization of liposomes by DLS, lipoplexes. And after this, the next step is to take this lipoplex library microarray, print cells, live cells into each spot on chip, and to start in this way a cell transfection experiment or gene delivery experiment. Culture incubate cells with these lipoplexes and then visualize the result by fluorescence microscopy, where we detect green fluorescent protein, GFP, which is expressed in the cells that have been transfected, successfully transfected by these lipoplexes containing plasmid DNA encoding GFP. So the higher the fluorescence, the better the transfection efficiency. And here you can see some results. We found some lipids that have been synthesized on chip that are more efficient than commercial transfection reagent. What is very important in this process as a kind of demonstration is that we have done on-chip synthesis of 75 lipids, on-chip characterization by multi-mass spectrometry, on-chip lipoplex formation, on-chip screening with cells, and identification of efficient transfection reagents, and all of this has been done within just 3 days and about 1 milliliter of solvents and liquids. And this demonstrates that this unification, combining chemistry with biology in the same platform, miniaturization, can really significantly accelerate drug discovery of the process of, in principle, screening, chemistry, and biological experiments. Because if you would have to do it one by one using a normal way with 75 lipids, that would take several months at least. Let me show you now another example of a related project, which is continuation of the previous project, where we wanted to improve the system to avoid the sandwiching steps and all transfer steps basically, and also to demonstrate and optimize the possibility of doing on-chip characterization. On-chip characterization of the synthesized library, which is important. To solve this problem, and what I didn't mention at the beginning, we use sandwiching to merge organic droplets with aqueous droplets to form lipoplexes. And to solve this problem, we have to improve the omniphobicity and omniphilicity of the surface. And this was achieved by changing the surface functionalization. We went from a monolayer of silence to a dendrimer functionalized surface with higher density of functional groups. This was done by starting from an alkenes functionalized surface, then thiol-in reaction, then in the second step esterification, and again thiol-in reaction. In this way we can build a dendrimer coated surface with double bonds at the end of the dendrimers that are reactive, that can be patterned with UV light. And the dendrimers are important because in this way we achieve a much higher density of functional groups, and this density is important to improve omniphobicity, as shown here. When we increase the generation of dendrimers, we increase the advancing contact angles or the static contact angles on the fluorinated surface and reduce the static contact angle on the hydrophilic surface. And in the extreme case, we have very large hysteresis, kind of apparent hysteresis between a fluorinated surface, between advancing contact angle on the fluorinated part and receding contact angle on the hydrophilic part. It's almost 130 degrees. And this is important to make stable droplets on the surface of different surface tensions, from water to organic solvents. Here you can see how the thickness of the coating increases with increasing the level, the generation of the dendrimers. The surface is transparent and we can make very precise patterns on the surface. And then we developed a method to coat ITO slides, conductive indium tin oxide slides, with this method. And since the surface is conductive, now we can do the synthesis and then we can use the same substrates for mass spectrometry, for multi-mass spectrometry. We can quantify synthesized compounds or characterize them by MALDI. Here you can see that the produced substrates are extremely well suited for MALDI, because we can have very high sensitivity, down to 0.1 femtomoles could be detected in a single spot, which is very, very high good sensitivity. And this is important because it's important to miniaturize chemistry to accelerate it. And with miniaturization, many methods become unacceptable or not compatible, like NMR becomes impossible, more or less. HPLC becomes very difficult, slow. So we are become limited in terms of the methods available for characterization of femtomoles. Yeah, we work with femtomoles, nanoliters in a high throughput. So multi-mass spectrometry becomes one of the few methods that are available, but luckily it's a very powerful method and we can detect very small quantities of compounds. Another advantage of ITO slides is that it reflects IR light, and we can characterize compounds on chip using IR, and we can quantify, not quantify, but characterize different compounds using on-chip IR spectroscopy. And here you can see that on-chip UVV spectrometry becomes also important by building such a simple sandwiching device. In this case, we can do transmission UVV spectroscopy on chip and can look at UV spectra from each single droplet using a plate reader. And this opens a possibility to do on-chip parallel monitoring of reactions. So how the reactions proceed using UV spectrometry, which is demonstrated here. So we can use this model reaction that leads to the lipids, but it also leads to a side product, which is a pyridyl thiopyridone, which has a very distinctive absorbance and UV spectrum that can be detected easily by UV spectrometry, which we do here. And here you can see the result of a on-chip parallel optimization of the reaction using this method, where we analyze 50,000 data points to get to this plot. And this is important for high throughput optimization of chemistry. So in summary, this method opens different possibilities. It enables us to do on-chip chemistry, on-chip mass spectrometry, on-chip IR spectroscopy or UVV spectroscopy, as well as on-chip biology, so to do biological cell experiments. And this is really important for accelerating drug discovery and accelerating biological experiments. So I have one more small project to present. I want to ask, how much time do I have left?
Yes,
you have
sir.
another 15 minutes.
Yeah,
15
another
minutes.
15 minutes is left.
Okay, thank you. So I have a short project, probably less than 15 minutes, but it's relevant to this and I think quite interesting. It has been just accepted for publication in advanced materials. It's not online yet. The project is called Assembly of Multispheroid Cellular Architectures by Programmable Droplet Merging. And it's schematically shown here. So, and the main idea is that in the case of droplet microarrays, we don't have plastic walls between. We have a flat surface with basically just a super hydrophobic barrier between the droplets. And this means that we can, in principle, we can increase the volume of individual droplets by liquid dispensing, and we can merge these droplets in a controlled way. And this merging allows us to basically kind of bridge two neighboring droplets containing cells or cell spheroids, maybe different cells, in a controlled way in a high throughput on chip. And here you can see an application for this. So here you can see that we can merge these two droplets or three droplets or four droplets, and if each droplet contains a single cell spheroid, so this is not a single cell, but it's a 3D cell spheroid, these spheroids will come into the same medium and they will fuse. They start to adhere to each other and they start to fuse together, and which is you can see in this SEM image or in this microscopy image. And by using this method, we could demonstrate that we can fuse different numbers of cell spheroids. Here you can see up to 4 cell spheroids, and we could find all different architectures because you can fuse spheroids in a linear way or they can fuse in a different way. It's based on statistics, but we could see all different architectures. And this method basically allows us to make a very relatively complex 3D architectures by merging, by fusing single cell spheroids. And what is interesting here is that we can start from different cell types. We can fuse, for example, 2 different cell types to form a 3-dimensional architecture based on 2 different cell types, or even 3 different cell types, and to make this interesting complex architectures. And what is also important here that we can do this in a high throughput in parallel on chip, and this is important for screening such complex architectures with drugs, or to investigate these architectures for tissue engineering. What is also should be noticed mentioned that this fusion process, it's a dynamic process, and cells or spheroids, they start to fuse and they keep fusing, they keep pulling to each other, and this process can be modeled. So in a collaboration with another group, we modeled this process and showed how cells come to each other, how they continue to fuse, but they don't form a spherical architecture at the end. They stop at such a kind of ellipsoid. And finally, an application of an example of application of this process is related to investigating cell signaling processes. And Wnt is a protein. It's a cell signaling molecule, probably one of the most studied molecules, and at the same time one of the most important signaling molecules because it's involved in the development of embryos, in excess patterning and cell fate, cell proliferation, migration, diabetes, cancer, and so on. And On the other hand, despite this importance, it's still not completely understood how the signaling processes happens, how the propagation of the wind molecules happens, how cells communicate via wind signaling in 3D, in 2D, or in vivo, and we wanted to demonstrate the potential of the method by looking into this problem. So what we did, we formed cell spheroids that release wind 3A molecule, and then also cell spheroids that that respond to wind molecules, so top GFP reporter cell line, and then we fuse them together on the droplet microarrays. So here you can see a control experiment where it's just identical cell types and there is no activation of wind. So as soon as the top GFP reporter cell line is activated by wind, it starts to express GFP, becomes fluorescent. But if it's not activated, it's not fluorescent. As soon as we have this system together, then we can see visually how wind molecules activate the reporter cell line, how they kind of go from one cell part of this double spheroid to the other part, which can be quantified, can be visualized in time. And here you can see also more complex architectures where we have three different spheroids merged together. And this demonstrates again how this method can be used to investigate, for example, cell signaling processes in 3D using these relatively complex but defined architectures. And also what is important, this process can be studied now in a high throughput way using parallel method and maybe combined with some screening experiments and different microscopy techniques. So with that, I would like to thank my group, my students who worked on these projects, our collaborators at the KIT, also funding. Thank you for your invitation again, and I will be happy to answer any questions. Thank you.
Thank you, sir, for your valuable talk. Now we have one question from Amir Suhail. So his question is, can we use the same idea to flow fluids on top of the hydrophilic parts on a hydrophobic substrate, a dynamic system of the same sort?
Sorry, I didn't quite understand. Could you repeat the question, please?
Yes, sir. The question is, can we use the same idea to flow fluids on top of the hydrophilic parts on a hydrophobic substrate?
Can we use the same idea to flow fluids on top of the hydrophilic parts on a hydrophobic substrate? If I understood the question, but I'm not sure I understood it correctly. So the idea is whether we can combine it with flow and microfluidics in some way to, although the question is about dynamic system of the same sort.
I don't know, like probably, Pavel, I think the participant probably asking, whatever you have shown, that is hydrophilic patches on superhydrophilic background, right? I think he probably wants to know whether you can make it other way around, where hydrophilic background and superhydrophilic patches, so that it can tunnel. I think so, but I don't know.
Hi, Uttam. I mean, it is possible to change the pattern in principle. Might be interesting for something, but not for confining liquids. Let's say you would form a sort of microfluidic network on the surface, which is interesting for some applications, but not for what I have demonstrated. It is possible to change chemistry, so it's a very dynamic, let's say, flexible system. You can make functionalize this hydrophilic part with different chemistry, with reactive groups, or can make different geometries and so on. So, any other questions?
I have some. So, can I go ahead?
From my side? Yeah, sure.
Yeah. So, thank you so much for accepting this invitation and giving this wonderful talk. It's amazing the way you are, taking a direction and doing all of this, which is probably in a few years probably materialized, right? So I have a few concern about like how you control this fusion of the spheroids, like in the last part of your talk. So where basically one to one and probably like two patches and then another diagonally, so the 3 or 4, like how you control those selective fusion of those arrays, subset of those arrays.
So, yeah, that's a good question. So, in the fusion of droplets is done by basically increasing the volume of individual droplets. So we use a liquid dispenser with print. We can deliver liquids into any droplet on the surface in a controlled way, the sort of inkjet printing method. So we basically slowly increase the volume, and then at some point they fuse because they touch each other. The question, another kind of related question would be whether we can control how spheroids are fused then inside of the droplets. And they are fused by themselves. That's what we cannot really control. So as soon as they touch, they start to fuse because there is a part adhesion between the cells and they start to merge. So it's a kind of self driven process of cell interactions. There we cannot really control it so well.
Okay, so you said that that's the reason I want to know. So look, when you are adding the droplets, basically you are creating some convection, right, in each small dimension, right? So that convection can also somehow accelerate to fuse those two spheroid, isn't it? So isn't it like in a way you are forcing them to come and meet each other?
It's not based on convection. I think it's based just on the physical volume, and since at some point they start to touch, the droplets basically contact each other and they fuse. Sometimes we print a liquid droplet onto a hydrophobic part between the two droplets, and this helps. Yeah, so you have two droplets and then you print in between, and they merge spontaneously.
Right. So no, but like I raised this thing because when I at some point I work on these pattern interfaces for microfluidics applications. When you put two droplets, basically once they meet, once they like try to mix each other, then they'll have a convection. So if you also take two droplets and if you allow to diffuse, you will see that convection and it's mixing whole process, and that create a momentum inside that small confined zone. So that is my concern. So it might be minimal, but that probably something is there, probably, right?
I mean, there is definitely convection inside of the droplets. They are quite dynamic in this respect. You can see this, yeah. But the cell culture is a process of hours or sometimes days. So as soon as it's merged, then we culture it, incubate it, and the cell spheres, they precipitate into the bottom of the droplet. Yeah.
Right, because as they precipitate, that is my concept. Because the cells are precipitated, right? They're forming those spheroids, right? So unless and until physically they meet and hit, it's very difficult to like come on their own and then fusing it. So that was basically the whole idea. Like I was kind of skeptical about that. But anyway, like you can think about it.
That's a good question. Yeah, maybe it's good point. Yeah.
Okay. Another thing is like, in your middle part, like where you are talking about doing some chemistry in that confined space, right? Now, the major challenge probably, like if it is aqueous media, there probably we can add a little bit of glycerol just to prevent the evaporation of this reaction media and all, right? But when it's come to organic media, like yes, there have certain organic medium, like I have seen, like you use DMSO, which is there is no concern. It can remain for days and weeks without evaporation, right? But when you're looking for like THF, acetone, which is common organic solvent, then probably the problem, right?
Yeah, you mean evaporation?
Yeah. If the
Yeah,
reaction solvent is not there.
It is definitely an issue, and simple systems like we are used to work with acetone in a flask, yeah, or in a vial, yeah.
Right.
This is like one of the aspects. We have to, in the future, let's say, to miniaturize this process, use similar processes as in biology. In biology, they don't use milliliters, they use microliters since many years. In chemistry, we have to do the same principle. One of the problems is evaporation. Another problem is characterization. Another problem is purification, and so on and so on. So this has to be solved, and yeah, it depends. You can solve it by in different ways.
Right. Great, Pavel. Great. Thank you.
Thank you. So I see some raised hands.
Hello, sir. Actually, I was asking about microfluidics. So instead of a channel or something, if you can have a hydrophobic surface, and if you have a particular path or on the surface of like on some particular locations where there will be some super hydrophilicity. So can we, like, instead of the fluid being stagnant, can we like pump the fluid and get a microfluidic flow through that, or like some nanofluidic flow through that through the on the top of this substrate where the hydrophilicity is more? So how much flow rate, or like is it even possible to flow the fluid than fluid being stagnant in particular locations? That was my question. Thank you.
Thank you. So as I understand, your question is whether we can use this method to make microfluidic channels, basically, and generate flow inside of the channels. So if this was the question, then yes, you can make hydrophilic patterns.
Not channels on top of, not exactly channels on top of a substrate on particular locations.
Yeah.
Like just flow fluid. That was like if you can make this particular whatever like small volume we have created. So if we can make a passage and pump the fluid from one side, can we get can we do that? Will the this hydrophobicity and hydrophilicity, will that forces will be sufficient to confine the flow through that particular path, like flow of fluid?
I think the hydrophilicity and hydrophobicity by itself cannot be used to generate flow. What can be used to generate flow is so-called Laplace pressure. So if you make a pattern of a certain geometry, then
yes, sir. Actually, for generating flow, I was thinking like if you can use some pumping other pressure-driven forces. But I was asking for confining the fluid to the passage. Can we use the difference in hydrophilicity? This for confining to the passage.
To insert it the system into a microfluidic channel and then confine fluid using a I don't think it might be possible to a certain extent unless you have higher pressures, because the surface tension confinement is not pressure resistant, let's say. It has limits. So if you have a flow, which I usually used in microfluidics, it will probably also go across the hydrophobic parts, not only hydrophilic parts. but to some extent, it's possible to achieve.
Okay, thank you, sir.
Thank you, sir. Okay, now we have with us Professor Ravinder S. Dahiya.
Okay, thank you. Bye-bye.
Thank you, sir.
Bye, Kapil.
So now, continuing our session, now we have with us Professor Ravinder S. Dahiya, who is a Professor of Electronics and Nanoengineering at University of Glasgow. His group, Bendable Electronics and Sensing Technologies, also known as BEST, conducts fundamental research on high mobility materials based flexible and printed electronics, leading to electronic skin and their application in robotics and wearable systems. Professor Dahiya has published more than 350 research articles, 7 books, and 15 patents. He has given more than 140 talks. He has led many international projects funded by European Commission, EPSRC, the Royal Society, the Royal Academy of Engineering, and the Scottish Funding Council. Professor Dahiya is the President and Distinguished Lecturer of IEEE Censors Council. He is serving on the editorial boards of several leading journals. He holds the prestigious EPSRC Fellowship and have also received in past Marie Curie Fellowship and Japanese Monbusho Fellowship. He has received 9 Best Paper Awards as author or co-author. He also received IEEE Censor Council Technical Achievement Award and Microelectronic Engineering Young Investigator Award. Now, I would like Professor Dahiya to take over. Sir, we welcome you to join us and give your valuable talk.
Thank you very much. Can you hear me well?
Yes, sir. It's audible, sir.
All right. Thank you very much for the kind introduction and for inviting me to share my views about flexible electronics. The title of the talk is Soft and Squishy Electronic Skin. So the title itself tells that I will be starting with the current technology, current electronics, which is rigid, stiff, and moving on to flexible, and then I'll move to the soft electronics. And the discussion will be through electronic skin, which in context with robotics. So that sets the context, because in robotics you need all these features. You need electronics to be conformable, you need electronics to be flexible, stretchable, and soft, etc. So before I start, quickly, what we do in my group, we try to understand sense of touch in humans, and using this knowledge to develop electronic skin for robotics. And that's the technology that becomes as a platform now, is now being repurposed in many ways, brings back to improve quality of life, either that is through wearable systems or prosthetic limbs with tactile sensory feelings, etc. And we also develop our the interface between virtual and real world through volumetric display, holographic, pseudo holographic display that you see here on the screen, where we are able to display virtual objects in 3D, that's mid-air display, and we are able to control them either through gestures or via haptic feedback. So what you see is a gesture-based control, and now you can interact with 3D models also. That is shown in the video on the screen. And after that, you will see a virtual button, a push button, which you can press and can feel also the pressure when air hits the hand. You can feel the pressure also. And because it involves aerohaptic, you can also have a temperature feedback, which is something new. No other pseudo holographic display or holographic display is capable of doing that. So that's a kind of interface using electronic skin, using touch sensing as an interface between real and virtual world, and bringing them together. So I would like to, before I go to the main part of the presentation, I would like to thank my group members. Most of the work that I'm going to present is their hard work. So here is the outline of my talk. Why electronic skin? The first part, I'll spend a couple of minutes to describe why electronic skin is an important topic. And then I'll move on to the second part, which is electronic skin technologies. What has been done, particularly in context with robotics. I will take this discussion, but this applies to flexible electronics in general in many other application areas as well. So the discussion is structured as what can be done on a time scale, what solutions exist today, what can be done in midterm, and then what is possible in long term, how the electronics can take this on a time scale, how this can be developed, and also on the dimensional scale, looking at starting with nanowires and printing nanowires to develop electronics, leading to electronic layer, and then making electronic skin and taking it towards the system, and finally using it in the application. So that's the kind of journey I'll take you through. And third dimension of this this talk is what I already said, is related to starting with the rigid electronics and going towards the soft electronics. And these are the parts. Then I have included third section. I don't know if there will be enough time. If time permits, I can give some example of how this electronic skin technology or flexible electronics is being used in other areas, such as in humans, where we either through wearables or some disposable devices. Okay, so and then I'll conclude my talk. So coming to the first part, the why electronic skin. If you think about all 5 sensory modalities that we have, touch sensing actually starts from is the first modality that any the moment that life originates, the touch sensory modality is the one you come across, and you start to feel the world through touch sensing, even when audio and vision are not there and other sensory modalities are not there. So such is the importance of touch sensing as a mobility, as a modality. And in real world, try to this type of experiment, try to put hand on ice block for some time and then grasp an object nearby. You will see that because temporarily touch sensory modality is not there because of numbness. You will see objects slipping out of your hand, and you can see this happening, but you have no choice. You cannot do you can do nothing much can be done about this. And this then highlights the question, why are we developing machines? Why are we developing prosthetic limbs? Why are we developing robotics without electronic skin or tactile skin? If we have tactile skin on machine, it will be much safer, and that has a real life implication as well. If you see this example on the screen, that shows robots working in a car manufacturing unit, and you can see that robots are working sort of in cages, and in these cages you don't see any humans, and by chance any human sensor enters into this cage, you also come across this news such as robot kills man in Volkswagen plant, or one example taken from India where robot was blamed for the death of a human, of a skilled worker, even though it was accidentally human enters into the workspace. So the question then arises is, why are we developing machines which can be unsafe? And that leads to the question about electronic skin itself. If there was electronic skin, if robot's body was covered with something that can either sense the soft touch or proximity or the touch itself and stop appropriately, then it will lead to a much better outcome. It will lead to next generation of robotics, which is evolving fast. We talk about Industry 4 today, and Industry 4 where human and robot are supposed to work together. And that's when you have physical interaction, you need to have a safe interaction, and that's where again electronic skin comes into picture. Not only this, there are several other push factors today which are pushing the research in electronic skin or flexible electronics in general. And there are several pull factors which you see on the right side of the screen. So examples I have taken are from the medical, biomedical, where haptic feedback, the surgical instrument that you see on the screen, or the minimal invasive surgery, where we expect electronic skin to be wrapped around surgical instruments and giving pressure feedback, etc., pressure mapping. And that has implication on people have started to use pressure mapping concept in automation sector, in cars, etc., putting them sensors on the seat and getting pressure maps and studying the behavior aspect. So it has multiple dimensions. In future, if you look at future looking forward, this could have implication in the retail sector, where you will be able to, for example, display that we see today. Through display, you may have haptic feedback, and you will be able to feel the texture of clothes you want to buy online. Likewise, doctors will be able to palpate and feel the tissues remotely, and that particularly is of huge importance in current context when we talk about touchless interaction because of COVID. And looking at further ahead, you may also think of neural control of limbs with soft robots, where distributed electronics, and this distributed electronics and sensing certainly would be seamlessly integrated, and stiff electronics may not work always in such scenarios. And at the center of all these lies the scientific question, which we need to understand. And these originate from the variety of features that real world objects offer. Some objects are hard and soft, some feel the texture is rough or glossy. Like that, you have several questions, how we interpret, how our brain interprets. So that leads to another dimension, not just the technological dimensions, but also the perceptual dimension, where you have to look at how to handle the data, how to compute, and that makes electronic skin very attractive, the topic, because you are dealing with the technology, advancement of the technology. You are also dealing with scientific questions which have implication on computing. For example, neural-like computing, have implications on big data, have implication on communications. So all these come together as a package. And you can see on this slide that this package has various strands which you need to look into to make the skin effective. And when I say effective, I mean that it's something that can be used in real life, and not just a benchtop sensor or set of patches you continue to characterize in lab. It has to, you have to look at various functionalities. You have to look at hardware, both from mechanical point of view, from electronics point of view, and that's where mechanical flexibility, stretchability, etc. they come into picture. This is where your computing, energy, fast data transfer, etc. they come into picture. Robotics adds to the challenges, because in robotics you need to have sensor data, you need to have feedback, and based on that feedback, there is some computation need to take place, and based on this computation you drive the actuators, and action takes place. So it's a complete action reaction cycle that you need to think about, and sensor must fit into, or electronics as a whole must fit into that action reaction cycle, and it must be fast enough to give a real a human-like response, if you have to look at humanoid, or it could be other robots that are being explored currently. And finally, you have to look at the manufacturing aspect also. It must be low cost, it must be manufacturable, reliable, and easy to maintain, etc. So looking at these, one has to then start looking at if you consider if you compare electronic skin or tactile skin with respect to other sensory modalities, there are several other challenges, and one of them is that you have to have multiple type of sensors to feel different parameters that real world offers. And also these multiple type of sensors are not just located on the fingertip, they are actually distributed throughout the body. This raises integration challenges too. This brings wiring complexity related challenges also. Unlike CMOS images, which has been a key technology, a distinct technology for vision, there is no unique technology that applies to electronic skin. And this is one thing that we have to consider. And then comes the question related to electronics itself. Can we use current electronics? So if we were to use current electronics, we will have to redesign the robotics. We have to have planar surfaces, and on that on those surfaces you can put current electronics. But that's not the future. If you look at from the psychology point of view also, we appreciate we are able to connect to objects which look like us or which look like animals around us, and that's the real world thing. Real world is curvy, real world is soft, is flexible. There are so many objects around you which require those features, and that means you have to then think of putting electronics in such a way, or developing electronics such a way that these requirements can be met. I mentioned about data energy. If you look at number of sensors that are mechanoreceptors, which is equivalent to pressure sensor or touch sensors in the body, then in the palm area itself, you will have more than 5,000, and these are rough estimates. Number can be even bigger. And I have not included any temperature related or pain related receptors in this discussion. So if you were to read all these simultaneously, then for 5,000 sensors you need 10,000 wires, and that leads to a different problem in terms of using it. So you want to develop electronic skin, you get the useful data, you get real-time data from all sensors at the same time, but also it is not allowing the hand to move, because 10,000 wires, try putting 100 wires on your hand and then try doing this experiment, you will see you won't be able to move the hand. The fingers dexterity will be sacrificed in the process. And that leads to then practical problems. So you are collecting huge data. It's difficult to handle the data. It's difficult to interpret. There is a limited communication bandwidth. You cannot transmit all data to the brain. So you have to reduce the data. That brings the other techniques here. And all sensors associated with electronics, if you take all of them together, then you have to deal with the energy. Also, current research, people talk about that large area is a problem, is a challenge. And because of the issues that I'm mentioning, then it becomes they call it as an energy sink. But later I'll explain, I'll show you some of the recent results from last month only. We published a paper where we have shown that it's not a challenge. Actually, skin is an opportunity to generate energy and operate other devices as well. So this brings me to the definition of what I mean by conformable or flexible electronics. And this patch that you see here, you could call it as an electronic skin, particularly when you have physical sensors for robotics. But if you put electrochemical and other sensors, then it can be applied to other areas. So having various type of sensors integrated with other functionalities such as energy harvesting, distributed memory, signal conditioning, possibly wired or wireless connection as needed with the application. All these functionalities integrated on flexible substrates is what makes the skin patch or conformable electronics. Sometimes it may be difficult to have these all functionalities on same substrate, because of either substrate does not allow the processing of the material, or the materials are to be processed in different ways to avoid affecting the performance of other devices. So you can also think of a stack, a flexible stack, and that's like 3D ICs, but in this case, it's a flexible version. So with this, I go to the next step, and that's about what are the technologies available. We looked into all these aspects, challenging aspects that I mentioned a while ago. And with that, we started looking into what are the potential solutions. We also need to consider the performance, which I've not discussed so far. There are so many groups talking about plastic electronics these days, and mainly it is about organic polymers, organic devices, which has not lived up to the expectation when we compare in terms of performance. Robotics is a good example for all computing and action reaction type cycle, where you need fast response. High performance becomes a key indicator there. So you have to then look at the materials and method in this context, that you want to develop electronics which has performance at par with current silicon electronics or current rigid electronics. And that led us to explore several methods and materials. We looked into organics, we looked into inorganic. I have not included that slide here, which compares the various materials on different attributes, both process parameters as well as performance parameters. But answer was very clear that silicon was the clear winner because of high mobility, because of mature technology, and the material purified material that is available today. So with that background, we focused on most of the time, our mostly research is focusing around high mobility materials. So we started looking into silicon and ways to get thin silicons or printing silicons, etc. And this is what I'm going to explain as we go forward. So next part is what can be done on the time scale. We looked into what can be done with current technology. So existing technology is not bendable. Can we use it and to sort out some of the problems, some of the challenges that we are facing in robotics? With that intention, we got this EU project funding from the European Union, and the project is RoboSkin. where the challenge was to develop the electronic skin for this robot, which was the outcome of another EU project. So this electronic skin, the robot interaction was primarily without any tactile sensory feedback. But then there are so many tasks in real life where you need feedback from entire body. As an example, if you have to lift a heavy sand bag, you don't use fingers, you just grab it and use the contact from the body parts. You plan it and you pick and place it. A simple task such as pick and place, you need feedback from the different body parts, and this is simultaneously coming. And this project was we were looking in this project when even mobile phones, they did not have multi-touch sensing. It was just about even mobile phone had at that time switches which were, you press one at a time. And that led to the area contact was an interesting aspect. This led to this opened in robotics itself. This opened a new area where people started looking into whole body as an opportunity because of the skin itself. Before skin, they never looked into this aspect that whole body could be used to exploit or to plan the movements. Animals do that. If you look at example of snakes, they don't have any limbs, but they rely on entire body feedback and they execute the task. And this is a soft body I'm talking about. So that's the example where skin is completely, you can see the body, most of the body parts are covered with the skin, and this is nicely packaged skin, which is commercially available too. The key main point here was the fuller map concept that you see. The body, if you look at body, there are parts such as shoulders, which are spherical, then there are arms, which are cylindrical. So you have a combination of body parts, body shapes, and you need to cover all these shapes with electronic skin. I guess most of you are today in this meeting, most of you are familiar with the technology that you realize most of the time electronics, it's easy to realize on flexible planar substrates, but when it comes to curvy substrates, because most of the technology that we have is light based, it makes it challenging to have electronics on curvy substrate. So one way to do that is to realize it was to why not convert a curvy surface into a planar surface, realize electronics for that, and then reconstruct the whole 3D surface again. And that example is on the screen now, which you have a spherical surface. Fuller map concept has been known for a long time. That leads to triangulation concept. So if you take 2D triangular projections, it becomes triangle, and then you can make a 2D layout. So once you have 2D layout, you can fill up the most of the parts and rebuild the whole surface again, and that leads to electronic skin. With that background, we got the 2D map of the entire body. We developed the triangular patches. So triangular patches originates from the triangulation technique. You can make big and small triangles to cover most of the body parts. And wherever we could, we place the skin on the body. This is the example on flexible printed circuit boards, and that's why I mentioned that this is based on existing technology. On the back side, you have capacitance to digital converter chip, which is shown here. On the top side, you have the capacitive, the lower electrode of the capacitive sensor. Then you place a soft polyurethane soft material on top of this, and the top layer is a conductive fabric. So you have the conductive fabric, which is acting as a top electrode. When you press it, these two electrodes come together. Capacitance changes, and that leads to the detection of amount of pressure that is applied, as well as location because of the arrays. And wires were routed through these flexible printed circuit board. These joints that you see here, these were the places where these wires were connected. And this was done in such a way that you can cut it and use it as required. So for example, if this triangle is not needed, you can cut from here and here, and you can still use it. So it can be tailor-made to fit any body shape. And this was the outcome of this first experiment, where skin was implemented with off-the-shelf components, and we were able to then this experiment. This was not my experiment. My part is mainly related to tactile sensing. Here, colleagues here developed this technique where they were able to integrate vision and touch together. And that led to multi-modal sensing in robotics. This was technology. This is still ongoing process, but the problem with this technology is, number one, is related to the flexibility itself. Flexibility is limited. You try to bend these triangles a little bit more than for a lower curvature, then the soldering will come off. The chips which were integrated on the backside, they won't be functional anymore. So there are reliability questions when we talk about using skin in real life or real experiments. And to that extent, we then looked into next aspect. Can we further improve the skin? And that led to the next work, which is ultra thin chips. So if you can have chips which have same electronics on it, but is more flexible. With that intention, we started exploring this technology, and we developed the technique, which is a combination of wet and dry, mechanical grinding and wet etching. By that, we led to ultra thin chips, this is a wafer scale process. On the silicon chips, which are about 12 to 13 micrometer thick, they are embedded in polyimide, and you can have access to contact pads. In this case, on the figure you see dummy wafers, there's no electronics on them. These are the samples where only electrodes were present, but now we have more samples more devices where active electronics is also present. So you can have access to contact pads, and we do have, instead of using direct bonding, conventional bonding, we just use printing techniques to have access to bonding pads. And this is the outcome. You can see the same silicon, which is brittle, and you try to bend and it cracks. When you thin it down to about 10 to 15 micrometer, it becomes ultra flexible, and you can advance the electronic skin approach that I presented earlier to a more flexible version, where you can even think of covering the fingertips or finger, and curvature smaller than fingers as well. But this is not the end. You have to look at the large area concept. So the chips is our interesting point, but if you have to implement this chip to a large area, then economy also comes into picture, and that's where we have to look at new strategies. And this is where we started looking into printing of new forms of materials, and new forms here are essentially nano ribbons, nano wires, so essentially nanostructures. One technique in this regard is this transfer printing, which is pioneered by Professor John Rogers and others in US. So they want in this regard, you realize silicon on the silicon wafer or any other wafer, you realize the micro nanostructures on the wafer, lift them with PDMS, and then transfer on the final receiver substrate. Once you transfer them, you got the doped wires transferred. After that, you can do metallization, etc. and etc. on the flexible substrate and get device, and you can use the same layout strategies to develop the electronics. So these are some of the results. This is nanowire transferred on polyimide. This is using this approach. Some devices also used as a skin tattoo. But there is other technique also which does not involve these PDMS in between, and that's direct printing of nanowires, and we call it contact printing. In this case, you grow nanowires vertically. Here you have horizontal nanowires, and once you have these nanowires, you bring the receiver and the donor substrate in contact at certain pressure and certain speed. When you drag it, you transfer the nanowires on the substrate. Once you have nanowires on the substrate, you can consider it like a top slice of your wafer, and which is now transferred onto the flexible substrate. It's ultra flexible, and you can use this top layer, the semiconducting layer, now electronic layer, to develop rest of the electronics as you do in standard form. Now, in this case, because it is nanowires, these are all single crystal nanowires, performance will be far better than organics. It is still not at par with the conventional electronics, but somewhere in between. We calculated this would be good enough to lead to transistor switching frequency of about 1 gigahertz, which is where most of the flexible electronics applications, they require lower than that, and that makes it more attractive when it comes to eventual use application. In this case, you can rely on the single crystal silicon, and the performance can be really at par with the silicon. So, and then you can use this approach to go to large area. You basically are lifting from one part to other, and you can transfer them wherever you need. So that takes you large area, which can be larger than the wafer size standard wafer available with you. Here is one example using transfer printing approach where we developed this device. The mobility of device was about 650. And the performance was quite nice. This was an NMOS device. We have results I've not included today. We have PMOS, and very soon we'll be publishing papers which describe with CMOS circuits with this approach. And that would be, in my view, the first such result where CMOS circuits using flexible electronics will be reported, and the performance at par with silicon. When it comes to the contact printing, you can think of this as a step where you can consider, instead of stamping, picking and placing in large area, you can transfer, you can print directly over large area by defining sticky regions where you need wires. That pretty much comes from your layout of your electronics. And then you may have wires either on the rolls, or it can be on the planar substrate, and transfer them on the to form the electronic layers. You can have the high K dielectric and deposit. If you look at ALD, they are compatible with the compatibility with roll-to-roll technique is there, and then you can develop more devices such as 1T1M structure shown here in this diagram. This way you can develop all the electronics on the flexible substrates. We do have some examples using contact printing using this approach where we developed our custom setup. And these are the areas where the gray areas, the bright area that you see here, these are the areas where nanowires were transferred. We transferred printed zinc oxide and silicon nanowires. You can see that in the direction of movement, there was a good alignment. Alignment is very important here because performance pretty much depends on the alignment. And one example where these were, in this case, there was no transistor, it was simple resistors. Zinc oxide example was shown here. So these wires were transferred, and then we demonstrated this for variable dosimetry. Please, can somebody advise me about the time? Anyway, so I'll take this. Can you hear me?
Yes, sir. Sir, you have 10 more minutes.
All right, thank you very much. So this approach we are taking towards. So we are using this approach to develop electronic skin, where you have on the back plane you have the electronic layer. Scheme is shown here. You have nanowires. From nanowires you can develop the transistor by having a source and drain line and gate line here, and this can be on one of these the source one of the side you can have pressure sensitive rubber, which is essentially a capacitor or a resistive sensor. So that leads to this kind of configuration where you press this layer, that leads to change in the current in the device. The ID will change, and change in ID would reflect the touch, or this could be placed here. The capacitance could change. So instead of this, you can have on the gate area, you may have either a variable capacitor or pressure electrode, the capacitive electrode that I presented earlier, or you may have a piezoelectric layer here to get different dynamic or static sensors. And some of these could then also be used as a part of readout electronics. So this way we are developing electronic skin, but this is not the only thing that we are. That just leads to flexibility, large area. You have to. I mentioned about softness also. This is where we are exploring the soft side, where we have been able to integrate inside the structure the sensors. So there is no soft robotics at this point, where the sensors are seamlessly integrated. They are distributed like our skin is. In this case, the structure itself is a sensor, but the structure can move as well like insect. So that's the thing we are exploring. We are also exploring integration of these technologies with 3D printing, and doing 3D printing in such a way that you have integrated sensing and actuation. So as an example, we have developed this hand here, which prints the PLA, and then you print the conductive material. So we have developed our own custom setups which can do multi-material printing. So you can print the conductive layer, and then you print dielectric, and then again conductive layer on top, and then rest of the PLA, which leads to the structure itself becomes a capacitive sensor. So we are also exploring it to improve the robustness of the structure. Instead of placing electronic skin on the outer surface, the structure itself can become sensor in this case. So we are, in that sense, you can call it material integrated sensing, that takes care of several things that I mentioned earlier. For example, wiring issue is partly resolved here. You are not You are using structure itself as a sensor, so you don't need dedicated wires which are outside. So this sensor is not in direct contact, so it improves the robustness also. And electronics can be also housed inside the structure. The 3D printed hand that you see is just an example on all the phalanges that you see here. These are these capacitive sensors that they have been turned into capacitive sensors, and these are the responses. In terms of intrinsic computing, I mentioned about printing in nanowires in an electronic format. And one example is shown here where we, instead of using one gate, we develop using nanowires. We are developing multi-gate transistor, and this is reported here in Frontiers in Neuroscience. So multi-gate transistor can have, you can view the functionality similar to a synaptic junction. You have multiple inputs coming in, and there is summation takes place. The floating gate ensures that there's a summation takes place. These can be excited simultaneously, or one can be excited, others are disabled as needed from different inputs. You can consider input directly coming from touch sensors. So based on multiple touch, if this threshold, if it crosses the threshold, then the transistor will work and it will pass on the signal to the next stage. You may also consider putting memristor here to make it to bring the dynamic, changing the weight dynamically. So those things, those things are all we are exploring. Example is shown here how we do it. So 6x6 capacitive sensor array is shown here. In this case, the transistor part is not shown. You can assume there's a input coming here, and all this thing is this part that you see here has been implemented in software. So basically what happens, you get multiple inputs coming in. There's a summation takes place, summation of weighted inputs. Weight is decided by the capacitance. You can see the dimension of these capacitors are different. So these are hardwired weight. We are currently looking at the ways to dynamically change these weights, and for that some circuitry are brought in between sensor and the gate terminal. That way you are able to then scale down the data. You are able to handle data more effectively, and this is also efficient in terms of power. So what happens then, why I say the data we can handle, because simple one touch you can detect multiple things. You can detect point of contact, you can detect the direction of the contact, and you can detect the location of the contact. Otherwise you have to send all the data to a computing unit, and you'll have to extract this same thing you have to extract this from the data. Our body is pretty the way sense of touch works in humans. We do distributed computing, and only high level data goes through high level processing to the brain. Otherwise it is scaled, it is scaled at every stage, it's scaled down at every stage. And it would be difficult to send all the data also because of two communication channels that our body has, and there's a limited capacity. So because of bandwidth issues as well, it would be difficult. I'll quickly touch this point and I'll stop after this, which is the energy autonomous skin. I mentioned about energy. This is an important aspect which has been blocking the electronic skin research. Because people consider this is a large number of sensors and associated electronics would mean it's a current sink. So sorry, energy sink. In that regard, we developed first thing was that we developed graphene based touch sensors, and the intention, the reason behind using graphene was the transparency. If you compare most of the energy harvesting options that are available, either piezoelectric, pyroelectric, triboelectric, solar, etc., you will see that solar is clearly the winner in terms of the energy that you can generate, and the technology also is also quite mature. But for solar cells to work with the touch sensor, you need to cover solar cell with something that is transparent, something that is sensitive, and that brings graphene. But then skin also has a requirement of large area, so you need graphene on large area. So we developed, first aspect was to transfer graphene on large area. We achieved that, and after that we using blade cutting, we did not use lithographies intentionally here because of damage it causes to graphene. So using mechanical automated blade cutting tool, we got these structures, so coplanar capacitors, and we got quite sensitive with respect to the similar devices obtained from other metal. You got quite sensitive sensors, and then integrated in on these sensors were integrated on flat solar cells as well as flexible solar cells later on. And we noted that graphene allows 98% of light to pass through. So that's the small loss that we have in terms of output. Otherwise, solar cells were generating more power than required, and then the additional power was also used in the supercapacitor underneath the solar cell were integrated, and they were able to store the additional energy. This example shows the importance of skin again. So the graphene skin is integrated on the hand, and you can see when the tactile feedback is disabled, it squeezes the soft object, but when you have tactile feedback enabled, the interaction or the grabbing is much better. Of course, there's a lot more to be done. And this video shows the solar cells, and under solar cells you have the supercapacitors. So all the energy that was developed, that was generated by solar cells is stored here, and then supercapacitors are then used to move the motors in the hand. So this was first example where we demonstrated that electronic skin can be an opportunity to not only power the skin, but also to use some of this power to operate the robotic hand. And that has an implication where robots for battery power robots or robots in unstructured environment, you can consider skin to be a way to extend the lifetime or operational time. I'll skip this part. This is one example of a triboelectric energy harvester. So from partial skin, we have now moved to the skin which does not consume any energy. Previous graphene-based skin has a touch-sensitive layer on top of solar cell and requires 20 nanowatt per centimeter square. But our new result is that we don't need any skin, but we can use solar cell itself as a sensor. And the principle is pretty simple, that if you cover a solar cell, it will stop generating energy. If you don't cover, then it will generate energy. So that means it's an indication of touch or no touch. Depending on the shadow, you can reflect the area of contact, etc. With that principle, we developed this prototype for the palm of robot that's on PDMS. The solar cell, these are all commercial solar cells. So miniaturization of solar cell, integrated them on PDMS, and this array of solar cells was then integrated on robotic hand. And we also integrated IR LEDs, which were also powered by these solar cells. So that was the intention, was IR LEDs was that so that the skin can also be used during dark time when you have no light. So this was developed. We noted that the palm area alone could generate 380 milliwatt. And if we have to scale it to whole body, which is if such a skin goes to human body, which is about 1.5 meters square, then you should be able to get more than 100 watt, which is good enough power to operate many of the motors on robot. So we also use it for object imaging. So this is, you could view this as a skin that can see the object coming. So it's not vision is not as good as camera, but because of the object imaging, which is possible due to shadow, you can see what type of object is coming, and then robot would be able to plan even before there's a contact. And these are some examples here shown where you have this kind of tool. You can see these are the areas which are not generating energy, and that's you could extract that, and that's on this basis action could be taken. These are some videos where solar skin is operating in shadow mode detection. This is the solar skin. So when somebody moves the hand on top of this, you block the light, and you can see they are not generating. Rest will generate the energy, and this was then used in the final prototype. Here we show the robotic hand integrated with solar skin, and this is used for shape detection, the example that was shown to you earlier. So you have a spherical object coming in, and you can see that these are corresponding the image. You can see the circular thing, and you can find the shape as well. So that's the linear shape. So that gives you a clear indication of the object that is coming in contact. So here is an example of solar skin. We call it solar skin because of the solar cells. So that's the proximity. If something comes in close proximity, that also can be detected. Here is an example which is grasping using the tactile feedback from these solar cells. So once you have in contact, robot is able to use this feedback from solar cells to move the motors and to complete the desired task, which is the grasping of object. And finally, an example of human-robot interaction, which is safe interaction. I started with this example, how the robotics is evolving in industrial setting. So you can consider this type of skin to be wrapped around these continuously generating energy for to operate some of the electronics or some of the motors on the robotic hand, and yet making it safe. So these are some examples that we have developed. I think in the interest of time, I'll just, there are several other examples that I could talk about, but in the interest of time, it's better I move to the last part, which is conclusion of my talk. Yes, so I would like to now conclude my talk by saying that tactile sensing is an important topic, which is important for the safe human-machine interaction. And electronic skin is a technology which provides this tactile sensing feature. It's an underpinning technology to advance several application areas, which include prosthetics, bionics, healthcare, Internet of Things, wearable systems, and you can connect to smart cities, etc. In fact, some of these could be even extended to applications such as environmental monitoring. We use printed electronics as a vehicle to obtain electronic skin, and in my view, that's another interesting dimension which will lead to in future cost-effective and eco-friendly route for manufacturing of high-performance electronics, much needed in several areas. I would say that at this point, I would also like to add that it's not something that I mentioned about silicon. I mentioned about high performance, but I did not mean to say that organic is not important. In my view, they both work together. There are some, they have their niche areas where they are better than each other. So a hybrid technology where both organic and inorganic semiconductor can work and can be integrated will be an interesting dimension. And as a whole, I would say that flexible electronics is a disruptive area. I gave an example of how it is advancing robotics field, but there are several other areas where it will have enormous impact. I mentioned about consumer electronics in the beginning. Aviation is another area, space electronics, so robotics, life science, military applications, etc., and sustainability too, from printed electronics point of view. So with this, I would like to thank my funders, EPSRC, European Commission, Scottish Funding Council, Royal Society, Royal Academy of Engineering, and of course my group members. And that brings me to the end of my talk. Thank you very much.
Thank you, sir, for your valuable talk. Now the participants can ask their questions.
Yeah, good afternoon, sir.
Hello. Well, I can hear you.
Yes, sir. My question. I'm Vimal Kumar Singh Yadav. My question is that if we use printed magnetic materials in your e-skin devices, so how it is going to affect the performance of the sensors? So in that direction, if you can add something.
Well, I would not say that, we have not used so far, we have not used any magnetic material in electronic skin, but using magnetic material is an important direction because it adds one more, it would be possible to detect more, let's say, parameters with magnetic sensing as well. It all depends on, there are challenges, there are problems, some cases when you are dealing with materials which are, for example, metallic materials, your sensor may not work very well. It is just that, as I was mentioning, you have to think about variety of sensors working together. And in that sense, if magnetic is not working, then you have to rely on other technologies. Eventually, it comes back to the application. If the application requires handling of objects which are non-metallic, then magnetic sensing would work very well.
Thank you, sir.
It's again connects to what I said. There is no unique solution for electronic skin as we have for vision or audio.
Yeah, fine, sir. Thanks a lot.
Hello, sir. Very nice talk. I am Udiptya. So I wanted to ask you that you have been working on wearable biosensors. So like what are the current challenges this field faces, and like what is how can it be commercialized in like solving these issues?
Well, your question has several dimensions. First is today I did not have much time to cover the biosensing part. That was one aspect. Coming back to the question, what are the challenges? Most of the important challenges, the reliable data. So you have so many sensors that are integrated on wearables. So many sensors you require conformal contact, but you cannot maintain that type of contact. You have even Apple watches, etc., which are quite sophisticated in terms of, but sometimes they also give you errors. So reliability of data is a challenge. In terms of commercialization, you can always, I give an example of Apple watches. There are several other wellness devices which are available. So there is already some commercialization happening in wearable biosensing sector, but using them for real clinical application remains a challenge because there is no certification so far, and that's a lengthy process. So that's also something from commercialization point of view you need to consider.
Sir, actually, those are most of the commercialized devices currently are basically physical sensors.
I said they
Sir,
are well,
yeah,
not for purpose.
anything like sort of electrochemical or that sort of like what are the challenges in that particularly in systems like that?
Well, I can cite an example. We have been hearing about contact lenses for several years with the biosensors integrated on contact lens. It's an interesting problem because it's a spherical substrate. You have to integrate electronics on spherical substrate. You also need to take care of the heating, etc. Because if you put a contact lens on eyes and if the sensor during operation you have heating, local heating, it may actually damage the soft tissues. So that example is there, and there are biosensors, pH, etc. have been detected, but the correlation between the body fluid and the blood is the gold standard. So most of the techniques are on blood. So correlation is poor. That poor correlation does not lead to guaranteed, or let's say outcomes which you can rely on, and that's where the problem lies. So sometimes it's the integration, sometimes it's the reliability of data, and reliability of data also includes the correlation type of thing. So you have to have a good matching with the standard techniques which are based on gold, on the blood.
Okay, sir. Thank you very much, sir.
Thank you, sir. Now, moving to our session. Now we have with us Dr. Debashish Datta. Dr. Debashish Datta is the director of RR Animal Healthcare. He has done his bachelor's on veterinary science and animal husbandry and management from IIM Calcutta. He has worked with Glaxo India Limited and Fort Dodge and is presently in Pfizer. He is in his entrepreneurship journey for more than a decade. His categories of activities comprises of research, manufacturing, and marketing. His innovations and executions include environment-friendly pest control solutions, antibacterial and antiviral solutions, litter or waste management solutions, and diagnostic solutions. Now, I would like Dr. Debashish Datta to take over. Sir, we welcome you to join us and give your valuable talk.
Thank you, madam. I hope I am audible to hear. Hello.
Yes, sir. It's audible, sir.
Okay. Thank you so much, and thanks for introducing me to the group. So, as Pranjali said, I am Dr. Debashish, and I'll talk on a various of topics actually, but most of the talks that we will be talking today is on a practical commercialization point of view, because we are basically a commercial organization, and we believe in working, or we believe in researching from practical commercialization possibilities point of view. So with me, I have Dr. Kole. Dr. Kole is he heads the research and development in our company. So some part I will present, and a part, the technical part, will be presented by Dr. Kole. So with this, I'll go to my presentation. Next. Church. Yeah, is the presentation visible? Hello.
Yes, sir.
So yeah, as I'm Dr. Datta, and with me Dr. Kole are there. So we are, if you see, the company is based at Hyderabad, but we are also there in Research Park IIT Guwahati. We are incubated, or we are a part of the research park that is there in IIT Guwahati. So this is about this is the front page of our company. If you see the name of the company, it is RR Animal Healthcare Limited. The name itself says that we deal with animal healthcare. When we tell animal healthcare, we mainly work with poultry, we work with aqua, and we also work with ruminants. I'm sure that I'm sure that many of the guests in this room must be hearing primarily for the first time, or very new to animal health segment. So I welcome all of them. I'm sure that this will be a different kind of presentations, maybe for many of you, maybe new for you, and maybe interesting for you. I hope so. Okay, our discussion segments we have divided into three. First, we'll talk about our company that we manage. We are into animal healthcare and also into human healthcare now. Then we'll discuss about the research work that we are doing regarding analytically more sensitive IVD development. And last part, I thought there may be many students in the group, so I have added few slides on entrepreneurship, like my experience and understanding on entrepreneurship in India as an Indian context. Okay, this is about the company. We started our journey in 2015. The company is headquartered in Hyderabad. We work all over India. We are incubated in CCMB Hyderabad. So CCMB Hyderabad is primarily our biological research headquarter, we say. And recently we are in IIT Guwahati also. So headquarter Hyderabad, incubated in CCMB for different research works. And if you see the works that we do, as Pranjali said, we are in antiviral products. We are in different segments actually. If you take the segments, went to disinfection, we are hugely into sanitization. We call it biosecurity. We mostly work with biosecurity or sanitization or disinfection of animal houses. We work hugely for better environment of animals, and we also work mainly on another main important area that we work is like to provide antibiotic-free chicken or antibiotic-free food for human. So that is our major area that we work in CCMB to give antibiotic-free animal protein for the human consumption. And also recently we went to diagnostics. This will come in the presentation. As I said, there are the different products that are our animal healthcare that we manufacture in market. We are into biosecurity segment. When I say biosecurity, it is mostly about a clean environment. It is lesser bacteria possible, lesser microorganisms, pathogenic microorganisms possible, lesser cleaner water, and lesser waste possible. So biosecurity covers bacteria-free, possible virus-free, possible fungus-free. So that kind of environment we try to create in animal houses, which is very similar to kind of the work that many people do in human healthcare for this operation theaters and all. Then also we work on productivity of the animals. Like when we say that productivity of animals, most important thing is that because animal production or farming is a commercial activity, so productivity is an important segment on that. And also we work with antibiotics. Like what we do in antibiotics is that this is a very unique work that we have been doing in animal health, and also it can be implemented in human health. What we do basically is we collect samples. We collect animal housing swabs from all over the country. We collect samples from all over the country, and after collecting the samples, we check the sensitivity of the antibiotics. Then we've divided the country into different zones, and we try to understand that in which zone of the country which antibiotic is working better. This year, what happens is that when an animal is sick, when an animal is sick, you can use a specific antibacterial product. That is how you can reduce the usage of antibiotic in animal husbandry. All of us know about the resistance issue, antibiotic resistance issue in human, and the reason of antibiotic resistance human. A part of that goes from animal husbandry. Also, all of us know that. So, in antibiotic sensitivity and antibiotic range, we work in that segment particularly. So this is what I was talking. We work on antibiotic-free therapy. So basically, what happens if you see, like, even in India or globally, if we more practically, if we talk about India, we have many research institutes. We have many scientists, like you have a handful of institutes and many scientists. We generally expect the chicken, eggs, or milk. When we talk about animal proteins, they're the primary: chicken, egg, milk, beef, and mutton or sheep. So we want those inputs to be antibiotic free. But when we practically see in the market, we expect them to produce antibiotic free, and we expect practically from illiterate people. Most of our farmers are not highly literate or not highly educated. So we expect them to produce, but they really do not know how to produce an antibiotic free input, how to produce an antibiotic free their produce. So we work on that. We are working on a comprehensive solution how India can go ahead for a safer food. This is a strategy that we adopt. Like this is called thermostable probiotic. Because probiotics, when you talk about beneficial probiotic, when you give beneficial probiotic, it can replace antibiotic to a substantial extent. So this is one of the approach. Apart from this, like we have discussed about biosecurity. So biosecurity is one, then thermostable probiotic is one. With this, you can reduce the bacterial number of bacteria around the animal and also inside the animal, with probiotic inside and with a better environment outside. So that is how the requirement of antibiotic will slowly come down, and we can go for a safer food. We do lots of bacterial genomics work. In this, what exactly we do is whatever samples we collect from all over the country, we bring those samples and we analyze those bacterial species. So that gives us a better understanding of what kind of bacterial population is there around the country and different zones to be more specific, and which antibiotic works on them. That is how we try to optimize the requirement of antibiotic usage in animal husbandry. Okay, the company, as I said, we are headquartered in Hyderabad. We have our manufacturing facilities in West Bengal and in Telangana, that is in Hyderabad. But we have offices in Hyderabad, Kolkata, Guwahati, and Karnal. Guwahati is the IIT. Guwahati is our official address for Northeast now, and also for research. When we come to biosecurity product, what happens is that if you see animals rearing is a commercial activity. Being a commercial activity, every animal owner or the farm owner, it will want maximum profit. So the animal owner will wish my animal grows very well, and animal only will grow well when there is minimum challenges from the these pathogenic organisms. So that is how the biosecurity or the bacteria-free environment, the try to bacteria-free or the minimum bacteria presence in an animal house is a very popular in animal husbandry. Every farmer, every veterinarian, they try that my farm or my area should have minimum bacteria so that the animals grow well and the animals do not fall sick. So in that way, we have been working with this disinfectants and sanitation for more than a decade now. After the onset of COVID, one of our products is RH5 Plus. RH5 Plus is a research product of RR Animal again. We have done lots of work on that. There are lots of published papers in different journals and all. So this was one of the first products that is recognized by Government of India as a start to combat COVID-19. This was in surface sanitation segment, and they appreciated the product. That they said that to minimize the spread of this COVID-19, RH5 will be a star in that segment. So that is how we started COVID actually. That is with RH5 Plus we started working in COVID. So if you see in this point after the onset of COVID, we are shifting from animal to human slowly. After this development, then had is known face for all of you, I am sure. Our respected director, sir, IIT Guwahati, and there is Professor Iyer and Professor Ghosh. So with the help of Professor Iyer and Professor Ghosh, we started working with IIT Guwahati. This is the MOU that we started with. And special thanks to Professor Iyer for helping us to get this MOU done and start our work with IIT Guwahati. So one of the basic works that we have done in IIT Guwahati was on COVID diagnostics. To start COVID diagnostic, one of the diagnostic tool that we started manufacturing after developing with IIT was the BTM, that is binary transport medium. Then also we have developed RNA extraction kit, the extraction of this COVID-19 virus RNA. This also was developed in collaboration with IIT Guwahati. You can see the logo of your institute in the packs and literatures. Then we have done this RT-PCR kits. So these three kits, this COVID diagnostics, if you see, technologically, these may not be the top of the class. Technologically, may not be the top of the class. But if you realize the importance of these kits for the society at that point of time, it was tremendous. There was a point when Government of Assam, it was very difficult for them to go ahead with the COVID testing because of unavailability of these kits. So that is the time they also requested IIT and we were there. So these kits practically helped the Government of Assam and many other governments. We have supplied a lot to the Government of Karnataka also. It helped them to maximize their testing. That's how the controlling the spread of COVID-19 in the respective states. So that is how we are into human now. We're into COVID diagnostics. We have done VTM, RNA extraction, and RT-PCR. These three kits are developed and commercialized. Most of the things you will find here are commercialized actually. Post this COVID diagnostic kits, what we thought that we can work a little more with diagnostics. So now we have taken license for 21 other products from this. It is called DGFT. So from DCGI we have taken the permission, Drug Control General of India. So all this kits. So apart from this COVID diagnostics, we are into another 21 kits. All of you can see in the slide, like for bilirubin test, HbA1c, amyloid test, triglyceride, and everything. So this also in RR Animal we have been manufacturing and marketing. All these kits, if you see friends, all these kits, principle of all these kits are very simple. All the 21 kits practically work on a very simple technology, and this technology is in practice throughout the world. This technology is in practice throughout the world and for a very long time, very long time. So all these kits we want to work on them to make it to make it more accurate, because these kits, like diagnostics, and diagnosis results, those are always plus minus numbers we have to calculate. So we want to make these kits more sensitive and more accurate. For this, we want to work with IIT Guwahati again on the principle of many of these kits. I'm sure, just imagine a particular kit, for example, hemoglobin. You go to a doctor and doctor asks you to test hemoglobin. You go to the lab, the lab guy takes your blood, they analyze, and then they give the report. If we have a handy system where you visit a doctor, the doctor can instantly, like we do for pregnancy diagnosis or this lateral flow kits. So if that kind of testing method can be developed with accuracy, that will be a great help for the humanity. So we are trying to walk in this direction with IIT Guwahati to make these kits more sensitive and useful for the humanity and the medical world. Now, apart from 3 kits that we have discussed for COVID, we have commercialized 3 other kits under ICMR validation now. I think this is a good news for many. One is magnetic RNA extraction, magnetic bead-based RNA extraction. One is rapid antigen test kit and rapid antibody test kit. So rapid antigen and rapid antibody test kits, these are very popular nowadays. Most of the states and most of the organizations, they are going for rapid antigen test kit. I'm sorry. Yeah. So we have developed all these kits again, and they're in ICMR for validation. While working on this rapid antigen test kit and rapid antibody test kit, our learnings were tremendous. So our learnings practically were that the kits are good, but they are probably not that sensitive, and they are probably not that analytically reliable. That has been our learning in till last week by working on rapid antigen. So based on that learning, we are trying to work how to make these lateral flow kits more sensitive and analytically reliable. With this, based on this learning, we are working on few technologies. Now I'll hand over the session to Dr. Kole. He will explain you the technologies and the direction that we are working. And also on the onset, I'd like to say that this is an appeal to all of you. Please see the direction in which we are working. And if anybody is working in this direction, or anybody is interested to work in this direction, we request them to join hand, and we are ready to work together. With this, I hand over to Dr. Kole to explain the direction that we are working now.
Hello, good afternoon. After approval, several kits and the practice in the different hospitals and others, which we found that the SARS virus, when it enters in the body, after 3-4 days, before 1 week, it will take for the establishment of this disease. So when it's established the disease, then there is a method for the diagnosis, that is the RT-PCR. So people take the swab, nasal swab, and can do the RT-PCR that can able to detect. But before coming to symptoms, it starts spreading. It seems that this virus starts spreading within 60 hours or below that. Okay, so RT-PCR kit works here, after establishment of the disease, and for the rapid kit works, after 1 week. So when we try to detect using the antibody detection kit, so that time already as disease has already established. So there is no meaning that you can prevent the disease and others. But what people are thinking, what I am thinking, that okay, if we can detect the disease before starting the infection, then it is a very good solution to the people. Theoretically, if I see that the virus has entered in the body, either nose or mouth, if we can and take that virus, okay, and able to detect, okay, then it's required very high sensitive methods, like the single like we have to develop a method which can detect the single virus. Okay, so for that, actually, just recently I got a paper. Recently I got a paper that somebody in the Osaka University, they have developed a novel methods which can detect a single virus. Actually, we are working in similar way. This was published in 2 days back, 28 November. So where we are working. So so if you see the lateral flow technology, that I am explaining using the ACG. When urine ACG, you add as an input, so it's bind to the immobilized gold and antibody. Then this antibody bind here another set of antibody and make a test line, and after that they have the control line. So here the reporting is the accumulation of gold here. So now to accumulate the gold, you need the minimum amount of the antigen. Okay, now for the COVID, if we take the bronchial lavage or the nasal swab lavage, okay, the amount of antigen probably will not be enough to that. Now there is a room to increase the sensitivity instead of using the gold, and instead of using an antibody and the reporting structure. Okay, so what we are planning, what we are doing. See, this is a single stage strategy. Okay, where only one kind of detection method you can use. Now, in each step, you can use all these three. Either you can you use a variable signal for the reporter, maybe the different color, or the broad selective recognition element, whether the whole part of the antibody or part of the antibody, or the whole part of the antigen, or part of the antigen, or the antigenic site only, or if you can use the different separation technique which help in the detection. So all these three combinations you can use, either spatial separation, either in blood recognition, okay, or that you can construct in the strip. Then with this permutation combination, you can use one or multiple signal, multiple outcome, and that generally happens in multiplexing levels. Okay, that's the multiplexing signals. So how we will be doing? Okay, suppose in normal ELISA, we generally immobilize an antigen. Then we use the specific antibody of that. Okay. Then we use the secondary antibody coupled with the probe. Here probe is enzyme, and then we add the substrate and give the color, specific color, and which is the readout. So here this is this is a color which is a readout which actually represents the presence of antigen or antibody. So instead of that, instead of visualizing as a gold particle, same thing we are using the color. Then the instrument will be different. That will be the ELISA reader or the spectrophotometer. Now instead of that, if we use some other technology which is called ECL, electrochemiluminescence. Okay, it is kind of in a well. In a well, you put different site for the different analytes. Suppose here you are trying to analyze 3, 3, 6, 7 analytes. Okay, you immobilize those 7 recognition analytes, and then you use 2 electrodes. These two electrode on a dielectric plate. Okay, and then you use a chemical, okay, which will produce the luminescence. Okay, and then if you read this and if you use the magnet from the bottom, and if you try to read with the PMD, then you will see the color. You will see the color, and that color represents the the detection of the antigen or antibody. This will be much sensitive than the single color detection methods. So this way, actually, we are trying to develop the COVID nucleocapsid and antibody. What we are using, we are immobilizing an antibody, and then we are using the secondary antibody coupled with some dielectric portion which we can detect through the ECL. Okay. Now this is one theoretical. Suppose in one lateral flow, we know this kind of things. If we can if we can if we can put the different spot for the different analytes. Okay, and using the same technology, the same sensitive technology, okay, then at a single go, we can able to analyze, possible analytes for a particular disease. Suppose in COVID, the cytokine storm is a phenomenon for the establishment of the disease. So here maybe the 8 cytokine is related, 8 chemokine is related, and 8 protein and other protein is related to that. So at a one go, with the different, as we have immobilized a different marker, at a one go, we can able to able to understand the what is the condition, what is the status of a patient immunogenic immunogenic property of that. So in there we can able to predict that patient is which state is susceptible for this disease, or he got good immunity and he will not get infected with the COVID. We are trying to develop multiplexing like this kind of things. Okay, now the sensitivity is the issue. Now each spot, if we try to develop the gold, that may not be very sensitive. There we need fluorescent level probe or further that. So recently the technology has come. This is called NELPIA. It's called NELPIA. NELPIA is that it will, instead of the adding the in a protein or an antigen, people add the amplicon after PCR. So what we are thinking, if we take the nasal swab and concentrate it in a microliter, maybe the one microliter. And this 1 microliter, if we use the ddPCR, that is a PCR, Bio-Rad they have, droplet rate PCR. So total PCR volume will be the one microliter or less than that. And after PCR, using the different primer, like for the one for the N protein, one for the RNSP, and another for the RDRP. Okay, and if this if this applicant, if we add this, in lateral flow pore, which is already immobilized, 3 antibody of specific tag, like this antibody for this tag, this antibody for this tag, and this antibody for this tag, and then it will bind that one, and each other primer, it is a biodynamic primer we use. Okay, and then we add streptavidin mixed with the carbon nanoparticle. So now if we add this one, it will increase the high readout, very good readout, maybe which is not colored, but we can use some device which we can read this nanoparticles. Okay, so we are looking for, who is working in this area, we are looking for high sensitive carbon nanoparticle and conjugated with streptavidin, that will very helpful for us. So this technology actually we are using now, and this is the total technology we are using. Either actually we are trying to develop with the collaboration of this different nanoscience group in the India or abroad. Okay, we are trying to looking for highly sensitive probes. Like instead of gold particle, we are looking for the nanoparticle which can sense or which can emit the electron or voltage, and that can be detected through the detection instruments or one kind of device like glucometer. So either this paper-based, chip-based biosensor, or that can be used in the paper-based biosensor, or film-based biosensor, or the graphene-based biosensor, or the thread-based biosensor. Actually, we are looking for actually sensitive biosensor. Then directly we can work on that. At this moment, this technology which we are practicing in our lab, it is a paper-based biosensor we are using. Okay, so if we do that one, after that, we have to plot the result of that, and this should be like with the any kind of technology you use, the graph should be like this kind of so tight and robust with the concentration dependent. Okay, so if we can develop this technology, it will not directly help to the detection of the COVID, but also detection of any analytes in the body which is very, very minute concentration level, and with the different biological activity, this concentration change, and this minute change we can able to detect. That is our aim of this study. Okay, so this is the total technology. Now I am handing over to the Devasis for the further discussion.
Okay, I had prepared a few slides on entrepreneurship, but I think we are out of time. Our time was up to 4:15. So maybe the organizers can suggest whether I should go ahead with the presentation, or maybe we are out of time, so we can go for question answer.
Go ahead, sir.
Go ahead. Okay. Thank you. Okay. Being an entrepreneur, I love to talk about entrepreneurship. And I thought, like, if you see entrepreneurship, this is the buzzword is in not only India, throughout the world. This is the buzzword now. So this is practically, I would love to address the student friends of IIT or any of the student friends. So this is why entrepreneurship, what to do, how to start, and financial management. Many people want to become entrepreneur, but it's really difficult what to understand key, what and how, and where from the money will come. See, friends, after academics, you have different options. You have job engagements. You can go for government job or you can private job. Everything is good, or you can go for government job or private job, or you can take the path of entrepreneurship. There are positive and negative in all. In government job, if you see, is highly secured. All of us know government jobs are secured. You have minimum work pressure till now. What I mean till now is that now many of the government departments in many of the areas, government is forcing them to work. If I can give you an example, in Andhra Pradesh, the departments they are putting that digital attendance. They are monitoring the employees through this webcam and everything. So, but in many of the places till now the work pressure is a minimum. Tomorrow it may increase, but there is an advantage: minimum work pressure. Then you have limited earnings. In government jobs, your earnings will be limited compared to other options. But you can get a planned retirement. Planned retirement is possible in government job, but availability. But how many government jobs are available in the country? And where from the government will give jobs? Government is finding difficult to manage the existing situations, existing employee and establishments. Those are only they're privatizing. So where is the availability that we will afford for government job? Because it's to being IITians, you certainly think ahead of other student communities. So you have to plan everything in advance. And government job, where is the availability and how is the future where they're already privatizing? So these are the positive and negatives of government job. According to me, it is not necessary that all of you agree to that. This is about my experience. I'm trying to tell you here. Then you have private jobs, more work. You have to work more earnings. Earnings are definitely, it can be much more only for the capable people. If you come to private, if your work produce results, earning is not a problem. Earnings will be much more secured. Private jobs are equally or more secured. I will say equally or more secured if you are capable. Because in private, if you have capability, you have research capability or whatever. If you have capability, more and more companies will come to you that please come and join us. So security in private job for capable guys who wish to work is not a problem. Corporate lifestyle. This is amazing. In private jobs, in big companies, blue chip companies, corporate lifestyle, 5-star lifestyle you can get from the beginning of your job. Then more chances of working post-retirement. In private, what happens that because you have huge channels here and there, and if you are capable, if you are still capable, 58 or 60 is not the age of retirement. Post-retirement also you can keep working with other companies or other groups. And work-family balance. This is a concern. Work-family balance you have to maintain. So that post-marriage it becomes an issue for many. So work-family balance. So in private you have more work, you have better earning, you have better post-retirement you can plan, corporate lifestyle, but you have to work more is one, and you have the work-family balance you have to maintain. So this is about government and private job. Then entrepreneurship. In entrepreneurship, the major problem is initial few years of hard work and financial risk. Initial 2 to 3 years, initial 2 to 3 years, you have to work so hard. You'll be working day and night. You must be working with very basic amenities. You will see that all other friends who have joined in government or private job, they will be ahead. They must be traveling in business class. You must be traveling in economy. So initial few years in entrepreneurship work is you have to really do very huge hard work. Initial few years can be compromised lifestyle. Yes, because entrepreneurship means you have to, in most possibilities, initially few years, at least 2 years, You may not have a very good income compared to your friends. So compromised lifestyle is a possibility for, but better earning of money. Money is like entrepreneurship can plan as per your wealth requirement. How much you want, you can plan. Money will be much higher. Flexible working as our own schedule. This is a possibility in entrepreneurship. Once your enterprise is ready, once your enterprise is marketable, once your enterprise is doing business, then more flexible. Then it's not warming, it's walking. Sorry, okay, yeah. So you can schedule your own work. You are in Guwahati or Hyderabad or Singapore or New York. It doesn't matter. You can plan yourself. Life at your own terms. Entrepreneurship allows you, like, okay, suppose you have a particular you have 50 million of your turnover. You can, from 50, you may say, no, yeah, 50 is good enough. I don't need to work as hard. Or somebody can say that, no, I want to make it a $500 million company or $1 billion company. They may continue to work. So life at your own terms, whatever you want, you can do. And one more thing, being a responsible citizen, India needs more entrepreneurs actually, because our unemployments are high, and jobs are required. Now, coming to entrepreneurship, the major problem, what I understand. This is, again, I'm telling to all of you that this is what is my understanding. Not necessarily everyone will agree, and it's true. Selection of industry, like. All of you are highly educated. What kind of industry you will select? According to me, there are two types of industries. One is highly challenging. When you say highly challenging, you have IT, you have automobiles, you have healthcare. Now, friends, you tell me you are a fresh graduate, or you fresh graduate master from an institute. You have some idea. You want to do research. Do you really think that it is possible to compete with the guys like Microsoft or Google or auto industry, somebody like Tesla or Toyota? So those are highly challenging industries, and those industries, in highly challenging industries, so much of work is happening throughout the world, and there are so big established players that you try to do something, they must have explored most of the things actually. And you try to do something, there are every possibility that tomorrow they come and they smash you. But the benefit of highly challenging, that if you really can do something which the big guys have not thought or not yet touched, if you can innovate in that, then you are through. You may not require to work in your lifetime again. So in highly challenging industries, these are the positives, and these are the negatives. Positives are if it touches, if you can, it can be huge.
I didn't have it on this low wall facing the house.
Then you have
I had it on the front of the house,
working
facing
areas.
the road,
Especially
and the cars
like
must be
this,
40, 50 feet away, and
on
yet
this, that
every single
traditional
car
means, I mean,
was
agri or
triggering
agri-allied industry.
this sensor. Basically, it's just
Now, how
far
many
too
IITians
sensitive.
or how many
But even in this
guys
position
from
here,
top
this sensor
business
is
schools or
far
top
too
educational
sensitive,
research
and the
institutes,
light's
they
just
want to
coming on
work for
all the time.
industries
So I
like
need
poultry
to
industry,
decrease the
working for
sensitivity of
poultry,
this
working
sensor. The
for
only trouble
dairy,
is,
working for
it doesn't
toy,
have a
handicraft.
sensitivity part.
Most of the times
Now,
you
at the
are
moment, this
totally
PIR
focused on
sensor and the little light
Samsung, Tesla,
are running from
Toyota,
a
and
12-volt
those are the
lead
blue
acid
chip.
battery. In fact, you can
In those
see
companies, you have lots of
it's
financial
disconnected at the
engineering. Those
moment
companies
because
will be there in
it
the
wouldn't take long to
huge
flatten
in
out
the
with this thing
share
on all
market.
the time.
One day $1 billion
That's
goes
another
up, tomorrow $1 billion
story.
comes
I'm
down.
kind of
Those
intrigued
kind
by
of
the
industry
possibility of a
we keep
remote
listening in the
battery,
newspapers and
but
all the
connected
media,
back to the main
and
battery
all of
bank
us are generally
through fairly thin wires. I
everybody
want
will
to
be,
investigate that. But
everybody's
the
mind will
first
go up with
priority
blue chip companies.
is to get the
But
sensitivity of
friends,
this thing
there are other areas.
down.
When you say
And as I
other
say,
areas,
there's no
it's traditional
potentiometer in there
companies.
for sensitivity.
Agri or allied
There are
agri,
two other
forestry,
pots, one
toy making,
for
handicrafts.
the amount of time the
Now I have
relay
written a
comes
few
on,
numbers here.
and
Poultry.
one for the light
Most
level
of the IITians or top guys
cover.
may not consider
Living in
poultry
here
as the
to live
area to work
out
for.
on the woody
But
bit,
the size of the
not in
poultry industry in India, 1.4 lakh crore, that's $20 billion industry.
but
The dairy
clockwise.
is a $150 billion
Sensitivity
industry.
of the PIR.
What is the textile industry in India? Around 150, $180 billion industry. What is the auto? It's a 200, 250, including everything. So what I'm trying to tell you is that these traditional industries like agri or allied agri industry, size of these industries are equally big. These are very big. Point number one.
Into this
Point
input
number
here,
two,
and
not many people
there's
are
mention
working
of
towards
in
innovation or
negative
entrepreneurship
one, in positive one,
in this
input
segment.
resistance
So
as well.
possibilities or chances of
So,
success is
and I'm
probably
thinking,
much
looking
higher,
at this stage here, now
and
they've got
entry barriers are very
be negative
much
feedback, and I was
lesser.
thinking,
So these are the highly challenging and they're the traditional industries. You have to select the industry where you want to, which segment you want to go. After going to the segment, you can
positive
think
input
about the
first stage op amp. So in positive.
So the direction of product line, you know, when it can be product or it can be service. You know what?
Grammar and
when
spelling
we think
are
that
important,
we want to do
but if
business,
you
we want to
want to
establish
write
our own
essays that
enterprise.
inspire,
What kind of product line
Circuits
you will
are
have?
very
It can be
tight
highly innovative.
pins.
So it can be highly
solar couple of
innovative.
390K.
So
Very similar.
Again, if you want to make it highly innovative, what is the percentage of success?
On the
Will
high
be less,
pins,
but if you can get, it will be huge. So whether it is possible to make a highly innovative, because you know, all entire Europe, US is behind innovation. So whether you want to go in highly innovation segment, that you have to think. And next is improvement of the existing system.
This all from the wrong angle.
You know, limitations and opportunities in Indian trading context. Okay, what I'm trying to tell is the improvement of existing system. If you see, you know, we go to dentists. I give this example. We want to go to dentist.
And now I have
even today they will put you this, you know, anesthesia, and they will pull the. It looks very cruel. It looks very cruel, and there are areas of work. So as I explained earlier, we are into 21 diagnostic products now. New diagnostic products, all those are decade-old technologies. There are not much working happening on the existing technologies or existing system of works. So there are lots of scopes that are existing system. There are lots of improvements or possibilities. You know, friends, one more thing, what happens in Indian context, in Indian business context, like if you see the Indian businessmen, they're trading communities or trading sentiment. So whenever in Indian market or Indian business circles, there's a product, you'll find a word like, no, it doesn't work out, pricings are high. It doesn't work out, pricings are high. Those people who says that these prices are high, this doesn't work out, I cannot sell this, I cannot do those business. Most of those people cannot find an alternate way of doing that. What I'm trying to mean is that there are many things which we import. In India, why do not produce? Because when we produce in India, the costing goes up. But what the country expects from IITs or people like you, guys like you can work and you can find an alternative way of doing the same work, and that is how the costing can be possibility in India. So this is one, the existing thing, doing it differently to make it cost effective and to take it to enterprising. Number three is working on need of poor and developing country. This is a high challenge according to me. You know, most of the innovations are generally happening in US and Europe. All of us should accept that. Now you have countries like India, and subcontinent, Indian subcontinent, or many Asian countries and African countries. If you think the countries like India, Pakistan, Bangladesh, Nepal, Africa, Afghanistan, and all these developing countries or poor countries, from these countries, entrepreneurship is not much. Number one. Number two, the developed countries do not understand, in most of the cases, the dynamics of these poor countries. Like our challenges are cleaning, hygiene. Our challenges and the challenges of European can be different, or it is different. So there is a huge work area. There is a huge work area in different segments which are needed in the developing countries. And this segment, country like India can do miracle. Can do really miracle, because China is also developed countries now. All are working on those development top things. Many of the things are not suitable for India, and many of the challenges, there are no solution for developing countries challenges. So that can be a huge area of work. So you have to think, you will be highly innovative, you will go, or you will go development of the existing systems, or you will try to find out some problems or many problems the developed countries are facing, because Europe and US are not working mainly on those things. So how to start? If you want to an entrepreneur, first you select the industry. When I say industry, you want to go to blue chip industry, or you want to go highly populated, very competitive industries, or traditional, less competitive industries. Chances are, that is one decision you have to take. Determine area of work. You want to do highly innovative works, or you want to improve the existing things, existing systems to make it enterprisable, or you want to do for developing countries. That is area of work you have to do. Then analyze skill and financial requirements. Innovations is only 10% of successful enterprise. This is a very important thing. Many of you may not agree, but this is true, friends. Okay. Any innovation, you innovate something, there is only 10% of a successful enterprise. 90% is all others. For an innovator, he only innovates. But when you become an entrepreneur, apart from innovation, what you need, you should have your, because you have to run a company, you need to know how to deal with people. That means you need HR skills. You need to know, example I am giving, you need to have finance knowledge, because you have to run the company. So that you have to, you need to have taxation knowledge, because you have GST, sales tax, income tax, and all those things. And you have to be accounting knowledge. You have to have, because accounting is a part of your business. You have to have manufacturing knowledge. You have to have civil knowledge. You have to have laws of the country knowledge, because if the government official will come and they say, why are you doing this and that? You should know the law of the land also. So when you go for enterprising, you need a set of skills. Apart from innovation, you need all other things that are required for a successful enterprise. So that is how you have to make a team accordingly. It's not only innovation, other things also you have to think, and the team building should be accordingly. And okay, team research side, this has become particularly good now in country like India, because government of India is startup they're promoting. So you have multiple CSR institutes, IITs, thanks to IIT Guwahati and especially Professor IR. So we are also there. So you have these institutes now who give you site to research. Actually, you can sit there, you can do your work, you make your product or service ready, you come out and go to market. So this is how you have to start. Finance arrangement. This is a very, very tricky subject, and this is a day-long subject. You can have your own money, always better. Number two is partners or relatives. You can borrow money for from your partners, or partner is not wife husbands. Not like if you have 2-3 partners in the company, the partners can share, and from relatives you can borrow for the time being. Third option always should be bank finances and government research finances. Government is doing lots, like BIDEC and all. They're giving lots of money nowadays. So this is how first 3 are the main source of finance that you should think to start doing. And if you have a disruptive technology in your mind, then venture capitalist. Always remember venture capitalist. When you approach them, capitalist, not a capital, venture capital, yeah. So when you approach them, they are also into business. So they will try to get out the maximum penny out of innovation, and you may or may not be as capable as a venture capitalist will be. So the other different finance arrangements you have to think about. So yeah, this is what I wanted to speak about entrepreneurship. I'd like to end here, like, if anybody has a marketable technology that they think RR Animal Healthcare and Spill Healthcare Limited, innovative technology in healthcare segment, I mean, if anyone has anything, we will be more than happy to discuss with those innovation partners, or anybody want to work in healthcare innovation segment, we will be more than happy to hear from them and work together. Thank you very much. Thank you so much. With this, I conclude my discussion today. Thank you.
Thank you, sir, for your valuable talk.
Thank you, ma'am. Okay, thank you, madam. Thank you so much.
Participants, if you have any questions, you can ask.
Dr. Datta, this is Parmeshwar. Nice talk.
Sir, good evening.
Good evening. Good evening. Good evening. I don't have a question. I want to request you one a small thing. Probably you should discuss or tell people. What are the little challenges that we have been facing. While in the commercialization or in developing this technology, or probably if you have some advice to young entrepreneurs, that is probably quite important. Especially in the Northeast, when you're doing something here in this region, I think that will be very useful for them.
Sure, sir. Northeast is a land of opportunities. Actually, when you see Northeast, it is a clear land of opportunities from quite a few parameters. One is natural resources. You have abundance of natural resources in Northeast. Number two, it is geographically also it is blessed in the sense. From northeast is the corridor for entire east. You have Myanmar, you have Bhutan. Okay, Bhutan is 6 lakh population, but you have Bangladesh. So northeast and neighboring countries itself is a huge market. The challenges that mostly an entrepreneur who wants to start faces is most of the time, and a person want to develop a highly innovative solution or highly innovative product, and most of the cases he finds like by the time he is working, many others are working the same globally, and competing with the global biggest is really difficult. So most of the time I find the entrepreneurs or the technical entrepreneurs fail in the selection of the segment, actually selection of a suitable segment. So that is the number one challenge. Number two challenge, as I said, once innovation, once industry is correct, sector is correct, then your product or service is also ready. The next challenge you will find that the marketing the product, marketing. You know what, in industry, academics and industry, this fight will be always there. In industry, your product is good only when the customers accept it. When you go to market and customer accept and customer repeats it, customer appreciate, then only your there's no amount of research papers will be helpful in marketing your product. Only your product has to speak. Only your product has to speak, nothing else. So marketing is a very important thing. Taking it to the customers, actually taking it to the customers with appropriate or correct messages. That is the second challenge the entrepreneurs face. That taking it to the customers with appropriate message. Number three, where they fail is most of the entrepreneurs. Most of the entrepreneurs, what I see on my experience is that many of them are not very good in accounting and finance. So they make a mess in that. Actually, many of them make a mess in that. So that is how that leads to failure of many budding entrepreneurs. So that's why I told in my presentation that one is segment marketing. I did not cover. Yes, one is a segment. It should be. You know what? You can always start with the traditional segment. So traditional segment, establish yourself, stand, then you go for your dream. Possible. But if you go for innovative and blue chip dream, you may fail and your enterprise may never start. So alternate is either you work for traditional or you start with traditional. Traditional is damn easy. And there are so much of blooming countries like India with 135 crore of people in population. You can actually not a problem. Yearly handsome income is not a challenge at all in traditional industry. So you can come to traditional, you can start a traditional, that you can go for a blue chip dream. So one is selection of the industry and the segment. There people fail. Then what I feel, sir, is like taking the correct message to correct people in correct way. People fail. Number three, what become more challenging is finance and accounting. So these three, I will say, the major areas of work when someone wishes to become an entrepreneur or wishes to make an own enterprise. Thank you, sir.
Any more questions? I think there are no more questions from the participants.
Okay.
Thank you, sir, for the interesting talk.
Thank you, Dr. Datta.
Thank you once
Thank
again.
you very much.
And
Yeah,
thank
I request you to be online itself. I am also here. Professor Ghosh will also be here. Probably, the questions that will come up for like a panel discussion. I think, if you be there, it will be good because we may discuss the same things, what we are doing together. So I think it's going to be
yes,
useful. If
myself and Dr. Kole will be here.
Yeah.
I would like to big thanks to the organizers for inviting us for this this session, and big thanks to the director sir IIT Guwahati for accepting our request to work with IIT Guwahati, and thank you IR sir for all the help and your dynamic help with it with dynamic, and thanks to Professor Ghosh for their always with the technical helps. Thank you so much, everyone.
Good.
So now we would like to move on with our next session, which is a panel discussion by Professor Siddharth Shankar Ghosh and Professor Parmeshwar Krishnan Iyer and their team members. So Professor Siddharth Shankar Ghosh is professor in Department of Biosciences and Bioengineering and Center for Nanotechnology, Indian Institute of Technology, IIT Guwahati. Professor Ghosh works in the area of cancer therapeutics, nanotheranostics, exploring new areas such as the EMT dynamics in microfluidics, exosomes, and notch signaling in cancer. Professor Parmeshwar Krishnan Iyer is professor in the Department of Chemistry and Center for Nanotechnology, Indian Institute of Technology, Guwahati. Professor Iyer Group is focused broadly on designing and synthesis of functional materials and exploring the physical properties of novel advanced materials, and to explore these structures for application in organic electronic devices, chemical and biological sensors, and brain therapeutics. Professor Parmeshwar K. Iyer and Professor Siddharth S. Ghosh has recently made an immense contribution in the fight against COVID-19 pandemic by developing high quality and affordable sterile SPILD VTM kits, RT-PCR kits, and RNA isolation kits. So now I would like to request the respective professors to take over.
Yes, very good evening to all of you. It is our pleasure to be part of this panel discussion. I would request Professor Iyer to initiate the introduction, then I will continue with this. Professor Iyer.
Rupa and all the organizers of this event. I think it's a great opportunity for all of us at IIT Guwahati, despite this pandemic, to interact with everyone. I think, we also get to know what are the challenges. In addition to that, it was also very nice to hear various participants, discussing about their research work, as well as, these CEOs and directors from various industries. We have been very intensely working with RR Animal Healthcare over the past, around 8 to 9 months. What started off as a small technology transfer of some device has now resulted into a major, joint work in a lot of different projects. Okay, so in fact, we are also very thankful to the entire RR healthcare team. Despite the pandemic, we could do a lot of work, and we got a real insight into what is the need or what is the lacuna in the Indian healthcare sector. Okay, and what we realized is, during the pandemic, it is not only the coronavirus that was troubling everyone, but over the 7 to 8 months, we realized that, even if you have any other ailment, okay, if you have any other diseases, the hospitals were almost inaccessible. So it was necessary that we also have a lot of other kind of devices, okay, for other detection of other disease biomarkers had also become very, very important. So that has been the focus of the Center for Nanotechnology over the years, to develop various portable devices by which we can detect various healthcare or disease-based biomarkers, plus various other environmental pollutants, gases, toxic gases. So that has been the focus. So all the participants who are here in this attending this NWNTD, on behalf of the center, I welcome you all. We are here to interact and see how we can progress and how you all can also benefit from this workshop. So with these few words, I once again thank Professor Ghosh also, who is here with me in the panel, and all the organizers for this opportunity. Thank you.
Okay, great. So actually today, right now we are having the panel discussion, and I got the message that we are going to miss the tea today. So that is one thing very important. The evening tea actually there we get a lot of inputs and stimulus from different department colleagues, and it's like becoming a culture, and these have been very much benefited because during the pandemic time we really faced some of the issues that I would like to share, and then Professor will talk about that. So the first thing, how this tea in this time is important, because some ideas we get and that we have implemented during the time of our kit development. The first thing we face mainly how we'll implement the program. As Dr. Datta from RRA Healthcare rightly said, that we have to choose the right problem to initiate, but this was given by to us by NBM, that is the National Disaster Management, to the director, and then it is trickled down to us, and we committed that we will make the kit. But we did not know anything about the kit, and we did not have any manpower excepting two students. One student is Raman, who will be presenting to you tomorrow by video demonstration, and another our student is Anil. And Anil actually completed his PhD, but he was not staying in the campus. Rather, that time every student were asked to leave the campus, and he left the campus, and he was living nearby in some other places in Naiper guest house with his friend. So the major challenges, we have to bring these two persons together, because Anil knew about the biological methods and Raman knew the chemical processes, but how to bring Anil? So what we have done, Professor and myself, we used to go in the morning to bring Anil, and in the evening we have to leave him there in the Naipar, and many of the people around did not know. Obviously, head of the Nano Center knew, some of the staff knew, and the director knew. But there was, that time we faced also some struggle. There was a team called DRT. It was made for the good efforts, and they are really protective. I would say they have done really good work, so to keep us protective at that point of time. But we had to make them full, right? Right now we can say that, otherwise we could not complete this task. So both of us used to go and bring Anil and drop him at night, and our car were unchecked and unmarked, so we could do that. This continued for 15 days. Then Anil we had allowed to stay in the lab, and both Anil and Raman slept in the lab. Then RR Healthcare support we got. Dr. Pankaj joined the team. He brought some of the workers who did not have the job at that point of time. So they were basically the tea seller, they were basically the fruit sellers, and they were other people around, the bookbinders in the code. So they joined in the team, and they were first trained. Now, how did you get that? Because those people have got DRT permission to enter into the campus. Okay. So by that, they entered into the campus and we took their help, and they were really charged up to see that this kind of drive we were doing for the Northeast, and so they took part into this program. And there was a training program for them. They were trained as a microbiologist. They used our facility and they did the all microbiological aseptic work. Initially, we did not give them, but later on, they got the essence, and then they started doing the VTM kit preparation by themselves. And they help in sterilization, autoclaving, cleaning, packaging. So there's many more things they were all involved, and then some other staff members also joined in this process. So as Dr. Datta mentioned, these were the part of unskilled labor who were trained to do some kind of work, microbiological aseptic work, and they did that, and they got minimum payment whatever was possible, because we are working as a non-profit organization. And there the RR Healthcare, all the team, Dr. Datta, Labarnamoy Kole, and Pankaj, they all chipped in and they supported us from the Hyderabad and many other places. The ingredients, the chemicals, the plasticwares, everything, and most of the reagents were made in our nanotechnology center in the lab, and this was done. So why I am saying this? Because most of the students at that point of time were not here, so you may not know. We have video recorded or videographed some of the events that has been taking place in this process. So again, coming back to the evening tea, what was important at that point of time, we used to get a lot of feedback. How to break the rule, how to bring the staff, unskilled labor, how to put them in that. So that was willingly we are breaking the law. Sometimes this is whether you call it disruptive technology, I don't know, but it was a disruptive effort for the good, will of the mankind and the entire Northeast. And actually, more than 9 lakh kits were developed, and that was given on time, and that has been very useful. And these kits were again, standardized in our laboratory, and it was validated in the course of time. Only few real-time PCR machines were left. We could manage to have our own machine so that we can do the RT-PCR standardization in the process. And this validation was done, and then ICMR certification was also possible. But that point of time, we also face, I think, some issues, because our ICMR center, the main center in the New Delhi, was shut down because of the COVID infection. So we had to get it done from the NICED. You may be knowing which is what is NICED. That is the Cholera Institute in Kolkata. That actually the vaccine testing is going on, right? So this thing we have followed up. There is a student actually who is doing PhD who was involved in the validation process. We linked with him and Anil, and then we talk regularly, got the feedback, then change our ingredients, and then we got ultimately the ICMR validation in all the kits. Once we got the validation, then the commercialization part was doing, and you cannot believe, in the night at 10 or 11:30, a big truck used to come, either our own vehicle, some hired vehicle, or NBM office used to send a big vehicle. So with that, in the night curfew, this whole package was we used to supply to the NBM office, that is in a distance place. So, and from there, another truck were almost ready. So the moment it was unloaded from the truck, the same material was loaded to the another truck to be carried out to the distance places in Assam. So this was a kind of a challenging experience for us. Both Parameswar and myself, we have really, went through that time, because we never felt that students are not here. because we never had a single day off, because we were involved so much into this deliberation, okay, preparation and everything. So basically that is how we have done in the process. So that's the background history, and there are technicalities in the process of making the VTM that was followed by WHO CDC protocol. Similar to that, RNA isolation kit, and again for the RT-PCR kits, you know, two viral genome has to be targeted. One was N gene, another is the RDPH, that is the reverse transcriptase gene. It was targeted, and one human gene actually was used as a control. And there are three primers. One is Hex, FAM, and ROS. The ROS primer actually given us a lot of problem, but at the end it was resolved. So it is all because of the student effort, all because of the some unskilled labor who become skilled at the point of time, and they keep on asking. I mean, yesterday also, what has happened to your kit? So by the time the student has come back, our duty is over, the lab is free, and we have almost supplied whatever the legitimate kit is possible to the society. So that's a kind of, you know, story, I would say. I cannot talk about the entrepreneurship. I do not know the model of the business. We are basic scientists, we can do the applied research, but it gives us a confidence when the time comes, it is possible to deliver it to the society, and that is what I think is our wish, and that is what we think our student will do. So this much from my point of view. Any questions you can ask me about the, I have shared our experience. Now I will ask Professor Parameshwar to share his view and take it further forward, because he was the main resource person who communicated overnight. He did not sleep. I remember several nights to communicate with the authority, with the NBM director, with the DC, and supplied the material. So he can share the experience. Thank you. Professor.
Thank you, Siddharth. You know, very nicely summarized. I think, you know, you have covered almost every aspect of the activities that, you know, we could do during this pandemic break, and acknowledged almost, you know, everyone. You're not left out anybody, you know, right from RR Animal Healthcare, everyone, to our students, research scholars, and the laborers, you know, who were, of course, overnight, you know, they were trained to be, you know, like scientists in the lab. Okay, I think, you know, it was a nice journey. And as you mentioned, I will just take another 2 to 3 minutes, and then we will ask the participants to post some questions to us. I think, you know, our interaction also in the same way in the campus during that time, as you all know, you know, everything was locked down, IIT campus. So we could not go out even for a cup of tea. And it was a time when, you know, early morning, maybe around 11:30, we were sitting outside my office. So that was the only place where, you know, tea was available. So I used to boil water, and we used to gather a few friends. And I got a message from our director that, you know, there is an urgent requirement from the National Health Mission. The director of the National Health Mission, he contacted our director whether, you know, IIT can help the GMCH and other these testing centers, the COVID testing centers in Assam, to develop this the VTM kits. Okay, because that was the major what you call the problem they were facing. They could not even procure these kits, and at that time, Indian government was getting it from China. Okay, so as part of the Atmanirbhar Bharat initiative of the Prime Minister and the government of India, they wanted to have this development even in some parts of India. So this was an opportunity, and as soon as we received that message, I got a message from director, and immediately, you know, I talked to Professor Siddharth Ghosh, and you know, he told that surely we can think about this, and I immediately, you know, we also started discussing with RR Animal Healthcare. And you all won't believe everything took shape within, you know, I should say within 10 days. In fact, you know, we had the initial samples ready, and we went and gave those samples to the GMCH store. Okay, and they started validating it. So this is how quick things started moving. And you know, we had to develop overnight. You know, we started looking for some facilities in the campus, And I should thank the Center for Nanotechnology head, Professor Dipankar, who was very, very prompt, was very helpful. Immediately he told, you decide the space where you want, in the center or any other space. You identify the space and you come to me, and we can immediately start there. This is where the shift as a researcher and as a opportunity to serve the society. We had this, and we had to do this as an IIT. So we had to really shift with whatever resources we had. We started working on this, and within a week, in fact, I should say, we could make the initial samples. Okay, and of course, the inputs and the help that we received from RR Animal Healthcare, it cannot be mentioned in words here. Okay, so without their support and without their very active participation, in fact, nothing was possible. So what I would like to say is, we got this opportunity. We had to shift from our normal research to this COVID research or COVID work, and everything fell in place. And we had a nice team, students who were very enthusiastic to contribute, and everything came together. And we started supplying this, and it was a big relief for the state government. In fact, sometimes they would call us in the night, 10 p.m. and tell that we want the samples at around 12 a.m. within 2 hours we had to give. And it so happened that we used to take the samples from IIT campus, and by the time we even did not have the opportunity to unload these kits. Directly it will be loaded in another vehicle and taken to all parts of Assam. So this was how the we tried to meet the crisis situation. So it was a great learning opportunity for all of us. And this is still continuing. Dr. Datta and their team have really expanded the reach, and now probably we are supplying to more than 18 hospitals and around 7 different states. So it has become like almost pan India, we can say. Okay, so what started off as a small initiative in our campus, so it's really truly an Atmanirbhar Bharat initiative, and we really enjoy being part of this. And we have got necessary approvals for the VTM and the RNA extraction kits from ICMR. The RT-PCR kits also, we are looking for the approval from ICMR very, very soon. And Dr. Datta probably did not mention about this. Along with them, there are other kits which are coming up for development along with RR Animal Healthcare. This includes, of course, the COVID detection kits, the rapid detection kits, but also for a lot of other diseases also, disease biomarkers also. So this is a journey we have just initiated. We could contribute something very quickly. But yes, there are a lot of other people in this group also who are going to be part of this as we move along the journey. And without taking much of the time, I request all participants to, of course, interact with us and ask us questions, or if any suggestions are there, we'll be very happy to discuss with you all. So thank you very much.
So participants, if you have any questions, you can now ask. Participants, if you have any questions, you can raise your hands.
Yeah, I have one question. Can I ask?
Yeah, I was expecting that. I was expecting. Yes, please.
Don't expect answer, but
We follow the rules, or we should break the rules?
Yeah, please go ahead.
So we should follow the rules or break the rules?
You should follow the rules.
But if we follow the rules, then we will not be able to do something like what you have done, right?
No, you can do. Still, you can do.
Can I answer?
Yes.
This is a kind of part of disruptive approach. So you break the rule, but people should not see that or know that you are breaking the rule, and that is for the good thing only, not for bad thing. This much we can say. Otherwise, that nothing could have been possible if Anil or Raman were not present in the lab.
So what students should learn? They should break the rule or they should maintain the rule?
No, again, let us not put the word in our mouth. That rule was broken for a purpose, and we are all part of that, right? So that was beneficial for the entire, societal work that has been done. So in breaking the strict rule or some kind of, somebody's wisdom is not a bad thing, I would say. But the rules are there and that has to be followed, because otherwise Biman is also listening, he will be angry.
So what is the future plan? What is next now? COVID is gone then?
The future plan is in your hand, Dipankar. Actually, we will all follow our leaders and we will make more such kind of a POC device in the center, and we wish, not we, all the students in the center and the participant, they will take part in this whole journey, and everybody should be involved in this process. Then only the development is possible.
Yes, Param, what is future plan?
There are a lot of things in the pipeline. So we are, of course, trying to make more. As I told you, the rapid kits are trying to get them ready. But a lot of other disease biomarkers are also being planned. We are another MOU has already been finalized. So there's going to be a multiple biomarker detection. So we have a few things are there in the pipeline with RR. So let us see how things shape up in the future. And we are trying to get, this trying to get this lab in this what you call our research park, getting it up and running. Hopefully, that will be done probably as soon as probably. Let us see how quickly we can do that. I'm not putting a date or anything, but yeah, we want that thing to start functioning properly.
So as you know, we have to increase the number of startups. So how can we increase the number of startups? What are the places? What are the opportunities?
Yeah, that's a good question actually. We have to sit and discuss. I have not had the opportunity to talk to RR or this thing, but this build is like a startup. Okay, RR was there, but now SPILT that we are seeing, it will be like a startup only, new startup. So probably in addition to 9, you can put it like a 10th one. Okay, so yeah, we are looking into various aspects. A lot of things are being discussed with them, and internally also myself and Siddharth are planning something. So anyway, we'll keep you updated. Of course, you will know. You will be the first person to know something comes up.
No, the target is like, say suppose can we do 25 startups? And what are the different directions? So after the afternoon meeting, I have started thinking on that direction, that how exactly, these numbers can be in, and maybe one of the ways, like, if we diversify to energy and environment, that is another way of involving more startups. Your opinions?
No, in healthcare itself, I had already interacting with a few other companies. You are also aware actually. So let us see if they can also be invited. I'm still. It is through some ICMR institute. I have submitted a few more projects. So they also have some industry interactions. I'm trying to rope them in for this also, if they can come in. But we are in the initial stage of discussion. So probably in another week or 10 days, I'll get more clear information from these companies also.
Yes, SSG, your views on more number of startups. How can we do that?
More number of startup, actually, what Paramasa said, that is correct. Healthcare, we can put a few more, and then maybe some allied area. We can also think of, may not be energy, but some local entrepreneurship we can think of. We will talk about this thing with other members also, and then we'll find out. Because from healthcare to energy to many things will be difficult, because these are almost similar. Maybe we can put one, but if you have to expand the number, then we need to have some kind of a local entrepreneur in different sectors. Maybe that we can think of. Maybe agriculture, maybe that agriculture, or no, some kind of a plantation. I know the agarwood and other things. These are actually alternatives we can think of. Even Biman can chip in. He's also there, the local entrepreneur.
Yes, Debashis is also there. Biman maybe, yeah.
Yeah, ma'am. Thank you. Goes the Professor Iyer, and of course, our animal healthcare and Mr. Datta and all. I mean, I was listening to your talk and the discussion, of course. Of course, it's a good step to move forward with startups, spin-offs as we call it. But again, my views are like, not only the number is something which is more interesting. The more interesting part is the business part, in terms of value addition and revenue generation. So any forward movement should be keeping in mind blue sky research, as well as common domains which are easily accessible, and there is a lot of opportunity to generate revenue as well. So because as per the parlance of in startups, they call it like the high hanging fruit and the low hanging fruit. So I think the basket should have both of them, so as to balance in the times to come. And I think IIT Guwahati and Northeast has ample opportunities, as was pointed out. And I think from health, environment, local needs, which can be highly profitable and revenue generating for the country. So I mean, that is what I feel at the end of the day. But again, reiterating on the point that revenue generation at certain point of time should be the aim of the startups, rather than staying on paper. So that should be one focus area where we should be emphasizing more.
Movement has been done in the direction of commercialization, right? Now, question is that how that knowledge can be expanded to a larger group of people, and what are the different directions, opportunities that are lying ahead?
According to me, there are two different points that I would like to add here. One thing is nothing succeeds like success, that all of us know. So the initial success that we have got with IIT Guwahati in terms of this COVID diagnostics, I had been telling in my presentation, that success has encouraged us, and we have gone with licensing with DCGI for 21 other diagnostic products, and those 21 products we are already talking to IIT, and we are on the way of finalization a MOU with IIT for working further on those 21 products to make them better, make them more sensible, actually more sensitive and more accurate. So success stories will attract people. Now, from institute point of view, that is most important thing is probably other people have to be informed. But sir, one thing that I'd like to tell here, because we are a part of CCMB Hyderabad also. We are a startup and incubated in CCMB Hyderabad for 3 years now, more than 2.5, close to 3 years. We may sometime talk in person when I visit Guwahati. I'll take your appointment or maybe over phone. But the facilities that we get in CCMB Hyderabad and kind of agreement that IIT Guwahati has, like for many startups, it may not be very attractive. So there are differences are vast actually. What CCMB says and what IIT in their policy in relationship with the startups. So that is one thing probably IIT can look into how other institutes are attracting startups.
Devashish, can I intervene? Can you explicit little bit? It will help us to give the feedback about this. It is the rent or something else?
See this. There one thing is rent. Number one. Hello.
Yes.
One thing is rent, but rent can be a minor concern. One concern that major concern what I find is when we are in CCMB, whatever instruments they have, we can those instruments we can use, and even CCMB it is an AIC center actually. So they have people to operate the machines. They help us and everything. But if you see the IIT Guwahati agreement, it is written that when we use an instrument in the central instrumentation facility, we have to pay for that. So that is one thing. See, companies get attracted to an institute to get incubated as startup mainly for the facilities. The major thing is facilities. So again, payment for that can be one constraint when a startup may think. And number two, this is if you see the agreement that we have done with IIT Guwahati for this research part space. I request you to go through there are some terms like if you are here for 3 years, this for 5 years, this. You know, general concept is when you are loyal for a longer period, your rental should come down. But in IIT Guwahati, when you're loyal for longer period, your rental per month rental goes up. So that is the kind of understanding that I got from the agreement. So these are the 2-3 points. One is the amount. One is when you are there for a long time, you expect some discounts on the rentals. And number 3, probably this one. The instrumentation facility should be allowed, like incubated company startup should be able to use the instruments freely. Reagents they have to do, and also regarding manufacturing, you know, the pilot level manufacturing of the industries we are talking about, maybe healthcare or healthcare. I know a bit. Other industries I don't have much facility. So pilot level manufacturing facility, what I think IICT is doing now. IICT Hyderabad, we had a meeting and they said that we are going ahead with even CCMB is doing. I think CSR is doing now. So they are giving facility. They are making the facilities ready. So startup will certainly go with the facilities are there where they can use the instrument at, without any cost, only on the rental that covers the usage of the instruments and also the pricings. So this is what I would like to. These 2-3 points are coming to my mind immediately, sir.
If I can add, Divankar here.
Yeah.
Yes.
Yeah. So Dr. Datta, what you raised is a very valid point, because no, recently last 6-7 months, I'm also walking the same path where we have licensed 5 technologies and a couple of them are in the market now. And this has been a major concern with the industries I have tied up, and where they have taken space in research building, and these were the requests. So I think this is already there, and what is not there is the case-to-case basis handling, which is not a good way to move forward. Currently, it is a case-to-case basis where the negotiations are to be done, and the companies and the primary person, faculty who is handholding needs to do a lot of negotiations, which is, I think, at certain point of time the institute has to go away with, so as to be on the same page with all the startups, spin-offs, or the joint collaborative associations with the industry comes, as it comes, because with times to come, I'm pretty sure the number is going to go up. But still, our rules, as you rightly pointed out, are on a defensively placed rather than kind of welcoming strategy, because of certain reasons which I also know, that there is no certainty. For example, the equipment use, as you mentioned, which is a valid point, that any startup or spin-off company would like to use the equipments in the community where they are applying with. But the authorities probably feel that if we give no fee or open, they may be misused, or how they will be regulated. I think those are still there, which should not be. But if no, in a more planned way, we go ahead and reach to the authorities and explain to them why these kind of welcoming steps are required to encourage more startups. I think that will be the best way forward.
Yes, Biman, I agree. Actually, we have to give the right feedback based on their, inputs. This is very important point, actually. One way we are saying that we need to have more startup, but we have to make the system conducive for them also, right? So these points we'll be able to take care. I hope so.
Yeah, when I started, it was unfortunate to say, but again, when I started, not even the agreements were in place. So I started with something which was not an agreement, like, it was not to be called an agreement. We had to, being not a lawyer, I could sit over it and look into all the rules and regulations and the loopholes, sealing it from both the sides, the industry side and the institution side. Now I think this document is being used as a standard template, but again, that should have been done more professionally. So this is one example I'm giving.
Yes, correct.
So Devashish, perhaps you can write an email to us highlighting all these points. So that will help us in a huge way.
Sure, sir.
Because until or unless we raise these points, it will remain hidden, right? So
sure, sir.
It can only be sorted out with discussions and all. Okay.
Okay, sir. I'll put the mail, sir.
So maybe SSG, it's about 5:30. So maybe you conclude with concluding remarks.
You have already done that, but anyway, it is very nice interaction. I would rather say it is an informal interaction. As we discuss, again, I will bring back the tea stall story. Today we are missing that. The similar interaction we had here, and we have shared our experience from different parts, and some of the genuine problems have been discussed. And as Devashish agreed to send us his inputs, we will try to channelize this input to the authority and try to make the system more conducive for the new startup, and some of the startups from different angles can be also be seen with help of others, and definitely we'll strive hard to do better, and all students will be involved in the process. That is my wisdom and that is my hope. Thank you very much.
Thank you, sir. Thank you, Professor Parmeshwar Iyer, and thank you, Siddharth Ghosh, sir, for such an insightful and inspiring discussion. So now with the end of the panel discussion, we now conclude the second day of NWNTD 2020. So tomorrow our session will start from 9 a.m. Thank you.
Thank you, sir.
Thank you. Thank you very much.
Thank you very much.
Thank you, Dr. Datta.
Thank you, sir. Thank you so much.
Thank you.
Thank you, Deepankar. Thank you.
Thank you, IIT Guwahati.
Thank you. Thank you very much.
Thank you. Thank you all. Thank you.